Dear respected participants, welcome to our second International Biological and Health Sciences Congress. Today is the third day of the Congress, and today is especially we are focusing on human medicine. And first session is going to be moderated by Professor Dr. Alptekin Tosun. He is working at Giresun Medical Faculty, and his specialization is about the radiology. Dear Professor, thank you very much.
Thank you, Dr. Ulaş, for your nice invitation.
I am not sure my voice was heard.
Now it's okay.
Thank you very much again for your support. We can start with our session, and it is under your moderation. We are listening to you.
Dear Dr. Ulaş, thank you very much for your nice and perfect organization. Good morning. I'm starting this first session. Dr. Sevgi Gezici, his work is glabridin induces intrinsic apoptic signaling pathway and cell cycle arrest in A549 non-small cell lung cancer and PC3 prostate cancer cells.
Yes, good morning, everybody, and good morning, Professor Alptekin. I want to share my screen, but the host, I think, doesn't give permission. Could you please give me permission to share my screen?
Okay.
Please.
It is opened. It is open now.
Yes, okay. Dear guests and dear participants, thanks giving us opportunity to share our research. My research is related to glabridin. Glabridin induces intrinsic apoptotic signaling pathway and cell cycle arrest in non-small cell lung cancer and prostate cancer cells. I am Associate Professor Sevgi Gezici, where I am working at Gaziantep University. In this research, we used glabridin. It is an isolated compound from the herbal medicine. As we know, herbal medicine is the oldest forms of the medicinal and complementary treatment used through the history. And also, medicinal plants contain various bioactive compounds against most of the disease. So they have a big potential to produce plant-derived drugs for highly active new drugs. They are also some of the plant-based biochemicals are the good candidate for produced new drugs. And in this research, we used the roots of the Glycyrrhiza glabra. As we know, these species is the most popular medicinal plants, and its members are now commonly used as feed and food. This genus is native to Mediterranean area, but also it is found in other areas such as India, Russia, and China. These species also known as the licorice, and it has been used in pharmaceutical and food industries, also used in manufacture of functional foods and food supplements. In Chinese and Siddha systems, in traditional systems, it's known that these plant species used for gastrointestinal problems, cough, bronchitis, and arthritis, also used peptic ulcers, respiratory infections, And it is also known the hypolipidemic, cardiovascular, central nervous, respiratory, immunological, anti-inflammatory, analgesic, antipyretic, and many other pharmacological effects, because it has it possesses many
Dr. Gezici, can you hear us? Dr. Gezici, we can't hear you. Dr. Sevgi Gezici, we cannot hear you. Would you please open your microphone?
protective, anti-inflammatory, anti-ulcer, anti-diabetic, hypolipidemic, and many other pharmacological effects. Licorice Glycyrrhiza glabra is contain many compounds. Glabridin is the one of the most major flavonoid compound extracted from the Glycyrrhiza glabra roots, and also this plant contained approximately 0.52% glabridin, and this is the chemical structure of the glabridin, and it has a wide range of biological activities also. However, the anti-cancer effects of glabridin on various cancer cells have been identified. Mechanisms of glabridin in cancer proliferation, migration, and apoptosis through signaling pathways still remain partly unknown. Therefore, we aim to investigate the cancer prevention potentials, cancer prevention effects of glabridin on lung and prostate cancer cells, and also we aim to identify the underlying molecular mechanisms using the glabridin in this research. And in the material method section, we use Glycyrrhiza glabra, and we extracted firstly we extracted, and then we fractioned these plants using different solvent systems. Then we isolated the glabridin for further analyzed. We used glabridin, we extracted, and then we make it powder using the lyophilizer and vacuum systems. Then we isolated glabridin, and we use glabridin to test, to analyze the anti-cancer effects against A549 non-small cell lung cancer cell lines, and also PC3 prostate cancer cells. In this research, we use MTT colorimetric assay for evaluating the cell proliferation and cytotoxic activities of the glabridin, and also then apoptosis rate of the cells were identified using annexin V, FIT-C, and propidium iodor staining using through flow cytometric assay. And finally, we performed the Western blot analysis to verify the quantification of the proteins. Because of the limit of the time, I could not give the details about our material and methods, so I want to summarize our results. Glabridin exerted significant inhibition of cell metastasis via decreasing cancer cell migration and effective inhibition of cell proliferation in lung non-small cell lung cancer and prostate cancer cells. You can see the figures of related to cancer cell lines. Also, in addition, it was demonstrated that glabridin treatment suppresses cell viability and angiogenesis in the cancer cells. So we use annexin 5 method for determine the apoptosis level of the cancer cells using the cytoplasmic, using the differences between cancer cells and other cells using the differences. And we use also flow cytometry for determine apoptotic levels. The results of flow cytometric assay show that glabridin induces apoptosis dose dependently via increasing sub-Q1 FAS cell, phosphatidylserine externalization, and caspase activation. Then we evaluate our results using Western blot analysis, and we use the caspase proteins, and we evaluated the results that glabridin induces apoptotic pathways and causes the apoptosis. So, cancer
time is limiting. Would you please?
Against our tested cancer cell lines, and also we use the statistical analysis to evaluation. And as we see the conclusion and future perspectives, accordingly, it could be concluded that glabridin induces apoptosis in non-small cell lung cancer and prostate cancer cell lines through affecting cancer-related signaling pathways and caspase proteins. Therefore, glabridin could be a potential phytochemical for cancer therapy. On the other hand, glabridin has antiproliferative properties against both of the cell lines, supporting the potential use of licorice and glabridin as an adjuvant cancer treatment. For this context, licorice anti-cancer results in vitro should be confirmed in well-designed human studies and should then be evaluated in terms of dosage in cancer patients in order to be able to calculate the amount of licorice as food to achieve its anti-cancer effects without reaching the threshold of toxicity following licorice consumption. And also, I would like to thank Izmir High Technology Institute for their technical support to perform our analysis. And in our laboratory, we can perform most of the analysis results related in vivo and in vitro cancer activities, and also neurodegenerative activities we can conducted in our laboratory, and we are open to make scientific collaborations to other scientists and researchers. Actually, in our laboratory, I want to give a detailed scheme of anti-cancer phytochemicals synthesis, optimization, characterization, and prospective use as cancer therapeutic agents, which is
our time is finished. Would you please summarize?
Studies conducted in our laboratory. Scientific collaborations. And thank you for your kind attention. If you have any question, it will be very appreciated to me for answer your question. Thank you again. Have a nice and fruitful meeting.
Thank you, Dr. Kesici. Any questions? Let's have a look at the chat. There is no questions. Okay, I'm inviting the next speaker, Dr. Adem Keskin. Effect of age, gender, cancer, and burden on homocysteine levels.
Günaydın saygıdeğer hocalarım. Çalışmamı müsaadeniz olursa Türkçe sunmak istiyorum.
Buyurun hocam.
Ekran görüntüsü geliyor değil mi hocam?
Şu anda başladı.
İsmim Adem Keskin, Aydın Adnan Menderes Üniversitesi.
Şimdi biz
Ses kesildi Adem Bey.
Şu an geliyor mu sesim?
Şu anda geliyor.
Ee,
Buyurun.
Adem Keskin, Aydın Adnan Menderes Üniversitesi Sağlık Bilimleri Enstitüsü Tıbbi Biyokimya'da doktoramı yaptım. Bu çalışmayı Samsun Eğitim Araştırma Hastanesi'nde çalışan Recai Acı biyolog arkadaş ile beraber yaptık.
Doktor Adem, İngilizce sunum yapabilmeniz mümkün mü acaba?
İngilizce sunum için müsait değilim hocam.
Tamam.
Çalışmamız pardon hocam. Çalışmamız homosistein düzeylerinin üzerine etki eden yaş, cinsiyet, kanser ve yatalak hasta durumlarının etkisini inceledik. Çalışmamızda amacımız kardiyovasküler risk markör olarak gözüken homosistein düzeylerini etki eden yaş, cinsiyet, kanser ve sekonder yaşamın etkisini incelemek amacıyla yaptık. Çalışmamızda 266 katılımcı vardı. Bunların yaş aralığı 3 ile 93 yaş aralığındaydı. Çalışmamızdaki 266 kişinin 109'u erkek, 157'si bayandı. Aynı zamanda çalışmamıza katılan 266 kişinin 120'si yatalak hastaydı. Diğer 146 birey sağlıklı bireylerdi. Bu 120 hastanın yatalak olan 120 hastanın 78'i kanser tanısı almışken diğerlerinde herhangi bir kanser tanısı bulunmamaktaydı. Çalışmamızda homosistein düzeylerinin immunochemistry model yöntem kullanılarak COBAS 8000 analizör kullanılmıştır. Elektrokemistri teknoloji altında. Çalışmamıza bakacak olursak homosistein düzeyleriyle yaş arasında bir pozitif korelasyon gözlendi. Homosistein düzeyleri yaş arttıkça yükselmekte olduğu saptandı. Çalışmamızda erkek bireylerin homosistein düzey ortalaması 13.97 bulunurken kadın bireylerin homosistein düzey ortalaması 15.17 olarak saptandı. Aralarında herhangi bir fark olmadığı gözlendi bu sonuçlarla. Çalışmamıza katılan hastaların kanser tanısı alanların homosistein düzey ortalaması 21.02 gözlenirken kanser tanısı olmayan bireylerin homosistein düzey ortalaması 12.98 olarak saptandı. Aralarında kanser tanısı olanların kanser tanısı olmayanlara göre daha yüksek olduğu saptandı. Çalışmamızda yatalak hastaların homosistein düzey ortalaması 22.19 olarak saptanırken herhangi bir yatalak hasta olmayan bireylerin homosistein düzey ortalaması 11.0 olarak saptandı. Yatalak olan hastaların yatalak olmayan bireylere göre daha yüksek olduğu saptandı. Sonuç olarak homosistein düzeyleri üzerinde yaş, kanser ve yatalak olma durumunun bir etkisi olduğu saptandı. Cinsiyetin ise herhangi bir etkisinin olmadığı saptandı. Çalışmamı dinlediğiniz için teşekkür ederiz.
Okay, thank you, Dr. Keskin. Are there any questions in the chat salon? No. Dear participants, if you have any questions, would you please write on chat salon, and we can see the questions and ask the presenters. Okay, for the next term, Dr. Efsun Somay, the evaluation of many parameters, concentrations in synovial fluid and in patients with temporomandibular joint derangements and effects on success of arthrocentesis. Okay, Dr. Somay, it's your turn.
If the Dr. Somay is available here, maybe she can press on the button of the reactions and we can open her voice, because in the name of Efsun Somay, nobody is available in the salon. If she is here with another name, can you?
Ulaş hocam, also there is a request in the WhatsApp group from Pakistan. I think his battery is gonna die and he wants to present right now. If you would like to see the WhatsApp group.
Is it possible, Professor Dr. Tekin Tosun?
Yes, it's so possible. Okay.
Okay.
Now, who is which term?
Which I will check the WhatsApp group and I will tell you.
Shahid Shah, I think.
Shahid Shah. Ovarian carcinomatosis, microenvironments induce epithelial mesenchymal transition and uptake regulate protease procoagulation activity in mesothelial cells.
Okay.
Okay, Dr. Shahid, it's your time. Welcome.
It's okay. Okay, I'm Dr. Shahid from Government College, University, Faisalabad, Pakistan. First of all, I would like to thank to the organizer who provided me the opportunity to present my work on this platform. So today I'm going to present the research topic, that is ovarian carcinomatosis microenvironments that induce epithelial mesenchymal transitions and upregulate protease procoagulant activity in mesothelial cells. Okay, so in peritoneal carcinomatosis, usually cancer nodules are present on the peritoneum, and the cancer cell clusters are present in the peritoneal fluids. So epithelial mesenchymal transitions is a transformation of epithelial cells into the mesenchymal cells by downregulating the epithelial markers and upregulating the mesenchymal markers. This EMT is very important in mesothelial cells that plays an important role for the cancer cells to attach on the peritoneal cavity. So here you can see the endothelial protein C receptors, that is anticoagulant factors, which downregulate the thrombin generations by blood coagulation pathway via binding with protein C that are very helpful. And the next is the tissue factor, that is an initiator of coagulation via extrinsic pathway. Both these factors are very important in peritoneal carcinomatosis expansions. So these are our results. First, we try to characterize the peritoneal membranes via immunohistochemistry and scanning electron microscopy. So in the non-invasive region of the peritoneal cavity, you can see here the mesothelial layer is intact due to presence of ecadrine, but in case of invasive regions, this layer is broken. And similar results were found by scanning electron microscopy. Here you can see the mesothelial cell layers is intact, but here the phenotypes of mesothelial cell layers has been changed. So here is in situ anatomopathological study of peritoneal wall that was stained with hematoxylin and eosin. You can see the non-invasive regions that is in good conditions, while in the invasive regions, the fibrin deposits begins to form. Here, the cancer cells begins to attach, and here cancer nodules begins to form. So fibrin deposits as well as the cancer nodules are very prominent in the invasive regions of the peritoneal membranes. Here you can see the in situ scanning electron microscopy of the peritoneal wall. In the non-invasive regions, the mesothelial cells are intact, but in the invasive regions, the phenotype has been changed that we called deformed mesothelial cells. And these deformed mesothelial cells has been exfoliated from the peritoneal surface. Here you can see the intermesothelial space, and after this, the fibrin deposits begins to form. And in the D section, you can see the cancer cells trying to penetrate on the fibrin deposits that is located on the peritoneal surface. So here it's a comparison of non-invasive and invasive regions of the peritoneal membrane via immunohistochemistry, and we found the number of blood cells and lymphatic vessels in invasive regions. Here you can see invasive region is significantly higher as compared to the non-invasive regions. So next is when mesothelial cells are treated with the ovarian or the ascitic fluids, we found a change in phenotypes. So ascitic fluid induce a change in phenotype of the mesothelial cells, as well as it induce the cell proliferations as well as the cell migrations. Next, you can see here the histogram that shows comparative release of cytokines from the mesothelial cells in their supernatant after treating with ovarian ascites. And when these mesothelial cells are treated with exogenous cytokines, we found an upregulation of metalloproteins like MMP2 and MMP9s, nepriolizins, and tissue factors, and fibrinolactins. That means that plays an important role in peritoneal carcinomatosis. Similarly, when these mesothelial cells were treated with the ascites, we found a significant decrease in the epithelial markers and increase in mesenchymal markers. That shows that epithelial mesenchymal transitions are affected by the ascitic fluids that are present in ovarian carcinomatosis. So here you can see similar that after treating the mesothelial cells with the ascitic fluids, there is a significant increase in procoagulant factors like tissue factors, like MMPs, MMP2, 9, nepriolizins, and a significant decrease in endothelial protein C receptors as compared to control. And similar results were found when nepriolizin activity was checked in modified mesothelial cells. And here, the diagram D and E shows the protein expressions in the ascitic fluid that shows the presence of mesothelial cells in the ascitic fluid of the peritoneal carcinomatosis patients. Okay, this is simple diagram that when the mesothelial cells are treated with peritoneal fluids, they undergo epithelial mesenchymal transitions. As the result of this, they change their phenotypes that we call the modified mesothelial cells. And the nepriolizin and MMP2 and 9 plays an important role to detach these mesothelial cells from the peritoneal surface. So here you can see the cancer cell clusters that we found in the ascitic fluids. You can see here the cancer cell clusters that are surrounded by the fibrin fibers. This is simple microscopy by methylene blue. Here the phase contrast media and even the transmission electron microscopy that shows the fibrin present an important role in cancer cell cluster integrities. Here you can see the in vitro study of cancer cell interaction with the fibrin clot. These are the fibrin clot. Here the cancer cells try to penetrate the fibrin clot. That is evidence that peritoneal carcinomatosis may progress. Here is simple cinematography photographs the cancer cells that becomes attached to the fibrin deposits and then divide to form cancer cell clusters that are found in the ascitic fluid. Here is just a diagram that shows graphical presentation that detached fibrin filaments against time. So this is our main working model that how cancer cell clusters and nodules formed. You can see here the cancer cells first attached to the fibrin deposits and then they start to divide. When they start to divide, they multiply themselves, and perhaps the fibrinolytic enzymes like tissue plasminogen activator and urokinase plays an important role for the detachment of these cancer cell nodules that appear in the ascitic fluids. So our results conclude that the change in mesothelial cells by tumor microenvironment induce secretions of neprilyzins and proteolytic enzymes, which are responsible for their detachment from the peritoneal surface. Number two, the modification of peritoneal procoagulant activity via mesothelial tissue factors on the peritoneal membranes for fibrin networks and generations. And the last one that we conclude our experiments that the formation of cancer cell clusters or nodules on the fibrin deposits, it may lead to carcinomatosis expansions. Okay, thank you. So if you have any questions, I am available to answer your questions.
Any questions thank you for Dr. Shsy, your excellent presentation. Thank you.
Thank you.
Any questions? I think no. Thank you very much, Dr. Shsy. Okay, now going to the next session. Dr. Efsun Somay will present us the evaluation of parameters concentration in synovial fluid in patients with temporomandibular joint derangements and effects on success on arthrocentesis.
I think she's not available.
Okay. Next speak Canan Eroğlu Güneş. Are you here?
Yes.
Okay. Dr. Canan Eroğlu Güneş will present us his work, her work, a cardiac glycosid of Nerium oleander L-dome regulates parameter scans in human endometrial carcinoma cells. Okay.
Can you see my screen?
Yes, okay.
Okay. Hello, dear researcher. I'm Sedef Akçalan. I'm studying at Necmettin Erbakan University. My presentation is a cardiac glucoside of Nerium oleander downregulates TLR1, TLR6, and MID88 genes in human endometrial carcinoma cells. My presentation's content: introduction, cancer, tool-like receptors, a cardiac glycoside oleandrin, aim, materials, method, result, and conclusion. Introduction: cancer is one of the major diseases that cause human death. Cancer is a group of diseases that undergo unregulated cell growth and proliferation without stopping. And worldwide, an estimated 19.3 million new cancer cases and almost 10 million cancer deaths occurred in 2020. Endometrial cancers. Endometrium cancers rank as the sixth most common neoplasm in women worldwide. It occurs in women predominantly after menopause, and hormones play an important role in the etiology of human endometrial cancer. Endometrial cancers, unopposed estrogens are a key contributor to each of the established risk factors for the disease: early menarche, late menopause, nulliparity, menopausal hormone use, obesity, and the other risk factors include diabetes, hypertension, and family history of endometrial cancer. And what is tool-like receptors? Briefly, TLRs. TLRs are a well-known family of pattern recognition receptors that play an important role in host immune system. TLRs are potent activators of inflammatory response, and the immune system can control tumor progression and growth. Tumor progression involves complex interactions between tumor cells, immune cells, and the tumor microenvironment. And chronic inflammation and infection can contribute to the development of tumors. TLRs may also cause the development of cancer and inflammatory diseases. And TLRs play anti-tumor or tumor promoter roles in different cancer cells. TLRs are expressed not only on the surface of immune cells, but also on cancerous cells. In humans, TLRs 1 to 10 play very important role in diseases progression, and the TLR signaling have been well studied in various diseases, including cancer. And olandrin. Nerium olander is one of the most poisonous of commonly grown garden plants. And olander, which is a member of Apocynaceae family, has been historically used in the treatment of leprosy, hemorrhoids, and snake bite. Olandrin is one of the cardiac glycosides obtained from an olander. And plant-derived natural products such as phenolics, alkaloids, and terpenoids have recently gained attention due to the growing request for discovering novel anti-cancer drug candidates. This study aimed to investigate the effects of olandrin on tool-like receptors pathway genes in Ishikawa human endometrial carcinoma cells. Material methods, cell culture and toxicity assay. Cell culture, Ishikawa cells were proliferated and in RPMI medium and incubate cells at 5% carbon dioxide atmosphere and 37 degrees centigrade. And toxicity assay, cells were treated with 8 different doses between 7.5 and 300 nanomolar doses of olandrin, and the toxic activity of olandrin was detected in Ishikawa cells by using XTT colorimetric based assay. And the calculated IC50 dose. IC50 dose was used in cells after 2 days of olandrin treatment. Total RNA isolation from cells and cDNA synthesis. And real-time quantitative PCR analysis, expression profile of the TLRs 1 to 10 and MID88 genes were determined by a real-time quantitative PCR. Briefly, each reaction contained cDNA, forward reverse primer, and cybergreen supermix. And the result, according to XTT proliferation test results, IC50 dose was determined as 75.3 nanomolar at 2 days in Ishikawa cells. And these graphics show that the expression change of TLR genes. According to these results, olandrin treatment significantly decreased the expression of TLR1, TLR6, and MID88 genes, and the other TLR's expression did not differ significantly. And conclusion, olandrin was found to be cytotoxic and significantly reduced the viability of in Ishikawa cancer cells. In conclusion, olandrin affected on TLR signaling pathway by changing expression levels of TLR1, 6, and MID88 genes in Ishikawa cells. However, further molecular biological analysis are required to explain these mechanisms. Thank you for listening to me.
Dr. Akçaalan, thank you for your nice presentation. Any questions? No, in the chat room. Okay, for the next presentation, I would like to invite Dr. Melek Bilgin. She will present her work, Melatonin Levels in Women with Systematic Lupus Erythematosus. We can see your screen, Dr. Melek Bilgin.
Günaydınlar. Hocam çok özür diliyorum. Ben de sunumu Türkçe yapacağım diye öyle hazırlanmıştım ama hani
kongre dilimiz İngilizce hocam. Mümkünse İngilizce
yapabilir
olur.
miyim? Sadece o zaman sonuç bulgular kısmını Türkçeydi benim. Diğerlerini İngilizce olarak hazırlamıştım ama hani
Tamam hocam. Buyurun. Sizin sıra.
bu şekilde olacağını bilsem onları da öyle yapardım. Kusura bakmayın. Efendim?
Buyurun hocam.
Yapabiliyor muyuz?
Tabii.
Efendim?
Olur hocam.
Yapabiliyor
Buyurun. Sunabilirsiniz.
Tamam. Kusura bakmayın tekrar. Özür diliyorum bundan dolayı. Samsun Eğitim Araştırma Hastanesi'nde mikrobiyoloji uzmanı olarak çalışmaktayım. Doktor Melek Bilgin ben. Çalışmamız sistemik lupus eritematosuslu hastalarda melatonin seviyelerinin incelendiği bir araştırma. Niye büyütemiyorum? Bildiğiniz gibi sistemik lupus eritematosus herhangi bir organ sistemini etkileyebilen, nedeni tam olarak ortaya konamamış, kronik otoimmün ve inflamatuar bir hastalıktır. Sıklıkla deri, hematolojik, kardiyovasküler, nörolojik, eklem ve renal bulgular ile ortaya çıkabilen,
Hocam,
özellikle de bu sistemle...
Özür dileyerek bölüyorum. Sorry for interrupting you. Üst kısımda tekrardan ekran paylaş derseniz slaytınız ilerleyecektir ya da sol taraftan slaytın üzerine çift tıklamanız lazım. Yani şu anda slaytlarınızı da göremiyoruz ilerlemesini. Sadece ana ekranı görebiliyoruz.
Ha şu an bende gözüküyor gibi ama çok... Yeni paylaşım mı diyeyim hocam? Şöyle.
Artık hocam özür diliyorum. Dün de benzer teknik bir problem olduğu için.
Oldu mu hocam?
Şu anda ilerlemiyor slaytlarınız hocam.
İlerlemiyor.
Hocam aşağı kısma gelip slide gösterisinin üzerine öncelikle bir tıklayın. Daha sonrasında
Şöyle oluyor hocam.
sizin ekranınızda tam gözüküyor ama bizde gözükmüyor. Sonra
bende gözüküyor da o yüzden diyorum.
Sonra üst kısma gidin hocam. Üst kısma gidin. Üst kısımda yeşil tekrar paylaş kısmı var.
Yeni paylaşım var diyor.
Evet. Ondan sonra orada slide'ı seçin hocam. 2 tane PowerPoint var. Slide kısmını seçin.
Şunu. Tamam.
Tekrar paylaş
Seçtim.
yeri açılmış.
Oldu mu şimdi?
Açılmadı. Oldu hocam.
Oldu mu hocam?
Şu anda oldu. Buyurun.
Heh.
You can continue.
Okay. Tekrar başlayayım o zaman. Sistemik lupus eritematosus çoğunlukla kadınları etkileyen kronik otoimmün bir hastalık. Patogenizi tam olarak aydınlatılamamış olmakla birlikte genetik duyarlılık, çevresel faktörler ve hormonların etkileşimi immün toleransın erken dönemde bozulmasına yol açmakta ve sonrasında antijen sunumu, T ve B lenfosit aktivasyonu ve plazma hücrelerinden otoantikor üretimi gerçekleşmekte. Sonuçta immün kompleks ve anormal T lenfosit ve diğer immün hücrelerin aktivasyonu aracılığıyla organ hasarı meydana gelmekte. Otoimmün bozukluğun bir sebebinin de sempatik sinir sistemi ve çeşitli hormonların dahil olduğu stres yanıt mekanizmalarının bozulmasından kaynaklandığı düşünülmekte. Bu hormonlardan biri de bildiğimiz gibi melatonin hormonu. Bu bağlamda biz çalışmamızı neye amaçladık? Biz SL'li kadın hastalarda melatonin düzeylerinin seviyelerine baktık ve bu melatonin düzeylerinin hastalık aktivite şiddetiyle ve laboratuvar bulgularıyla bir korelasyonu var mı onu inceledik. Samsun Eğitim Araştırma Hastanesi'nin etik kurulundan izin alarak gerçekleştirdik bu çalışmayı ve hastanemizde mikrobiyoloji laboratuvarında yaptık. 28 SL kadın hasta ve 20 tane de sağlıklı romatolojik bir şikayeti olmayan hasta kadını kontrol grubu olarak çalışmamıza dahil ettik ve günlük serum melatonin seviyelerini inceledik. SLE tanısı bu hastalar tabii ki hastanemiz romatoloji bölümünce takip edilen, SLE tanısıyla takip edilen hastalardan oluşmaktaydı. SLE tanısı da 1997'de Amerikan Romatoloji Koleji tarafından tanı kriterlerine uygun olarak 2 ve daha üzeri bulgular var ise SLE tanısı konmakta. Buna göre konulmuş hastaları dahil ettik çalışmamıza. Ve romatoloji uzmanımız tarafından da melatonin için kan aldığımız gün gelen hastaların hastalık şiddeti skoru yapıldı. Eğer bu skor 0 ise hastalık şiddeti yoktur. No aktivite olarak sınıflandırıldı. 1 ila 5 arasında 1 puan almış ise hafif aktivite, mild aktivite olarak sınıflandırıldı. 6 ila 10 arasında ise 1 puan moderate yani orta derecede aktivite şiddeti var denildi. 11 ila 19 arasındaysa da yüksek aktiviteli yani hastalık şiddeti yüksek olan grup oluşturmaktaydı. 20'nin üzerinde ise yüksek aktivite, çok şiddetli aktivite vardır denilmekte. Ancak bizim 28 hastamızı ancak 4 gruba ayırabildik. Çok şiddetli aktivite gösteren hasta grubumuz yoktu bizim çalışmamızda. Kan örnekleri saat 8 ila 10 sabah alındı. Tüm hem kontrol grubundan hem de hastalık grubundan. Melatonin konsantrasyonları ELISA kiti ile değerlendirildi. Antinükleer antikor tanıda çok önemli bir kriterdir SL'de. Biz de indirekt floresan antikor yöntemiyle antinükleer antikor testlerini değerlendirdik. Anti-DS-DNA da yine hastalık şiddetinin takibinde kullanılan bir parametredir SL hastalarında. Bu da mikroELISA kiti ile değerlendirildi ve kit prospektüsüne göre de 30 internasyonel ünite bölü mililitre üzerinde olanlar pozitif, 20-30 aralığında olanlar ara değer, 20'nin altında ise de negatif olarak değerlendirildi. Çalışmamızda 28 kadın SL grubu olarak 20 tane de kontrol grubu olarak dahil ettik demiştik. Kontrol grubunun yaş ortalaması 40, iki SL grubunda 44 olarak tespit ettik ve istatistiksel olarak anlamlı bir fark yoktu. Her iki grubun melatonin düzeyleri bağımsız örneklem testiyle karşılaştırıldı. Kontrol grubunun melatonin düzeyi 42,70 pikogram bölü mililitre iken SL hasta grubunda ortalama melatonin düzeyi 16,56 pikogram bölü mililitre olarak tespit edildi ve bu da istatistiksel olarak anlamlı sonuç olarak değerlendirildi. Burada da şematik gösterimini görüyoruz. Demiştik ki hastalık şiddeti hastalık şiddet aktivasyonu da skorlanmıştı demiştik. Bu gruplandırmaya göre 8 hastamızda aktivite yok olarak belirlendi. 15'inde hafif şiddetli, 3'ünde orta şiddetli, ikisi de yüksek şiddetli olarak gruplandırılmıştı. Melatonin seviyelerine baktığımızda aktivite olmayan grupta 15,79 nanogram bölü litreydi. 15'i 17,26, 3'ü 17,87, iki tanesinde de 12,50 nanogram bölü litre. Bu pikogram bölü mililitreye eşdeğerdi tabii aynı zamanda. Karşılaştırma sonucuna göre gruplar arasında anlamlı bir fark saptanmadı. Yani melatonin seviyesi SL hastalarında anlamlı derecede düşük. Ancak ve şiddeti yüksek olan iki hastamızda da melatonin seviyesi en düşük olarak bulundu. Ancak gruplar arasındaki hasta sayımız yeterli olmadığı için istatistiksel analize gruplar arasında anlamlı bir fark saptamadığımızı gördük. Bu da şematik olarak gösterimi. Yine SL hastalarımızın 2 tanesinde anti-DS-DNA testini negatif olarak saptamıştık. Bunlar zaten aktivite olmayan gruptaki hastalardı. Anti-DS-DNA ortalaması 120 ünite bölü mililitre olarak tespit edildi. Peki melatonin seviyeleri ile anti-DS-DNA arasında bir korelasyon var mı diye baktığımızda istatistiksel olarak bir ilişki saptayamadık. Melatoninin otoimmün hastalıklarda yapılan, araştırılan çok çalışması var. İşte sistemik lupus eritematosus'ta, sistemik skleroziste, romatoid artritte, otoimmün tiroid hastalıklarında ve multiple skleroz gibi. Ancak SL hastalarında melatonin araştırıldığı çalışma sayısı kısıtlı. Ve bu çalışmaların sonucunda şöyle bir şey söylüyorlar. Melatonin çok önemli bir role sahiptir. İmmünomodülatör ve antiinflamatuar etki olarak ve bu hem doğal immüniteyi hem de adaptif immün yanıtı etkilemektedir deniliyor. Mesela ilk çalışma melatonin seviyelerinin sağlıklı kadınlarda ve SL hastalarda yapıldığı ilk çalışma Robe ve arkadaşları tarafından yapılmış. Bundan 111 SL hastası, 46'da sağlıklı kontrol grubu olarak ele alınmış. Bunda da aynı bizim sonuçlar gibi melatonin seviyeleri SL hastalarında anlamlı derecede düşük. Aynı zamanda hastalık şiddetiyle negatif korelasyon, ters ilişki saptanmış melatonin düzeyleriyle hastalık şiddeti arasında. Yine başka bir çalışma var. Bu da Wang ve arkadaşlarının yaptığı bir çalışma. Burada 90 SL hastası, 90'da kontrol grubu değerlendirilmiş. Ancak bu çalışmada farklı olarak tamamen Robeva'nın sonuçlarından farklı olarak herhangi bir korelasyon bulunamamış melatonin seviyeleri açısından SL ve kontrol grubunda. Aynı zamanda deniliyor ki laboratuvar parametreleriyle de hastalık şiddetiyle de melatonin arasında bir ilişki yok denilmiş. Farklı ırklarda, farklı hastalık grubunda farklı da seyredebiliyor gibi bir yorum yapılmış. Peki biz çalışmayı bulduk. Biz çalışmamızda SL hastalarında melatonin düzeyinin anlamlı derecede daha düşük olduğunu bulduk. Ancak hastalık şiddetiyle korelasyonunu saptayamadık. Bunu da hastalığı çalışmamızın kısıtlı bir yanı olarak belirtiyoruz. Çünkü sayımız az. Ondan kaynaklandığını düşünüyoruz. Bu yüzden bu konunun daha iyi anlaşılabilmesi için melatoninin SL'li hastalardaki rolünün daha iyi irdelenebilmesi için daha fazla sayıda hastaların kapsamlı daha geniş çalışmalara ihtiyaç olduğunu düşünüyoruz. Bir öncü çalışma olabileceğini düşünüyoruz bu konuda. Teşekkür ediyorum.
Okay, thank you, Dr. Bilgin. Are there any questions? No. Okay, next presentation. Mehmet Emin Çelikkaya. Is he here? Mehmet Emin Çelikkaya? No. Okay, next presentation. Dr. Burcu Çaykara. Okay, welcome, Dr. Burcu Çaykara. She will present her work, altered microRNA expression profiles in the postmortem prefrontal cortex of the alcohol dependence.
Just give me a moment. I will try to give a presentation. I think you will see my screen as a presentation section. I hope you see, all of you see this. Isn't right?
Yes, okay.
Okay. We would like to thank you and respect to the scientific and the organizing committee of the Congress, and dear Dr. Tosun and dear Dr. Acargöz and dear participants. I will present today altered microRNA expression profile in the postmortem prefrontal cortex of the alcohol dependence. Alcohol is probably first discovered pharmacological agent, and the alcohol addiction goes back to the ancient times in the human history. Alcoholism is a complex disease affected by genetics and responsible for the deaths of the 2.5 million people annually at the global level. Alcohol and addictive substances change the molecular mechanisms in the human brain, affecting the expression of the certain genes in the brain. In addiction, gen expression interactions that occur in the different parts of the brain at a certain time intervals should be known. MicroRNAs that play important role in the control of the gene's expression are the short RNA molecules and are encoded from the DNA, but it's not translated into the protein. You can see the right section. This figure shows the circle shows the nucleus, and the other parts of the shows the cytoplasma. DNA, as you can see, microRNA is encoded from the DNA, and the precursor microRNA translate to the cytoplasma, and in there it's included to the RNA-induced silencing complex, and they show the effects as translation, degradation, or deadenylation, and the effects to gen expression. By binding to the target gens, messenger RNA, they can cause the mRNA degradation and the translation inhibition. Many microRNAs were found responsible for the development of the morphogenesis in the dendritic and addiction in the research studies. We aim to analyze two expression of the MIR, is the short name of the microRNA, 132, 133, 3B, 130, 180, 1A, 119, and the 212 in the postmortem prefrontal cortex tissues of the people who died with alcohol abuse. These people were the very alcohol abusers, not the social drinkers. Material and methods. The prefrontal cortex tissues were collected within 20 hours of the date and frozen in the liquid nitrogen and stored at minus 18 centigrade. 30 prefrontal cortex from alcohol users and 30 prefrontal cortex non-alcohol users were grouped as alcoholic and the non-alcoholic group. Total RNA isolation, reverse transcription, and quantitative real-time polymerase chain reaction were performed. Data CT methods were used for the determining expression levels, and or expression levels were not normally distributed. That's why Kolmogorov Smirnov manual tests were used. When we see the alcoholic and non-alcoholic group, almost nearly similar. I mean, it's not the statistical difference. While the mean age of the starting to consuming alcohol was the 27 years old, the duration of the alcohol was the almost 25 years. We found increased levels of the mere 133, 140, 119, and 1, 2, and 12 in alcohol abuse. On the other hand, one microRNA, 132 and 119 were downregulated in the alcohol-dependent prefrontal cortex tissues. No correlation was found between age, duration of alcohol consumption, and the expression of the microRNAs. We couldn't reach any other demographic values of the people who died with the alcohol consuming. That's why we couldn't have any chance to add to our results for the correlation. This shows the, in general, table of the gene expression differentiation between the groups. Under the one, it's like the decrease level, and when you see like the 133B, 4 and 44, it's like the above the one, and it's the increased levels. And this chart also shows the differentiation between the microRNA in the alcohol and non-alcoholic groups. Conclusion: consequently, microRNA may involve in the alcohol and substance abuse. To our results, many of these studied microRNAs may be associated in the neuronal pathways of the alcohol consumption. Thus, these microRNAs and the associated mRNAs should be studied at the pathways of the alcohol use and should be clarified. And these are all my end of the presentation to do, keeping short to presentation duration. And thank you for listening. If you have any questions, please let me know.
Okay, any questions? May I ask you a question, please?
Okay.
How do you classify the alcohol usage and non-alcoholic people, or how many liters per day, or the consumption, and also the final, for the dead people, the final diagnosis were alcoholic intoxication or heart failure or chronic alcoholic failure?
Yeah, you know, as there's many samples as collected when if one people died with a suspicious death, you know, and the urine, one of them, especially they were looking for the like the substances degradation molecules, and the alcohol, one of them. They are looking for the alcohol in the blood, these people in the blood system, and also urine. These people like to not 1 cup of alcohol in a week. It's they had family stories, and their family is approved. Yes, they were like the drinkers, were drinkers, like a bottle in the week, and like that. That's why we correlate these results with the family story. That's why we choose these people, alcoholic people. And the others one, is healthy people, and there's no drinking stories from the approved by their family, and they especially were defined as the Muslim people. That's why the non-alcoholic groups is what completed the non-alcoholic groups is the very easy to do alcoholic groups, of course. But we also through like the 2 times one of them is like blood and the urine test. The other one is the family story.
Okay, thank you, Dr. Shaykara. Any questions?
You're welcome.
Okay, thank you. For the next presentation, I would like to invite Yakup Önal, Dr. Yakup Önal, the relationship between cultural sensitivity and sexualities among university students. Okay, Dr. Önal.
Welcome to my presentation. My name is Yakup Önal. I am first-year nursing student at the Faculty of Health Sciences at Barton University. The subject of my presentation is the relationship between cultural sensitivity and sexual mates at the university. We aim to examine the relationship between cultural sensitivity and sexual mates among university students in this study. Method. This cross-sectional study was conducted with 1081 university students in 2021 and 2022 academic years in Barton, Turkey. Data collection tools were personal characteristic for sexual mate scale and intracultural sensitivity scale. To analyze the sexual mates by participants' characteristic, we utilized t-test and ANOVA and the Banfuruni test during post-hoc compression. To identify the relationship among the variables, we utilized the person's correlation coefficient. Finally, to verify the mitigating effects of participants' cultural sensitivity in the studied relationship, we utilized a simple regression analysis and multiple regression analysis. Result. The average age of the students is 20.17 years. 58.4 female and 41.6 male, 4.2 were in the preparatory class, 34.2 in the first grade, 34.1 in the first grade, 18.9% in the first grade, and 8.5% in the first grade. Male students believed in higher levels of sexual mates. Students living in the metropolitan cities for a long time believed in sexual mates at a lower level than students living in villages, towns, districts, and cities. The students who did not think that being together with different cultures contributed positively to you believed in sexual mates at a higher level. The students who found your level of knowledge about sexuality and sexual health sufficient believed in the sexual mates at a higher level. The total mean score of the cultural sensitivity scale was a significant predictor of students' sexual mates. 11% of the total variance regarding sexual mates refers to students' cultural sensitivity. A 1-point increase in the intracultural sensitivity scale causes a minus 382 decrease in the sexual mate scale. Conclusion: it is recommended to plan and open the lesson on sexual health and cultural sensitivity in the curriculum in university. Thank you for listening.
Dr. Ünal, thank you for your presentation. Are there any questions to Dr. Ünal? No. I'd like to invite a new speaker, Dr. Turhan Turan. He will present his work, their work. First, molecular detection of two giroviruses from domestic cats in Turkey, chicken anemia virus and girovirus galga 1. We are listening to you. Okay, Dr. Turan, welcome.
Teşekkür ederim Sayın Hocam. Evet. Benim
Turan Hocam, sunumunuzu tam ekrana alabilir misiniz?
Tam ekran görünüyor.
Artık hocam teknik problem olduğu için müsaadenizi açıklamak istiyorum. Turan Hocam, slaytınızın üst kısmına gidin. Yeşil bir bölge var orada. Yeşil butona tıklayın. Tekrardan paylaşım yapı seçin. Orada 2 tane sunumunuz gözükecek. Slide olanı seçin hocam.
Bir paylaşım.
Slide olarak açılmış olanı seçin hocam.
Şu an?
Yok, olmadı hocam. Ha, oldu. Oldu hocam.
Şu an oldu değil mi hocam?
Buyurun hocam.
Tamam. Tamam, teşekkür ediyorum. Evet, bugün benim size yapacağım sunum başlığı Türkiye'de evcil güvercinlerden chicken anemi virüs ve girovirus galga 1'in ilk moleküler tespiti. Buradaki girovirus galga 1'deki galga, gallus gallus'u ifade ediyor. Yani bildiğimiz tavuk. Evet, chicken anemi virüs ve girovirus galga 1 Neuroblastoma familyasının girovirüs cinsinde oluyorlar. Girovirüsler küçük, sirküler, iplikli DNA'dan oluşan bir genoma sahipler. Günümüze kadar girovirüs cinsinde 10 farklı tür tespit edilmiş. Bu 10 farklı girovirüs türü tavuklara yanı sıra insanlar, emirgenler ve çeşitli evcil hayvanlarda da tespit edilmişler. Bu çalışmada bizim amacımız ilk olarak sağlıklı olan evcil güvercinlerde chicken anemi virüs ve girovirus galga 1'in varlığını ortaya koymak. Materyal olarak Elazığ, Sivas, Elazığ, Malatya ve Tokat illerinden az önce de söylediğim gibi klinik olarak sağlıklı 11 farklı evcil güvercin sürüsünden Türkiye'de güvercin sürü veya kümes olarak bahsedilebiliyor. Yani 11 farklı evcil güvercin kümesinden ve her kümesten, her sürüden 9 ila 10 adet olmak üzere toplam 103 adet dışkı örneğini topladık. Her dışkı örneği steril PBS ile %10 oranında seyreltildi ve homojenize edildi. Homojenatlar 3000 RPM'de 10 dakika santrifüj edildi ve ticari bir DNA izolasyon kiti kullanılarak bu santrifüj sonucu elde edilen süpernatamplardan DNA'lar ekstrakte edildi. Harita üzerinde örneklemeleri yapmış olduğumuz illerin konumlarını gösteriyor burada. Tokat, Sivas, Malatya ve Elazığ illeri. Biz çalışmamızda kullandığımız primer setleri, nested primer setleri kullandık biz. Hem chicken anemi virüs için hem de girovirus galga 1 için. Girovirus galga 1 için viral protein 2, chicken anemi virüs için ise viral protein 1'i defledik biz. Chicken virüs VP1 geninin PCR testleri için kullanılan primer bağlanmasılar ve döngü parametreleri sunuda, ekranda görüldüğü şekilde uygulandı. İlk tur 51 santigrat derece, ikinci turda da nesnede yani 50 santigrat derece annening ısı uygulandı. Virüs galga 1 viral protein 2 geninin PCR testleri için ise kullanılan primer bağlanmasılar ve döngü parametreleri yine nasıl da görünüyor şekilde. İlk turda 50 santigrat derecede, ikinci turda da yine 50 santigrat derecede annening ısısı uygulandı. Bulgulara geldiğimizde çıkan virüs VP1 geni için PCR sonuçlarımız etidyum bromür boyama ve ultraviyole ışığıyla görselleştirildi ve amplikonlar agrojel elektroforezde yürütüldü. Ekranda işte pozitif örneklerin tespit edildiği çıkan virüs için bir gel görüntümüz var. Aynı şekilde virüs galga 1 VP2 geni içinde PCR sonuçları yine etidyum bromik boyama ve ultraviyole ışığı altında görselleştirildi. Bu PCR sonuçlarına göre 103 dışkı örneğinin 23'ü yani yaklaşık %22,3'ü anemi virüs yönünden, 78'i yani %75,7'si de virüs galga 1 yönünden pozitif bulundu. Yüzleme ve filogenetik analiz, bunu biz hizmet alanı şeklinde yaptık. Yazıda görülen kitler kullanılarak ve cihaz kullanarak AB 3100 cihazı kullanarak sekans analizi yaptırıldı. Çalışmada elde etmiş olduğumuz çıkan virüs suçlarının diğer çıkan virüs suçlarıyla kısmi VP1 gen sekans benzerlik tablosu şekilde olduğu gibi buna göre, bu şekle göre çıkan virüs suçlarımız kendi aralarında %96 ile %99,7 aralığında benzer bulundu. Bankta mevcut diğer Türkiye tavuk suçlarıyla 96 ile %100 arasında bir nükleotit benzerliği. Bankta mevcut diğer ülke tavuk, evcil kedi ve insan suçlarıyla bu oran %92,7 ile %99,2 aralığında bulundu. Virüsler aynı zamanda insanlarda da tespit edilen Anelloviridae familyasının virüs cinsindeki virüsler. Ve çıkan virüs suçlarının kısmi VP1 gen bölgesinde yaklaşık 355 bas p'lik filogenetik bootstrap konsensus ağacı oluşturuldu. Bu ağaç Zeus Prime adlı bir yazılımın 2002'ye 0.2 sürümüyle gerçekleştirildi ve burada neighbor joining yöntemi kullanılarak bu ağaç gerçekleştirildi. Özellikle 4 suçumuz birbiriyle oldukça yakın görünürken bir tane özellikle 45 nolu suçumuz diğer Türkiye izolatlarıyla daha yakın olduğu tespit edildi. Evet, çalışmada elde edilen virüs galga 1 suçlarının diğer virüs galga 1 suçlarıyla kısmi konusu da şekilde görüldüğü gibi oluştu. Suçlarımız kendi aralarında %100 benzerdi. Tamamen birbirlerinin aynısıydı. Bankta mevcut diğer ülke tavuk, evcil kedi, yılan ve insan suçları ise bu oran %96 ile %100 arasında bulundu. Virüs galga 1 suçlarının kısmi VP2 gen bölgesine yani 560 bas p'lik filogenik bootstrap konsensus ağacı yine Zeus Prime 2022 sürümüyle ve neighbor joining yöntemi kullanılarak gerçekleştirildi ve başta Brezilya, İtalya, Çin suçlarıyla aynı konu üzerinde oldukları tespit edildi. Sonuç olarak çalışmamız Türkiye'deki evcil güvercinlerle yakın virüs ve virüs galga 1 prevalansına ilişkin bilgileri sağlamış ve evcil güvercin dışkı örneklerinden elde edilen resmi genomik verileri ortaya koymuştur. Bu aynı zamanda Türkiye'deki evcil güvercinlerde chicken anemivirus ve pirovirus galgabile dair ilk rapor olarak kayda geçmiştir. İlginiz için teşekkür ediyorum. Sunumum burada tamamlandı.
Dear Professor Turan, thank you very much for your detailed work. Any questions to Dr. Turan? No. Okay, I'm going to the next presentation. Dr. Sevgi Şengül Ayan, estimating currents from action potentials using single and multi-output neural networks models.
Hello, Professor. I'm going to do our presentation. My name is Selim Süleymanoğlu, and let me share my screen. All right.
No,
You
wait.
can see it now.
No.
Says I am screen sharing. All right, let me try the whole screen then. Now,
Wait, please. We cannot see. We are still on Dr. Turan's screen.
maybe he needs to stop sharing.
Sorry. Is it your screen?
Yes, I'm sharing my screen now.
Okay.
All right. Hello, everyone. Welcome to our presentation on estimating currents from action potentials using single and multi-output neural network models. We worked with cardiac action potentials, which are a recording of a cell's membrane potential versus time that we measure from ion channels. During each cardiac cycle, ions move back and forth across the cardiac cell membranes to create membrane voltage changes. These voltage changes are largely dominated by the calcium, sodium, and potassium channels. Here we see the phases of each of a cycle. In the first rising age, phase 0, sodium ions start to get in and out of the cell, and after them, during phase 1, potassium channels open, and then as we reach phase 2, we reach a plateau, and calcium channels also open, and they slowly start to close, and then we reach phase 4, a resting phase. In our model, we fit real data that we gathered at Akdeniz University to a nonlinear ordinary differential equation system. We used changes based on the, we used data that we collected based on conductance changes. Our formula in its simplified form is as shown on the screen. Here we use the ion currents and an external stimulus current and divided by the membrane capacitance to get our most valuable value, which is the membrane voltage divided by the time change. Here is a representation of our figure of our system. On the left-hand side, the potassium, sodium, and calcium channels are represented. On the right-hand side, we see the background channels. On the bottom, we see the different potassium channels, which are rapid, slow, and transient mainly, and an invert rectifier potassium current. Here on top left, we see our action potential. And the other ones are the currents that are generated depending on this action potential. The red lines represent the control groups. The black lines are 880 sets of currents and action potential. So we have different channels such as sodium, slow potassium, rapid potassium, and here L-type, long-lasting, calcium, transient potassium, etc. And here are the other ones. And we have high frequency current and color background to finish those up. The method we selected was Bayesian regularization. We compared this method with Levenberg-Marquardt and scaled conjugate gradient backpropagation. Scaled conjugate backpropagation was the fastest one, but it was the least accurate one, so we eliminated that one. The Levenberg-Marquardt method was also decent, but not as good as Bayesian regularization. That's why our artificial neural network model uses Bayesian regularization. Our results were obtained via perturbations to 5 independent variables. 4 of them are conductances, and 1 of them is the membrane capacitance. Our main question that we tried to answer was, given only the action potential, can we predict ion channels? The results of this question are shown on the screen. The blue lines represent the predictions made by the model, and the red lines are the real value. Here, although just as we enter phase 0 at the start of the rising edge, sometimes we see jagged lines, some inaccuracies. But other than that, the accuracy is close to 100%. Here are the next 4. As you can see, sometimes at the beginning of the rising edge, again, there is some inaccuracies. But other than that, we are glad with the results we got. And here are the next 4. So what's in the future? Our main purpose is to make it available to everyone, that they can get their data, action potential data, put it into a system. This can be as simple as a mobile app, ideally. And then they can get the predictions for the ion channels. This can be useful for disease detection. For example, endocrine cells, when they release a hormone, they make use of these action potentials. But also heart diseases, as we used cardiac action potentials here, that is closely related to that. And these can be used for making medicine in pharmacology or some other experimental researches. Thank you for listening to our presentation. If you have any questions, we can take them.
Okay, thank you, Dr. Süleymanoğlu. Are there any questions? No, there's no. If you have any questions, you can use the chat chat room. Okay, now we are going to the next speak. Dr. Semiha Dertli. Personnel working with disabled children in special education institutions, first aid, information and practices. Dr. Semiha Dertli, you can share your screen. Okay.
Good morning, everybody. Welcome to my presentation. My name is Semiha Dertli. I am research assistant at Fırat University. My topic of presentation, personal working with disabled children in special education institutions, first aid, information and practices. Spatial education institutions maximize of abilities of children with disabilities and ensure their integration into society, carries out studies to develop basic self-care skills, independent living, and professional skills. Disabled children spend most of the day in the institutions, and therefore they may be exposed. It's of great importance that the personnel are sufficient in terms of first aid application and in accident that occur in institutions. This research was carried out as a descriptive study in order to determine the first aid knowledge level of personnel working with disabled children in special education institutions and the factor affecting their knowledge levels. This research was conducted with 145 personnel working in special education and rehabilitation centers affiliated to the Directorate of National Education in a province in the east of Turkey and volunteering to participate in the research. Introductory information questionnaire were used to collect data. Descriptive statistics, Mann-Whitney and Kras-Kalvais tests were used in the analysis of data. It was determined that the average age of personnel participating in research was 29.41 plus or minus 7.08 years. 66% were women and 84.1% were underage ages. It was found that tenure of personnel in institution was 4.18 plus or minus 3.85 years and 66.2% of them worked as teachers. It was determined that 8 of the personnel work in institutions received first aid training and 53.1% of the trainees received this training from the school they graduated from. It was stated that 49 of the participants rarely encountered events that required first aid in the institutions. It was determined that 62.1% of them evaluated their first aid knowledge as moderate, and basic first aid practice questionnaire average score was 14.22 plus or minus 2.66. It has been found that most common types of accident encountered by personnel working with disabled children in special education institutions are falling hit and fainting seizure. In the study, it was determined that average score of the basic first aid practices questionnaire of personnel working in institutions was high. It was determined that the average score of personnel who received training and related the first aid education level as good was higher, and difference between the groups was statistically significant. In with this result, it's thought that a child's life can be or lifelong qualified training programs to the personnel working in programs.
Okay. Any addition, any contribution, or any questions to Dr. Dertli? No. Okay. Thank you, Dr. Semih Dertli.
Thank you.
Now for the next presentation, I would like to invite Dr. Cüneyt Yavaş. Whole exome sequencing results in follow-up patients with nephropathy, a two-center retrospective study.
Thank you. I'm sharing my screen. Do you hear my voice?
Yes.
Okay. Sorry. I'm taking a little time. Thank you for coming today my presentation. My name is Cüneyt Yavaş. I'm Chief Technical Officer in Başakşehir Çam ve Sakura Genetic Disease Assessment Center. Today I will present our findings on nephropathy patients who investigate exome results. As you know, a normal kidney has about 1 million filtering units. Each unit, called a glomerulus, connects to a tubule which collects urine. Conditions such as morphologic and physiological change take a toll on kidney function by damaging these filtering units, collecting tubules, and causing scarring. It is the clinical reflection of glomerular disease that occur clinically as a result of change in the selective permanently of glomerular capillary wall and progress with proteinuria and edema and hyperlipidemia at the nephrotic level. These changes are caused by genetic variation or non-genetic variations such as high blood pressure, minimal change, disease and diabetes or lupus, etc., etc. But diabetes is the leading cause of chronic kidney disease and end-stage kidney disease in the United States and worldwide. About 26 million people in USA were reported to have diabetes, and more than 200,000 people with end-stage renal disease due to diabetes were either on chronic renal dialysis or had a kidney transplant. Diabetic nephropathy occurs 20% to 50% of these living with diabetes. Although diabetic nephropathy is more common in the studies, but we want to examine present and genetic distribution of non-diabetic nephrotic patients in our study. Also, one of the important factors determining the prognosis in children with nephrotic syndrome is genetic factors. We are using MGI DMB-Seq next generation sequencer in our laboratory. I want to brief information about the workflow. We isolate from peripheral blood obtains gDNA. The gDNA concentration was measured with qubit, and we recommend that the concentration of isolated patient gDNAs 50 nanograms per microliters, and after that gDNA samples were fragmented and bead purification was performed after barcoding. Every patient's barcoded with different barcodes. The resulting library products were converted into the DNA nanoballs and loaded into the device. We take FASTQ data from the device for analyze. I recommend twist human compressive exome because the other kits very bad about the coverage of all genes. Also, we detected variation were confirmed on the AB3500 device, and segregation was performed in those families whose could be reached. Not every family, but we can reach families. We performed that. Also, this is our results. The average rate of findings clinically relevant variation in patients with nephropathy was between 10% and 40%. According to the result of our study, pathogenic or variation of unknown significant variation associated with kidney disease was detected 24 of 50 patients. This rate is very higher. The other studies, it's about 48% are founding genes. This results was found to be above the average kidney disease genetic studies. In addition, in our studies, 9 variations were identified as a novel variation. We are first finder this variation in nephrotic patients. Also, the mean age of the patient was 11 years old, and they ranged to 0 to 60 years old. It was observed that the majority of patients with nephrotic syndrome were under the age 17. It has been thought that genetic factors is important in adolescence and earlier age. Also, most of the detected variation were found to be de novo. We had been determined that patients with familial inheritance are few. As you see, the most causes damage to kidney, Alport syndrome, and polycystic disease are we determined. It has been understood that the genes with the most variation are PKD1, PKD2, and Alport associated genes, C4A3, C4A4, and C4A5. Therefore, Alport syndrome and polycystic kidney disease genes should be especially looked in the patient with nephropathy. I want to introduce shortly brief about the Alport syndrome genes. Alport syndrome has a spectrum of phenotypes ranging from progressive renal disease with extrarenal abnormalities, hematuria, non-progressive or very slow progressive course. The PKD, about PKD, there are 2 types of inheritance of PKD, autosomal dominant and autosomal recessive inheritance. Autosomal dominant polycystic kidney disease is generally late onset and multisystem disorder characterized by bilateral renal cysts, liver cysts, and increased risk of intracranial aneurysm. Autosomal recessive polycystic kidney disease belongs to the group of congenital hepatorenal fibrocystic syndromes and is a cause of significant renal and liver-related morbidity and mortality in children. The other ones is very few about in patients, nephrotic syndrome, FCGS, and nephrolithiasis, fibronectin deposits, and familial Mediterranean fever. Thank you for your attention and your patience. Further information and questions, you can connect with me as you see my email. Thank you.
Thank you, Dr. Yavaş. for your nice presentation. Are there any questions to Dr. Yavaş about his work, their work? I think no. Okay, thank you, doctor.
Thank you.
For the next speaker, for the next presentation, Dr. Lubna Dafrallah, a Moroccan family with distal hereditary motor neuropathy and the C452CT variant in the small heat shock protein HSPB1. Okay, Dr. Dafrallah.
Okay, can you hear me?
Yes, we are hearing you.
Hi, everyone.
Could you please share your screen?
Okay.
If possible. Also, you can open your camera if possible. On the top, there is a yellow, green bar to share screen.
Can you see now?
No, we can see your camera, but we can see your, you don't share your screen. We cannot see your slides.
Mm.
Dear Professor Artekintosun, may I describe the process to the researcher? Dear Dr. Lubna, first you have to open your presentation. Afterwards, after opening your presentation, you have to go Zoom screen, Zoom. Then at your screen, at the bottom, there is a green button. Just click on it. Just click on it. Afterwards, you will see a window. In this window, you have to select your presentation, PowerPoint presentation. If you need more time, we can continue with the next speaker. Then during you can understand the process.
Okay. Okay. You can pass the.
Okay. For the next speaker, I would like to invite Dr. Berna Özdem for their presentation on investigation of the apoptic effects of indometazine on SHSY5 neuroblastoma cell line.
Hi everyone, dear mycologists. I will share my presentation first of all. Okay.
Would you please share your screen?
Of course.
Thank you.
Could you see my screen?
Yes, now we are seeing your screen.
Okay. Okay. First of all, hello, dear mycologists. First of all, I would like to say I'm very pleased to be here. I am Berna Özdem. I'm a PhD student at Indonesia University Medical Biology and Genetic Department. Our study will be about the cancer, neuroblastoma cancer, and I will start with some information about our cell line. We studied in vitro. First of all, SY5 cell line. It's actually the metastatic neuroblastoma cell line. This is generated from a metastatic bone tumor from a 4-year-old cancer patient in 1970. So, and I would like to talk about another thing, Indometazine, because when we searched the literature, we found that some results about the Indometazine. And as you know, maybe, Indometazine is a non-steroidal anti-inflammatory drug, and this drug is generally used for the swelling, tenderness, pain, and fever. These are signs of inflammation. It's used to treatment of inflammation disease, such as arthritis and tendinitis and etc. And you can see from here, this image, you can see the structure, molecular structure of the Indometazine and molecular weight. And you can see in literature, in one study, the researchers stated that the Indometazine, that is induced apoptosis in doxorubicin resistant small cell lung cancer cells via MRP1 dependent mechanism. This is very important for the resistance in cancer, because the doxorubicin is a cancer drug, as you know, and this MRP1 multi-drug resistance associated protein 1, and this protein that is found in humans, it's encoded by the ABCC1 gene. This protein, this multi-drug resistant protein 1, is an ATP binding acid, ABC polytopic membrane transporter of important clinical importance that is the confers multi-drug resistance in tumor cells by reducing drug accumulation via active efflux. So it's important. And another study, when we searched the literature, we found that some researchers that found that the Indometazine was shown to induce apoptosis in EC109 esophageal cancer cell line by releasing the second mitochondria-derived caspase activator and activating caspase-3. As you know, there is a 3 pathway in the apoptotic mechanism, and this is mitochondrial-derived caspase-dependent pathway. So we decided to search investigation effect of Indometazine on proliferation and apoptosis effects on this neuroblastoma cell line. So our aim was that. Then what was our hypothesis? Our hypothesis was different concentrations of this Indometazine have a positive effect on reducing the neuroblastoma cancer, have a negative effect on the proliferation and positive effect to induce apoptosis. And of course, if I summarize our hypothesis, this drug is a potential anti-cancer. So because of that, we planned some experiments, and this is our methods, MTS, as you know, for the proliferation test, cell viability. First of all, we used this MTS because to calculate the IC50 value, and flow cytometric analysis with annexing PI. Because of that, we want to find the apoptosis rates and Western blotting. This is our first results after the MTS. You can see this viability and concentration ratios. We used also vehicle as a DMSO as a vehicle, because we sold in the Indometazine, we sold in the DMSO. Because of that, we used as a vehicle group. and 50 micromolar, 100 micromolar, 200 and 400 micromolar. We used these rates, these concentrations, and after the 3 days, we calculated this graphic, and as we calculated IC50 value, we found that IC50 value was 30 micromolar. After that, we started to flow cytometric analysis. This is non-treated group. This is with our vehicle group, DMSO, and IC50 value, 30 micromolar group. As you can see from here, this area is meaning the leaf cells. This ratio shows us the leaf cells here, and here totally showed us apoptotic rates. As you can see from this picture and graphics, statistically, we found to significant because our p-value lesser than 0.05. Here you can see the apoptotic rates, non-treated group, vehicle group, and our Indometazine, 30 micromolar. This is our graphic significantly. So we found yes, our hypothesis was true because it induced apoptosis in this neuroblastoma cell line. But we want to prove that results with Western blotting. And Western blotting, in Western blotting, we use the PARP1. Why we use the PARP1? Because PARP1, as you know, during the apoptosis, DNA strands breaks led to PARP activation. So we use the PARP as a apoptosis marker. As you can see, this bonds, non-treated vehicle and 30 micromolar Indometazine. In here, PARP, during the, as I said before, during the apoptosis, PARP1 will be cleavage. Because of that, we want to this actually, and we got these good results. Cleavage PARP1 was cleavage, and as a control group, we used the beta-actin. You can see. As I summarize this study, of course, we need to develop our study with another things. But as a result, in this study, that is showed us the effect of Indometazine vary depending on the dose. Indometazine showed a really stochastic effect on this neuroblastoma cell line, and with the Western blotting and our flow cytometric analysis, we proved that this is also Indometazine increasing the expression of the cleavage PARP1 protein expression. So, and considering these results, the importance of future studies for Indometazine is better understood. Therefore, actually, it was concluded that it can be used as a precursor compound to develop new drug combinations to treat neuroblastoma cancer. And thank you so much to listening to me. If you have any question, I can answer.
Thank you, Dr. Özdem. Are there any questions in chat room? No. Okay, thank you.
Thank you.
Dr. Lubna Dafrallah, are you ready? Dr. Lubna Dafrallah? Okay. I'm going to the next speech. Are you here? Are you ready?
You can, yes, I'm here. You can see? You can see now?
No. Just hearing you.
No. Okay.
Okay, I think it's okay. Now we can see your screen. Dr. Lubna Derfrallah, you are welcome. To their work, a Moroccan family with distal hereditary motor neuropathy and the C452C2 variant in the small heat shock protein HSPB1.
Yes.
Okay.
Thank you. Hi, everybody. I hope you guys doing well. I'm Lubna Dafrallah, PhD student in biology, working on neuromuscular disease in the laboratory of human genetics in Pasteur Institute of Morocco. For my objective is to identify the genetic cause, to identify the mutations of the distal hereditary neuropathy and the mutations of small HSP proteins HSPB1. The distal hereditary neuromorphopathy, also known as spinal form of Charcot-Marie-Tooth disease, comprise a clinical and a genetically heterogeneous group of neuropathies characterized by the degeneration of alpha motor neurons in the peripheral nervous system, leading to slowly progressive distal symmetry limb muscle weakness and atrophy, frequently leading to foot deformities such as pes curvus, pes plans, and the cloud tubes are as you see in this picture. The distal hereditary neuropathies caused by mutations in a large number of genes, about 39 genes or loci associated with the distal hereditary neuropathy, with autosomal dominant, autosomal recessive, or X-linked models of inheritance. As you see in this picture, the different mutations, the different genes associated with the distal hereditary neuropathies. For the proteins, HSP proteins, HSPB1 or HSPB1 was perforated and characterized for the first time in 1987 and 1988, and presenting most neurons of the peripheral and the central nervous system were expressed constantly in more neurons in both the cytoplasm and nucleus. These proteins were described as stress proteins, act as ATP-independent molecular chaperones, whose functions is to protecting cells from various unfavorable conditions. These proteins are associated with the development of hereditary peripheral neuropathies, including distal hereditary neuropathies and Charcot-Marie-Tooth disease. HSPB1 also is involved in many cellular processes, including neuron cell homeostasis, which can prevent the accumulation of aggregates of denaturated or misfolded proteins, and as seen in the proteasomal degradation if they become irreversibly damaged after diverse stresses or environmental insults. Apoptosis inhibition regulates cell proliferation and protect the cell against oxidative stress and inflammatory mediators. You can see here the gene HSPB1 containing the conserved crystalline domain and the different in the C and the N terminal regions. We studied one Moroccan family having members with distal hereditary neuropathy phenotype to identify the leading cause of their disease. For the genetic analysis, we used the peripheral blood samples, was collected from probes and their available family members, and the genomic DNA was isolated. We used the whole exome sequencing analysis of the proband, and this analysis by whole exome sequencing of the proband revealed the mutations variants on the HSPB1 gene in heterogeneous state. In the present study, by whole exome sequence, we have identified the mutation and the HSPB1 in the family of Moroccan region. That's all. Thank you for attention.
Dr. Lubna, thank you for your presentation. Are there any questions? No, there's no question in the class. Okay, thank you, Dr. Lubna. I would like to invite Dr. Binnur Akkaya. Dr. Binnur Akkaya, are you here?
She's here. We are trying to.
I am here. Do you hear me?
I would like to invite Dr. Binnur Akkaya for their presentation, New Heparin Mimetic Material and its Anti-Cancer Activity.
Okay, I will try to share the
Okay.
presentation. Can you share the PPT?
No, no, just video.
Only
and voice.
so must I switch off the video, or maybe I? Share. I try to share, but I cannot share.
Dear Dr. Birnur, you have to open first your presentation.
Yes,
Afterwards, you have to come your Zoom screen, Zoom window.
Yes.
At the bottom of the window, there is a green button. Please click on it. Then you will see another window. In this window, you have to select your PowerPoint presentations. Just click on it, then it's going to be open.
First of all, I must start the video. Is it true? No.
First, you have to open your PowerPoint presentation.
Open now.
Afterwards, you have to go to your Zoom window. Then in this Zoom window, there is at the bottom a green button. Do you see it?
No.
Green button.
No.
Share screen, or it is written in Turkish, ekran paylaşımı.
No, I cannot see it. No?
After the video, microphone and chat room and share screen. And the button. Katılımcı sohbetin yanında ekran paylaşımı yeşil diye geçiyor altta.
Gördünüz mü hocam? İsterseniz WhatsApp'tan atarsanız biz de açabiliriz. Sorun yok. En alt kısmında.
Ben daha önce de katıldım aslında bu şekilde. Share dediğim zaman açılıyordu ama şu anda ben göremiyorum. Yani söylediğim...
Ekran paylaşımı düğmesine bastınız mı hocam? Aşağıdaki.
Aşağıda ekran paylaşımı yok ki. Yani
şu anda siz ne görüyorsunuz hocam? Karşınızda ne var?
Benim şu anda karşımda Zoom meeting var. Bu Zoom meeting'de live var, view var. Burada ne var? Share screen. Okay, sorry.
Evet, ona tıklayın hocam.
Tamam. Kusura bakmayın.
No problem.
Can you see now?
Not yet. Okay, now we can see your screen.
I'm so sorry for deletion. And, welcome to the Second International Congress on Biological and Health Science. I will try to present new heparin mimetic material and its anti-cancer activity. In recent decades, heparin and heparin sulfate derivatives and analogs have been synthesized from natural sources or through chemical synthesis. Sulfated glycosamine glycans are glycosylated glycans that have been sulfated. They are known as heparin sulfate mimics, and they play an important role in the body. Some recent advancements in cancer therapy have played a role. GAGs are natural heteropolysaccharides. As previously stated, they are found in all mammalian tissues. They are composed of repeated patterns. Disaccharide units are made up of sulfated or non-sulfated monosaccharide units. The size of their molecules and the type of sulfation present vary. They can be found on the tissue as free molecules or as part of the proteoglycan. In the current study, we synthesized a new heparin mimetic materials, and its anti-cancer activity was investigated on MCF7 cell line. Multiple investigations have shown that heparins cause cancer through in a variety of different ways, such as cell-cell contact is inhibited by preventing cell adhesion molecules, such as selectins, and extracellular matrix inhibition, heparinase, proteinase, and angiogenesis inhibition. Heparin, it is used therapeutically as an injectable solution as an anticoagulant, as you know. It has also been used in forms such as coatings in various biomedical devices, as well as for various indications. In addition to the properties of these polysaccharides, such as anticoagulation, tissue regeneration, anti-inflammation, and protein stabilization, they also contain the disadvantages of their use due to their various functions in the body. So materials that mimic heparin are widely investigated in therapeutic applications. In this sense, the synthesized heparin-like polymeric materials have found widespread use in the field of medicine due to their improved therapeutic properties and reduces side effects. Here you can see the heparin structure. Heparin is a linear, highly sulfated glycosamine glycan produced by mast cells. Its chemical structure consists of monomeric units of uronic acid and glycosamine linked by repeated 1,4 bonds. Also, you can see here in the structure that there is red colored the sulfate groups, blue colored the carboxylate group, and green colored of sulfamate groups. Polymeric heparin mimics can provide better control over structure, sulfation, and purity. It is also possible to obtain a narrower molecular mass distribution. Also, heparin mimicking polymers typically resist degradation, desulfation by heparinases, allowing them to last longer in the body. This can give positive or negative results depending on the application. Although the heparin mimic polymer has been studied extensively, chitosan was used in the study. Chitosan, dextran, cellulose, alginate, and there are a lot of another polymeric natural polymers have found a great place in heparin mimic studies because they can be found naturally and they are also cheap, and you can modify all of these polymeric materials. Here you can see the structure of the chitosan. You can see also the functional groups of the chitosan. Chitosan polybeta 1 to 4 and acetyl D-glucosamine is the acetylated form of the chitin polysaccharide. Compared to chitin, chitosan has higher chemical and biochemical reactivity due to the regularly distributed free amino group structure in the molecular chain structure. So you can modify the chitosan by modification of these functional groups, and you can mimic as heparin, and also you can use it for another mimicking molecule. Chitosan is a non-toxic, biocompatible, and biodegradable amino polysaccharide. For this reason, it has application in many areas, from clinical applications to the food industry. For example, it has been used frequently in wound healing, strengthening the immune system, drug delivery, and antimicrobial studies. In addition, since it has an important antimicrobial effects against fungicides, there are many studies on food industry in order to protect foods against fungi. Chitosan oligosaccharides is used because heparin mimics change activity according to their molecular mass that we mentioned before. Experimental section, there are a few slides about the experimental section, but I would like to mention this section later if the time is enough. So the chitosan sulfonation, surface morphology, cytotoxic effect studies, antimicrobial effect studies are the protein absorption experiments, are the experimental section titles. So we can mention to the results. You can see here the SEM results. Figure shows the SEM micrograph of both chitosan and sulfonated chitosan. It can be seen from figure that the surface of chitosan itself is porous and has a large specific surface area, and the surface of sulfonated chitosan is more rougher than and more porous than those of chitosan, which is due to the sulfonic group to reduce the cross-linking of density, thus increasing the capacity of holding water. In this presentation, you can see the absorption, absorbed amount of protein per polymeric bead, gram polymeric bead. At pH 7.4, a slight variation in the BSA absorption could be observed at the first time point. At this pH, both the protein and the chitosan present a high negative charge density and so reducing the electrostatic interactions and possible the amount of absorbed protein. At the same pH, raw chitosan has amino groups that are less electronegative than the sulfonate groups of modified chitosan, causing a decreased interaction with PSA. And we want to actually decreasing the protein absorption capacity. The addition of sulfonated groups creates a negative surface, then impeded the absorption of PSA and fibronesion also. In the current work, heparin mimetic material was synthesized by chitosan sulfonation. The result of cell viability test showed that this new material has cytotoxic effect on MCF7 cell line, and the result was suitable by the literature. The current study, in the current study, cytotoxicity of the chitosan oligomer, and actually we used here not the chitosan, we use in this study chitosan oligosaccharides. We can see that it is also the same with the chitosan oligomers, and the molecular weight of the chitosan oligomer is 6 kilodalton. So we modified it with the sulfonated groups. So we saw the structure gain heparin mimicking material, and in the current study, cytotoxicity of the chitosan oligomer and sulfated chitosan oligomer was determined by XTT assay, which was performed by using different concentrations between 1 to 100 microgram per milliliter at 24 hours. The table shows the IC50 values calculated from the concentration via cell viability percentage. In the current reported data, sulfated chitosan oligomer had an value, IC50 value for MCF7. The cell viability test of chitosan and its degraded product or derivatives on some cancer cells has been reported in the literature. In the literature, sulfated chitosan was studied with breast cancer cells like here, and the results show that IC50 value was 35 micromolar. However, in the present study, sulfated chitosan showed that IC50 is 46, about 46 microgram per milliliter against MCF7 cancer cells. The literature also showed that sulfonated chitosan efficiently inhibited the MCF7 cell growing as those time-dependent manner via apoptosis. In this slide, we can see the results of the antimicrobial effect as showing the inhibition zone. The potential pathogenic bacteria such as Staphylococcus aureus, gram-positive bacteria, and E. coli as gram-negative bacteria was used. The active antibacterial property of sulfated chitosan against E. coli and C. aureus indicate that the sulfonic groups introduced onto chitosan backbone are very important. Inhibition zone is decreased for E. coli, like in literature, Kocabay and et al., like Kocabay and their co-authors researched, and increased for say aureus, like Hankett and their friends was worked. So we try to investigate about this new heparin mimetic material. If we only the morphologic screening and protein absorption capacity, only for the one cell line, the FCF7, and antimicrobial effects. And we try to continue with preparing the same heparin mimetic material with gaining the material, the magnetic properties, incorporated the magnetic properties, and we will try to investigate for the other antimicrobial agents and the different anti-cancer cell lines. Thank you very much for your attention and listening to me.
Thank you, Dr. Akkaya, for your presentation. Are there any contribution or any question to the researcher? Let's look at the chat room. No. Okay, thank you, Dr. Akkaya. I would like to invite the last speaker, the researcher, Dr. Abdur Rahman Sial. Can you hear me?
Yes, I'm hearing you.
Yes, would you please share your screen?
Okay. Just a minute, please.
Okay.
Just a second.
Okay. We can see you, we can hear you. Just share your screen, please.
Yes, please. My screen is shared with you.
Just a moment, please. Okay, Dr. Abdurrahman Sial will present their work. Exploration of gingiber officinale powder effects on performance, syrup constitutions, and good morphology in broilers. Okay, doctor.
Okay, I'm trying to just putting the computer on slideshow. I'm clicking on this option. Maybe it's available or not. Just a second, please. Okay. Hello, everyone. Good day. First of all, I would like to thank Dr. Ulash Ajarus and his team for organizing this wonderful event. Despite of the various limitations, especially the limitation of the COVID, he has provided us the opportunity to interact with the various scientists of the subjects. And I'm also thankful to the administration of the Kojotep University and his organization, especially Professor Mehmet Karakash, Rector of the University and Dean of the Faculty of Veterinary Medicine of Kojotep University. I'm also the alumnus of this prestigious institution. I did my PhD from this university of Kojotep University. So I'm feeling pride that I'm here again on this forum to present my talk. So let's start with my presentation. The topic, as the Professor Tosan already described, that I want to talk on the effects of the gingiber officinale in broilers. As far as my introduction is concerned, I'm Abdur Rahman, Assistant Professor in Animal Nutrition from University of Veterinary and Animal Sciences, Lahore. As far as the introduction of my presentation is concerned, or my the background of my study, actually the commercial poultry production was started in Pakistan in 1962, and then the growth of this sector was very rapid, and now this sector is contributing about 1.4% in the national GDP of our country, and more than 1.5 million people, they are employed in this sector, and the meat of the poultry is very important because it's easily available, it's cheaper, and it's providing a major segment of human protein, human diet protein, especially to the masses of rural and the poor, you can say poor areas. And the poultry meat is, of course, it's well understood that it's very balanced and it's good quality meat, having vitamins and minerals, of course, and has good quality fat as well. So with this background, we focused and we tried to study the effects of some, actually we can say we tried to investigate some alternative solutions of the antibiotics, because the antibiotics were commonly used and still being commonly used in various parts of the world as AGPs. I know that the European countries have banned the use of antibiotics, but still in the developing countries and in our country, we are still using AGPs. So keeping in view the subject of microbial resistance, we try to investigate some alternative solutions to be used to replace the antibiotics in the poultry farm operations. Here we can see the comparison of antibiotic growth promoters with the phytobiotics. We know how antibiotics acts as a growth promoters, and here we can see how phytobiotics works to enhance the growth or to promote the growth performance in the poultry farms. So the comparisons gives us a spectrum that yes, the phytobiotics can perform well. Among the phytobiotics, we can see there are many products which can be used to replace or as additives in the poultry feed. If you can see on the left side of this slide, among the herbs, flowering, and the non-woody plants, the ginger, the officinale, which is our target product, is coming in this category of the phytobiotics. The ginger, which is our target product, has different bioactive components of the desired effect. For example, it has gingerol and the geranyls and the gingerone and the gingerdione and the gingerdiol, paradol, and the 1-dehydro-6 gingerdione shagols and the 6-dehydroshagols. These are the different bioactive components that this product has. If we see the composition of this product, it has the protein and some value of the fiber and the ether extract and ash and NFE. Although we are utilizing this product as an additive, not as a nutritional value nutrient, rather we are going to use it as a feed additive to replace the antibiotics. The application, again, we have discussed that the ginger has antimicrobial activity and the stress alleviator, and it has the antioxidant potential. It has the potential to enhance the growth and has some activity of the secretion of the digestive enzymes. Some studies already showed that this product has potential to enhance the growth and some immune parameters and others. So with these objectives, we performed a trial on the broiler, and the design of the study was like that. We have used 1,500 day-old broiler chicks, which were divided into 5 groups, and each group was having 6 replicates with 50 buds in each replicate. The design was like that. the group A was fed with the basal diet, having antibiotics, which is controlled in our country, and the other group B was considered as the negative control, fed basal diet but without antibiotics. And other three treatments, the C group was fed D basal diet without antibiotics, supplemented with the target product ginger 0.3%. And then the group D was supplemented again with the basal diet B with the ginger at the rate of 0.6%, and the group E was supplemented with the basal diet B along with the 0.9% supplementation of the target product ginger. So the two types of the diets were offered to the birds: the starter diet and then the last phase, the finisher diet. The following parameters were evaluated. The pre-slaughtering evaluation was performed on the feed consumption, and then the body weight gain and the FCR. And the post-slaughtering evaluation was performed on the carcass characteristics, total bacterial count, and the gut morphology, immunological tests, and the blood serum biochemistry. Some tests from the blood serum biochemistry were performed. For the sample collection, especially for blood, sample were collected and the centrifuged, and serum were separated and stored at -80 degrees centigrade for further analysis. And the analysis of the serum triglycerides, HDL, LDL, and total cholesterol were performed. As far as the results are concerned, you can see the feed intake results showed that at the 5th week, the supplemented groups with the ginger shown the better results, especially the group E, which were supplemented with the ginger at the rate of 0.9%, has shown that the feed intake was significantly higher at the end of the study. And similarly, the body weight gain showed also the significantly improved body weight in the supplemented groups with ginger. But if we can see at the 5th week, the body weight of the birds in the group D, which were supplemented with the ginger at the rate of 0.6%, was significantly higher than the controlled, positive control, negative control, and all other groups. And then if we see the results of the feed conversion, yes, at the 5th week, again, the feed conversion ratio was better in the group T, which were supplemented with the ginger at the rate of 0.6%. Similarly, the carcass statistics, the parameters of the live weight and the eviscerated weight and the dressing percentage and the giblet weight percentage, these parameters remained unaffected, and there was no positive or no adverse effect of this supplementation of the ginger as compared with the control group. And then when we talk about the immune response of the dwellers with the supplementation of this product, again, we can see that the group D supplemented with the 0.6% ginger showed positive the better ND titer at the day 28th of the trial, but there was no effect on the we can say no significant effect on the 35th day. And then regarding the total bacterial count, the bacterial count was again lowered with the supplementation of the ginger product at the rate of 0.6% in the group D as compared to other groups, and especially as compared to control A and negative control B group. And the results regarding the gut morphology, the villi length was significantly better in the group D, and the villi width was again better in the group D, and again the same results, the same type of results were found in the group D, supplemented with 0.6% ginger. These are the pictures of the histomorphology. And then the serum biochemistry results showed again the triglycerides, cholesterol in the first column. The cholesterol was lowered, significantly lowered in group D and in group E as compared to others, which were supplemented with the ginger at the rate of 0.6 and 0.9% respectively. And then the triglycerides was also lowered in the same groups, and the HDL was better in the group D, the usual cholesterol, and LDL was lowered in the group D with the supplementation of the ginger at the rate of 0.6%. So as a conclusion of this study, we can conclude that the ginger as a natural affiliative can be used for the intestinal health, for the immune response, better immune response, and for better growth. Thank you very much. Yes, if there is any question, we can talk.
Dr. Sial, thank you very much for your nice presentation. I will take care for ginger for myself. Any questions to Dr. Sial? No. Are there any delayed presenters or any researcher more that delayed to the session? I think no. Before closing this session, I would, dear respected researchers, I would like to invite both of you to speaker section that will start 12 o'clock in Turkish local time, 20 minutes later. Your presence will honor us. Would you please just take a coffee break time now? And after coffee break, let's meet together at 12 o'clock, okay? Dr. Ejaros, any more?
Thank you very much, dear Professor Dr. Alptekin Tosun, for your presence and for your contribution and support. It was very, very nice to do something together. And thank you again for your nice moderation.
Okay. Okay, thank you. I'm closing the session.
Yeah.
Bye.
Bye. Dear respected participants, we are today with the third day of our Congress, and the session for invited speakers will start with the presentation of Professor Dr. Alptekin Tosun. He is a radiology professor from the Giresun University Faculty of Medicine. Dear Professor Dr. Alptekin Tosun, thank you very much for accepting our offer, and again for your support. We appreciate. We are happy to listen to you.
Thank you, Dr. Ulaş. Thank you very much for your excellent Congress organization. Can you see my screen?
We can see your screen, but can you do it full screen?
Now it's in full.
In your screen, yes, but for us not. Please go upside of your screen.
Yes.
Then click the green button. Then select the reshare. Then select your presentation. First, you have to make full screen in your computer. Then you have to go upside.
Okay. The
Then
green button.
Green button. There is two different options of your presentation. Please select the slideshow.
Paylaşım üstüne tıkla, paylaşım üstüne tıkla. Şunun üstüne tıkla.
Is it okay now? Okay.
Yes, perfect.
Again, thank you, dear participants, dear researchers, dear Congress organizers. You are all welcome. I would like to talk about the pandemics, novel subject, the diagnostic approach to COVID infection, what has changed, and what's the place of radiology. Which diagnostic tool? The diagnostic methods are first direct diagnostic methods. This depends on virus isolation, molecular tests, antigen testing tests. Also the part of the body, antibodies, serological diagnostic tests, rapid antibody tests, immunoglobulin M, G, ELISA, immunoglobulins, and other biochemical and hematological parameters depends on the body, the immune response. So blood counts, lymphopenia, eosinopenia, procalcitonin levels, ferritins, CRP, sedimentation rate, interleukins, D-dimer, lactate dehydrogenase, liver function test, creatinine, and also we apply to the radiology imaging methods. Which are these? Mostly depends on radiation, and X-ray is the first step. That is research for opacities and ground glass densities, and advanced method is CT, multiple patch infiltrations, peripherally disseminated interstitial changes. The virus intake to the body, and we get the infection, the peak infection, overt disease, and after recovery, and finally the immunity, if not that, the immunity is starting. So we are looking at the laboratory diagnostic tests, RNA tests, gold standard for antigen, SARS-CoV tests, and antibody tests, immunoglobulin M, because the acute infection period. And in recovery periods, we are looking for discharge RNA tests. And after immunity, we are looking for antibody tests, the chronic immunoglobulins, immunoglobulin G. After the vaccine development, we are looking for, without the infection, we are looking for the immunoglobulin G. Virus isolation, molecular diagnostic tests, rapid diagnostic tests. These are looking for antigen and antibody detection techniques, non-technical methods and biosensors. First of all, the nasopharyngeal or oropharyngeal swabs, they are looking for the nucleic acid base diagnosis. And in the part of the body, we are looking for the blood count and serological based COVID diagnosis. We are looking for the antibodies. Before the symptoms, the virus intake to the body, first of all, after incubation period, is starting the infection. So the symptom onsets in this period. After the symptom, the increasing probability of the detection is possible after the symptom onset. These methods are nasopharyngeal swabs, virus isolation from respiratory tract, bronchoalveolar lavage or sputum or salivation, or stool, GAITA PCR tests may be possible to detection. And after weeks, immunoglobulin M and immunoglobulin G antibodies detection is possible after the recovery period. In this period, the PCR likely is positive here, and especially symptomatic rays. Virus isolation, is it required or not? Virus isolation is required in a biosafety level 3 laboratory in advanced states. Virus isolation, virus culture is not recommended for diagnosis. When do we need virus isolation? Just on antiviral research on pathogenesis, virus stability research, neutralizing antibody test studies, or vaccine development studies. We do not use in daily practice. For PCR tests, the gold standard is nucleic acid amplification test, high sensitivity and specificity, and samples from upper and lower respiratory tracts are preferred. But in daily practice, most widely used real-time reverse transcriptase polymerase chain reaction. These are looking for the bronchoalveolar lavage, sputum, nasopharyngeal, oropharyngeal swabs. The exact location is important. Where can the sample locations are from the vestibule of the nose, anterior nares, mid-turbinate, nasopharyngeal place, and oropharyngeal anatomic place. Virus spread is important difficulty about the potential to spread. Do RNA positive individuals spread infectious virus? The virus replication stops 7 or 10 days after the onset of the symptoms. However, patients can be detected as RNA positive by PCR for 1 to 2 weeks after this point. This is the symptomatic days. The symptoms increasing, the symptoms is decreasing. These are the X line is the NAS and pharynx virus load. This is the infection starting, and this period after the symptoms ending, the PCR diagnostic window period. Should antibodies be tested after vaccination? Absolutely, it's no. The answer is no. It's not recommended to measure antibodies before and after vaccination in public vaccinations. Let's look at radiology. What's the place of radiology? Is it a gold standard diagnostic method or not? When do we need imaging methods? We are using radiology for pulmonary complication. First step is X-ray, and advanced stage is CT. It's X-ray, chest X-ray. It's easy accessible to practical and low radiation dose, but equals to low sensitivity, unfortunately. Don't forget, a normal chest radiograph, normal chest X-ray does not rule out the disease. X-ray is practical, portable, and also easy to disinfect, especially in hospitalization patients. Multifocal opacity with bilateral middle and lower region involvement in the patient's film can be considered diagnostic. Similar findings can be seen in other diseases. There may be atypical findings and further imaging may be required. The further imaging method is CT. MRI not is ready in this period. Most patients with COVID infection do not develop pneumonia. So CT or X-ray imaging is not necessary every time. In respiratory complications, we use diagnostic methods. Don't forget this. Imaging methods are not a diagnostic method in this disease, but an aid in diagnosis. First imaging tool, X-ray or CT. First, X-ray. the symptom is starting the first X-ray if there is a lung involvement. So in clinical incompatible, the advanced disease, we use CT. Let's compare them. The B line is X-ray, the A line is coronal multi-reformat CT in lung window imaging. And in B section, in early period of the disease, X-ray graphy is absolutely normal. You cannot tell anything about the disease. But on CT, in early period, same patient, same time, small ground glass areas are seen is visible and usually not reflected in X-ray as this image and are unlike to be seen. But in advanced stage, in the late period, the ground glass opacities and reticulations is visible on CT, coronary CT. Also in X-rays. Do we need, after this stage, do we need CT imaging? If clinical stable, we use X-ray for follow-up patients. Just X-ray, chest X-ray is enough for follow-up. CT is non-portable. It's not portable, especially in reanimation unit. You cannot use CT. You cannot transport it. X-rays to follow up, and CT has high dose, high radiation, and high risks. In the early stage, the sensitivity of chest radiography is low. Early CT scans are either normal or contain ground glass opacities. Let's look at the radiology terms. What is this ground glass density? What is the consolidations? The terms, let's clarify this. In ground glass density, if the increased opacity in the lung does not raise the underlying anatomical lung size, it's called ground glass density. But if the increased opacity erases the underlying signs and these signs are not visible, there is a superposition on both. It's called consolidation. In the later stage, especially in the periods when consolidation develop, findings begin to be seen in chest X-ray. This period starts about 4 or 6 days and is highest on 10 or 12 days. X-ray is useful to follow up in this stage. Don't forget. Radiologic findings are more prominent in chest radiographs on cases with moderate or severe clinical findings. This is the same patient follow-up graphs, follow-up X-ray imaging. First admission to hospital, and there is unclear opacities. On follow-up, say 8 days in reanimation unit, intubated patient, widespread consolidation is visible and it's developed. This is a typical COVID pneumonia patient, X-ray imaging, bilateral, middle and lower zones, mostly peripheral, increased densities, alveolar opacities, ranging from ground glass to consolidations. The difference between bacterial pneumonia and virus pneumonia. The bacterial pneumonia are usually characterized by consolidation in a single lung and a single lobe. Just on the left imaging, just single lobe and single lung. The virus pneumonia, especially COVID, are involving bilateral, the peripheral regions, mostly in the lower zones, middle and lower zones, and peripherally. That's the virus pneumonia. When do we need CT? In insufficient X-ray or normal X-ray but clinically advanced stages or uncertain X-ray, we apply CT imaging. And severe dyspnea, low oxygen saturation levels, we use CT. In this condition, guidelines recommend chest CT tomography. This also, don't forget, COVID infection disease is leads thromboembolic disease. The patient is possible for thromboembolism. So in dyspnea, and high D-dimers levels in blood suggests pulmonary embolism. And pulmonary embolism, we have to underwent patient contrast-enhanced CT imaging. In COVID pneumonia, it's not required in contrast material administration. But if you are suggesting a pulmonary embolia, you have to use contrast-enhanced imaging for diagnosis. This is A and this is B. This is enhanced thorax CT imaging. And this is main pulmonary trunk, ascending aorta, descending aorta, and right pulmonary artery. In the right pulmonary artery, there is a thromboembolism and filling defect that is visible just on the contrast-enhanced imagings. After the diagnosis, we follow up is also again X-ray. The differential diagnosis depends on other virus pneumonias. Atypical pneumonia agents can make similar findings. The differential diagnosis, aspiration pneumonia, pulmonary edema, and hemorrhage. Also organizing pneumonia and similar inflammatory pneumonias, drug reactions, and lung damage due to electronic cigarette use. Many events should be considered in the differential diagnosis. This is an disseminated infection in an intubated patient in a reanimation unit. On the left A image is the X-ray graphy, X-ray röntgenogram. And the B line is coronal reformatted CT imaging in the lung window, parenchymal window. Also additional findings. This is costophrenical sinus. This costophrenical sinus on the left side is normal and visible, but on the right side, it is blunting. It's not visible. So this equals to pleural effusion accompanying infection. And widespread parenchymal consolidations are also visible. Typical X-ray findings appear later than CT. And X-ray mostly provides sufficient information in follow-up hospitalized patients. And X-ray also widespread availability, easy to use, and reduces CT requirements and reduce contamination problems, and also is with reduced radiation dose. A structure also in CT imaging, there's CORAT's classification system. We use CORAT's classification system. What is CORAT's system? Imaging reporting data system. We are classifying in 5 degrees from minor to major. So, but it's like CT that has been suggested for X-ray, but there has not been a complete consensus. On CT image, the bilateral ground glass opacities is typical for COVID infection. Also, the other findings look at ground glass opacities and vascular enlargement is possible. The consolidation and air bronchogram with surrounding ground glass densities is visible. The diffuse cobblestone appearance and bronchiectasis is also visible. This in the high advanced stage we see like this, but in the early stage also subpleural ground glass opacities with pleural thickening also possible. This is the same patient, but in follow-up we use CT because this is the chronic stage. CT in after the disease, peripherally patchy ground glass opacities is visible. But 2 months later, follow-up, the fibrotic change and bronchial distortion and bronchiectasis and structural distortion and this chronic disease. Also, the other findings, subpleural curvilinear line and subpleural parenchymal bands. And normally, this air bronchogram and rounded by hollow sign. This hollow sign is the meaning this. Then the central patient, central component, there is a density, and the outer line, the peripheral rim, is radiolucent. But in reverse hollow sign, the central is lucent, the surrounding rim is dense. Round, oval ground glass opacities, and the bilateral you can see. And the reverse hollow signs, the bilateral reversed hollow signs is especially accompanying to the COVID-19 disease. And finally, take-home message. And CT has high sensitivity and plays an important role in early diagnosis in patients with high clinical suspicion and false negative PCR tests. We can apply CT. CT is also used in the evaluation of progression and response to treatments. Reverse transcriptase polymerase chain reaction PCR test is the standard reference diagnostic method. CT should not be used as a routine imaging method because it causes radiation, it's a high cost, and only in appropriate indications. The widespread and indiscriminate use of CT for unscientific reasons should be avoided. The molecular diagnostic methods are easily accessible, can give rapid results, and it's clear that there will be less need for imaging as their accuracy is high. Thank you very much.
Dear Professor Dr. Alptekin Tosun, thank you very much for your valuable contribution and for your nice presentation. Dear respected participants, do you have any questions to the presentation of Professor Dr. Alptekin Tosun? If you have questions, you can put them in the chat box, or if you would like to ask it directly, just go to the reaction button and we can open your voice. Also, if you are allowed, we can put Professor Doctor your email address in the chat box. The interested researchers may directly contact you for some possible questions and for scientific collaborations. Thank you very much. We are continuing now with the next presentation, which is going to be held by Professor Dr. Recep Keşli. He is from University of Health Sciences, Hamidiye International School of Medicine, and we have worked with Professor Dr. Recep Keşli also Afyonkarahisar University meanwhile together. Thank you very much for his acceptance for our offer for invited speaker. He is going to talk about human gut microbiota, gut brain axis, and medical conclusions. Dear Professor Dr. Recep Keşli, we are happy to listen to you. It is your turn.
Okay, thank you very much, Dr. Ajaros. I would like to thank all the audience and collaborators for organizing this symposium, and I will share my slides.
First, you have to open your presentation.
Yes. Okay. Full screen. In some situations, full screen
doesn't work.
Doesn't work.
You can.
Is it continue the slides? Is it continue?
No, it is the problem. I can explain you how to continue. Just make it in your computer full screen. It is full screen now in your computer.
No, full screen now. This is full screen.
You have to go upside. You have to go upside, and there is a green button. Just click on it. Did you find the green button on the upside of your presentation?
Upside.
You have to select it, and then you have to, and now it is full screen. Now it is full screen.
Okay. Are the slides continue now?
No.
Constant? Constant.
Hocam, üst tarafta yeşil bir düğme var. Tekrardan oradan onu seçin. Yeşil düğmeye basın. 2 tane poster sunumu gelecek karşınıza. Ha, şimdi oldu hocam. Tamam.
Okay. Okay. Good afternoon, everybody. I will present human gut microbiota gut-brain axis, and also medical conclusions, relationship between the disease and this communication, gut-brain axis. I would like to start with the basic knowledges, the genomics and transcriptomics, proteomics and metabolomics, the famous omics pathways. And when the DNA transcripted to RNA, and then protein synthesized, and then all the metabolic pathways concluded with different metabolomics and metabolites and end products. And quantity and quality of the protein synthesizing, also metabolic end products, is important. In fact, many of the diseases originated from the quality and quantity of the protein synthesized. Also, normal and abnormal end products. Macrobiota, eubiosis, dysbiosis, terminology, metagenome, metagenomics, transcriptomics, proteome, crosstalk, metabolomics. Now, in the normal and homeostasis, normal balanced position of the microbiota, termed as eubiosis, and density and diversity, two terminology important. Number of the bacteria located included in the metabolome important. Also strain diversity also important. A balance exists, normal position. If the balanced imbalanced position, named as dysbiosis. More than 1,000 or 2,000 strains included, and function and activity of the microbiota important. Also, composition means density and diversity together. Microbiota named as the second brain, and also a crosstalk or bidirectional communication exists between gut, bacteria, and the brain. Which one is managing our body, all the biological activities, by brain, or by gut, or by bacteria? In fact, gut means bacteria, and metabolic end pathways, and 4 mechanisms, 4 pathways exist for this bidirectional communication, crosstalk. Total DNA, approximately 6,000 belonging to viruses, and the other DNA total 93,000, including the bacterial DNA. So, gut microbiota, in fact, collecting all the strains, viruses, virobiota, and microbiota, but with quantity more than 90% of total DNA belonging to the bacteria. So this means that microbiota equal bacteria. Different functions exist of the microbiota: protective functions, metabolic functions, structural functions. And this is the normal situation. And 6 phyla important. Phyla, and the most important terms: phyla, name, and species. Actinobacteria phylum, Bifidobacterium strains originating from this phylum, and Firmicutes, including gram-positive bacteria. Clostridia, spore-forming anaerobic bacteria, important here, and Lactobacillus, also important, and Staphylococcus also included in this group. Bacteroidetes means gram-negative anaerobic bacteria, including Bacteroides fragilis, Vulgatus uniformis, Parabacteroidetes, Distasonis, and Prevotella strains also included in this group. Mainly Firmicutes, including gram-positive group, Bacteroidetes, including gram-negative groups. Proteobacteria, Desulfovibrio imported here, and also Bilophila wadsworthia, and Physobacteria, Physobacterium nucleatum, and Vericomicrobia, the last phylum, Akkermansia muciniphila, an important and famous bacteria. Sterility theory broken down. In the past, placental fluid and placenta semen obtained as sterile, but nowadays this sterility theory broken down, and useful bacteria including in this liquids, in this size of the body. Macrobiota, child's macrobiota, affected with intrauterine development period, also from mothers' different behaviors and social and physiological conditions, smoking, gestational metabolic abnormalities, and hygiene, antibiotic exposure, etc. In fact, macrobiota may be divided into 2 groups: changeable macrobiota and constant macrobiota. Changeable macrobiota affected composition is affected from environmental factors and changing, not static position. From especially intrauterine development is very important. Also genetic factors, diet and nutrition, exposure to antibiotics, etc. Many factors affecting the human macrobiota. Now, just a moment, please. Okay. Host is genetic, the most important factor, and then exercise, stress, antibiotics, age, diet, mode of delivery, vaginal action or sexual, geographic location also affecting the microbiota composition, and exposure to the chemical factors important, smoking, alcohol, diet, and these toxic factors directly changing composition and functions of the microbiota. Metabolic functions, immune system, and nutrient synthesis functions affecting, and many of the non-communicable diseases related with this composition changes, cancer development, neurologic diseases, diabetes, etc. In an animal study, after 2 days of antibiotic treatment, the mouse gut microbiota is altered and more susceptible to invasion of Clostridium difficile, causal agent of Pseudomonas enterocolitis, as you know. Unnecessary antibiotic use is very harmful. And microbiota also related with gut gut-brain barrier, and bowel-brain barrier. Two barriers exist, as you know, in the front of the brain. Each molecule shouldn't be reached to the brain. And bowel-brain barrier mainly maintained by TG proteins, tight junction proteins. As you know, cell-to-cell interaction, 4 different types of cell-to-cell interaction exist: tight junctions, adherence, distance, and gap junctions. And the tight junctions maintained by some proteins, TG proteins named as. Occludin is very important here. Occludin level should be raised. In this biases situation, occludin level is decreasing. And all of these factors, level of these TG proteins directly affecting maintaining of the integrity of the bowel-brain barrier. If the permeability increased and unwanted molecules such as peptidoglycans and LPS and toxins, etc., may be reached to the circulation and then also may be reached to the brain because blood-brain barrier also related with the gut-brain barrier. Intestinal microbiota, KLMAT, sophisticated intracellular, intercellular, and interkingdom signaling systems. This signaling system regulating intestinal barrier functions. Intestinal microbiota is playing a central role. Abiotic composition playing central role for regulating gut bowel-brain barrier, integrity of the bowel-brain barrier also, and unwanted, unnecessary molecules, harmful molecules may be reached after increased permeability of the bowel-brain barrier to the circulation. This is a high-risk position for developing unwanted many pathogenic mechanisms, pro-inflammatory cytokines activating, and then inflammation pathways activating, and then oxidative stress pathways activating. And many of the neurodevelopmental disorders such as autism spectrum disorder, or depression, directly has a relationship between pathogenesis of this disease and gut-brain axis and gut composition and microbiota composition. In fact, this period starting from intrauterine development, perinatal maternal dysbiosis definitely affecting the fetal newborn microbiota composition. At the intrauterine period, perinatal maternal dysbiosis concluding with blocking, inhibiting organogenesis, maternal immune activation occurring, and fetal brain development may be regressed. Also may be inhibited. Neuronal progenitor neural cells maturation and differentiation also negatively affected. In fact, perinatal maternal dysbiosis and immune activation, maternal immune activation against the proteins of the fetal brain proteins, concluding predisposing many serious diseases. Some kind of inflammatory mediators and cytokines aiding increasing the intestinal permeability, and the other ones, on the other hand, protecting intestinal tissue barrier functions. Nowadays, many of the diseases etiologic mechanisms proved and investigated with animal model studies and cell culture studies, human cell culture lines used. And also also studies performed with a child and adult human cases. And these groups of diseases, etiopathologist of these diseases related, proved with the originated from human gut microbiota and gut-brain axis. Metabolic disorders, obesity, diabetes, metabolic syndrome, functional gastrointestinal disorders, irritable bowel syndrome, ulcerative colitis, addiction, alcohol, and food dependence, neurodevelopmental disorder, autism, schizophrenia, neurodegenerative disorders, multiple sclerosis, Alzheimer's, Parkinson's, stress, depression, and anxiety. And nowadays, disorders and diseases included in this group. For example, COPD, acne vulgaris, skin cancers, lupus, etc. Also fibromyalgia included, and asthma, allergy, hypersensitivity reactions included in this group. Different abbreviations, different phrases I obtained from the articles: gut-brain axis, enteric nervous systems, enteric glial cells networks, gut-brain pathways, microbiome immune system, microbiome neuroendocrine system, intestinal gut and enteric neuropeptides. And this, I maintained this slide from titles of the articles. Gut microbiota, the conductor of the orchestra in the human. Human neuroendocrine communication. The host microbiota dialogue. Gut microbiota talk to the brain, language is enigma. When intimate friends turn into foes, etc. Hippocrates said in the past, all diseases begin in the gut. Microbiota composition, density, and diversity is very important, playing critical role for developing of the different kinds of the systemic diseases. How this crosstalk established? Up to now, 4 different pathways diagnosed, determined. Metabolic pathways, neuroendocrine pathways, neuronal pathways, and immunologic immune pathways. I will try to explain these pathways. Our knowledges, scientific investigations developing day by day. In the past, only enteric nervous system and only 2 neurotransmitters was diagnosed in the past with barrier function interaction. And then different neurotransmitters diagnosed and immune system also incorporated in this circle, in this reaction. Epithelium, immune system, mast cells, T and B cells, neutrophils, macrophages, and dendritic cells. And the microbiota is situation well diagnosed, well identified. Microbiota also means mainly bacteria directly affecting the epithelial functions, and then enteric nervous systems and enterochromaffin cell, hormone active cells of the gastrointestinal system, and glial cells incorporated in the circle system, and also stromal cells, fibroblasts also incorporated. And nowadays, this is the last identified situation here. Also, human gut microbiota epithelial barrier and the mechanisms directly affecting to the brain neurotransmitters and origin of the neurotransmitters, some essential amino acids, immune modulation system, and some metabolic end products of the, especially Clostridia and Bacteroideus strains, short-chain fatty acids, propionate, acetate, butyrate, propionic acid, acetic, and butyric acids. Butyric acids, all these 3 kinds of short-chain fatty acids helping protecting this barrier function, but especially butyric acid. But quantity of the short-chain fatty acids is also important. Excessive production of the short-chain fatty acids means that they enter into the circulation and reach into the brain. If entered to the circulation, stimulating and triggering strongly pro-inflammatory cytokines, TNF-alpha, IL-1 beta, and IL-6. 3 pro-inflammatory cytokines important here. And these cytokines activating inflammatory pathways, and also especially propionic acid entering to the blood circulation, and also triggering microglia activation. Microglia is a macrophage included mononuclear phagocytic system, a kind of macrophage protecting a member of the protective immune system in the brain, and also microglia cells activating on also as astrocytes activating. Astrocyte responsible for maintaining the blood-brain barrier, and astrogliosis concluding with very strong inflammation activation, and also neuroinflammation, myelitis, etc. Also, microglia responsible with synaptic purining. When the synapse is not purined by microglia cells, microglia is playing as an architecture of the brain, and also responsible with excitation and inhibition balance of the neuronal signal transmission from one neuron to the other neuron, also from coordination of the two hemispheres by corpus callosum. And also signal transmission is balance is broken down. Here, short-chain fatty acid, gut peptides, tryptophan metabolism, cytokines, corticosterone, lipoglycans, and peptoglycans affecting to the brain. And what this one's input for brain, and this ones are output, reaction of the brain, macroglia activation, neurogenesis affecting, monoamine turnover affecting, long-term potentiation. Long-term potentiation is a method for determining signal transmission system of between neurons. Maybe used as laboratory investigation. You can use as in vivo or in vitro. After sacrification of the animal models, sacrification of the brain, you can put into a conflict brain to Ringer lactate solution, and then you can assess the signal transmission, and also you can record long-term potentiation. Hypothalamo-pituitary adrenal axis activation and bowel-brain barrier integrity also affecting, and neurotrophic factors. Brain-derived neurotrophic factors, important key elements. Also, interactions with the gut microbiota, gut and brain axis, motility, autonomic neuronal system inputs, motility, secretion permeability microbiome from brain to the gut, but from gut connectome affecting immune neuronal and neuroendocrine systems. Stress is blocking and completely harmful. And hypothalamo-pituitary adrenal axis, and at the final situation, cortisol secreting from cerebral cortex, a stress hormone, and affecting all whole management of the whole body, immune system, and sensations. 3 pathways simulated in this figure. Neuronal pathways, also nervus vagus, an important at the neuronal pathway. Many different molecules may be reached from gut to the brain in the nervus vagus. Nervus vagus playing just a road for some molecules in order to reach to the brain from the gut, originating from the gut. Central nervous system and intestinal and microbiota is positions affecting each other. Also, factors influencing factor, diet, pathogens, inflammation, altered gut microbiota, concluding with different mechanisms and pathways. In this study, fibromyalgia, etiologic and etiopathogenic relationship established with sequencing of the gut microbiome profile, metagenomic analysis, 16S RNA, and in this table here, as you see, for example, this healthy control group, this fibromyalgia group, diversity of some strains of bifidobacteria absent in fibromyalgia. This means that the diversity, total population number of the bacteria strains decreased, maybe absent, because diversity should be protected. Very important. Strong evidences exist. Diversity and health. Strong evidences concluded from different studies. Bifidobacteria absent, and this bacteria Erythrobacterium strains absent. But for example, here a different strain exists. Rickenelsia alistepsis. This strain not exists in healthy control group. This means that gut composition directly affecting the triggering beginning of the pathogenic mechanisms of the diseases. This site is normal healthy position, and this site is dysbiotic position. As you see here, pro-inflammatory cytokines activating and post-stress hormone activating. Also, epigenetic and genetic mechanisms also activate, change it. Just as same as a circle, genetic, epigenetic, and environmental factors. These three factors important in the etiology of the many systemic diseases, especially diseases has genetic origin. Microbiota composition directly affecting genetic expression and also epigenetic activation of the epigenetic mechanisms. For example, in autism spectrum diseases, microbiota composition is different. In different countries, metagenome analysis established, and all the data may be maintained by scientists. How was the changes occurred in microbiota composition in autistic children? Actinobacteria lowering, Firmicutes lowering, Lactobacillus lowering, Clostridium increasing, especially in children, and Clostridium tetani increasing, Bacteroidetes increasing, etc. Microbiota also has a relationship with the synthesis relationship with the synthesis of the neurotransmitters. As you know, only a few essential amino acids playing as a source of the manufacturing of the neurotransmitters: tryptophan, serotonin, melatonin, and phenylalanine, tyrosine, L-dopa, dopamine, and norepinephrine and epinephrine. Here, I would like to emphasize some essential amino acids, source of the for manufacturing of the neurotransmitters. Also, microbiota functions. Gut microbiota also directly has a relationship between this kind of neurotransmitter production. Which bacteria has playing important role for production of the neurotransmitters? For example, GABA, Lactobacillus bifidobacterium, noradrenaline, Escherichia bacillus saccharomyces, serotonin, Candida streptococcus, dopamine, Bacillus, Lactobacillus acetylcholine. In fact, a newly identified newer neurotransmitters diagnosed, determined in the brain and also functions. I would like to emphasize this. We do not know today how was affecting gut microbiota and which kind of bacteria affecting this kind of production of neurotransmitters. This means that memory, behavior, and cognition functions directly or indirectly may be affected by microbiota compositions. Perhaps these relationships will be explained in the future investigations. Serotonin also important. Short-chain fatty acids important. Short-chain fatty acids maintained by two ways. The first one, complex carbohydrates such as inulin and pectin. This complex carbohydrate is not digested, only fermented by anaerobic bacteria in the gut. And end products all are short-chain fatty acids. The second way is short-chain fatty acids exist cell membrane of some strains of anaerobic bacteria, such as Bacteroides fragilis group and Clostridia. In the situation of overgrowth in these strains, also excessive production of the short-chain fatty acids will be concluded. The second way, this means that homeostasis of the also density of the microbiota are important. Short-chain fatty acids affecting peripheral immune system to modulate brain functions, and also immune modulators for these cells, neutrophils, dendritic cells, macrophages, monocytes, and T cells. Also, cell-to-cell interaction affecting has important roles with these functions of the body. Short-chain fatty acids, I mean, cell-to-cell interaction, neurotransmitter synthesis and releases, microglia activation, mitochondrial functions, and gene expression. Maternal gut, fetal brain axis. In fact, some congenital diseases, in fact, originated in the pathogenesis, started in the intrauterine period. Also, short-chain fatty acids regulates early neural system development. In neural cell culture investigations, human progenitor neuronal cell lines used and exposure to the short-chain fatty acids, propionic acid, with the control of unexposure to the propionic acid. In the conclusions, neuronal progenitor cell differentiation and maturation for developing normal neuronal cells blocked by exposing to short-chain fatty acids. I have a slide in the further presentation. Short-chain fatty acids also directly affecting gene expression and blood-brain barrier functions, and which bacteria responsible for production of which kind of short-chain fatty acids here. In short-chain fatty acids, I will show this slide. This slide is very, very important. This slide showing us transporter proteins. This transporter proteins transporting to the short chain fatty acids to the brain. And this slide showing us receptors of the short chain fatty acids. Which kind of short chain fatty acids binding to which kind of the receptor and reaching which parts of the brain. For example, in the past, these receptors named as coupling protein receptor. For example, GPR41. Nowadays, these receptors renamed as free fatty acid receptor 3. For example, free fatty acid receptors, receptor for acetate, propionate, and butyrate, and this receptor located in sympathetic ganglia, vagal, and dorsal root ganglia, and trigeminal ganglia. This means that, as you know, receptor determining tissue tropisms for many molecules. This means that this kind of short chain fatty acids easily can be reached these parts of the and cell components of the brain. As you see here, many receptors located in different parts of the brain and cells of the brain or neurons. This slide is very, very important because summarizing to us connection between the gut microbiome and the brain. The first part, immune pathways. The second part, endocrine pathways, vagal pathway, neuronal pathway, and other humoral pathways. Here, dietary fiber, probiotics and prebiotics, and changing intestinal microbiota composition, and short-chain fatty acids manufacturers as a metabolites from these metabolic actions. And these short-chain fatty acids also has a strong effect on regulation of these pathways. Here, as you see, and at the final outputs, motivation, mood, motor activity, depression, autism, schizophrenia, and learning and memory activities, depression and anxiety, stress, and hypothalamus pituitary adrenal axis activation activity. Food intake, learning and memory directly affected as a result of these 4 pathways. These 4 pathways also originated as a functions of short-chain fatty acids, or functions of the intestinal microbiota, or functions of the diet and nutrition. Diet and nutrition is very important, especially in these mechanisms. Anxiety and depression, stress, memory, learning, neurogenesis, and neuroprotection. Neuroprotective agents stimulating of the synthesis and manufacturing of the neuroprotective agents blocking, for example, brain-derived neurotropic factors. Also, gut-brain axis inflammation-associated diseases. Inflammation, in fact, a very important serious role in many of the diseases. For example, inflammation-associated disease and gut-brain interaction. Autism spectrum disorder, directly targeting to the brain, fetal brain, and neuroinflammation, massive and strong neuroinflammation occurring, and myelitis, and microglia activation, and astrocyte activation, astrogliosis, and also signal transmission is changing, and synaptic pruning is changing, and excitatory and inhibitory balance changing as a result of macroglia activation. Multiple sclerosis also, and stroke, and Parkinson, here macroglia activation Inflammatory bowel diseases, obesity, and here obesity, macrovector, and central nervous system inflammation. Here, systemic effects, intestine and brain, immune effects, immune pathways, and neuronal pathways, illustrated in this figure. This study we studied in the past and published as a summary. We investigated short-chain fatty acids by MSGC, mass spectrometric gas chromatography. We determined chromatograms in autistic children and compared with healthy control children. And we determined strongly statistically differences between healthy control group and autistic groups short-chain fatty acids excretion via urine. We collected urine samples and stored at deep freezers and studied with MSGC. Intracerebroventricular injection of propionic acids leading autistic behavior disorders in rats, male rats. We also performed a kind two studies with this method. I carried out 8 different animal models up to now with 3 model ways methods identified in the scientific publications. Also, I discovered a new method not identified and not published in a journal as an article or text in books, textbooks. A new model we we discovered and established. Okay, I'm summarizing because the duration is limited. Frequency of the autism in the US in some states higher than 4%, and economic aspects of the autism nowadays in United States, $95 billion for a year, and $2.4 million for a child during life if intellectual disability exists. If not exist, $1.4 million for during life. And microglia functions, microglia activated in autism, and synaptic pruning, etc., affected. Also, autoimmunity tendency increase in autistic children, and autoimmune antibodies detected from serum samples of newborns and amniotic fluid against these autoantibodies against different protein types exist in the brain. Some metabolites investigated, 59 metabolites investigated autistic children, and p-cresol found as higher level and GABA found as lower level. And also metagenome analysis with new generation, next generation sequencing methods, autistic children and mothers, healthy children and mothers, and compared poorly. Child to child, mother to mother, mother to child comparison exists in this study. And in this study, neuroinflammation, propionic acid induces gliosis and neuroinflammation, and human neuronal cell line cell culture study. And also this is another study, and this is animal study. Autistic children is got some fecal samples obtained, and microbiota composition transferred a female mus musculus mouses, and after gravity, after delivery, and all the offsprings showed autistic behavior disorders, and healthy control group established, and also fecal samples obtained from healthy children, and also microbiota composition obtained and transferred with oral gavage to the female mus musculus mouse, and then after gravity and pregnancy and birth, all the offsprings determined as healthy. We also carried out this study with normal microbiota of mouses, and also SPF mice, specific pathogen-free mice. We procached these mouses from Jackson Laboratories. This study, this last slide, this study carried out as post-mortem autobiopsies of the brain from humans. Autistic humans died, and then with special ethic approval maintained, and brain autopsy carried out. What are the results? CD4 T lymphocytes activated, CD20 B lymphocytes activated, CD8 cytotoxic T lymphocytes activated in the brain, CD4 T lymphocytes activated in the brain. Also, blood-brain barrier impaired, and microglia cells activated, and astrocytes activated. We also maintained all these results with the same method from animal models, sacrificed brain tissues. We investigated histopathologic staining methods with hematoxylin and eosin and NIL, two different paints, and also we carried out immunohistochemistry methods and searched different types of proteins. We also maintained the same results from the animal models' brain tissues. My slides is finished. I would like to thank for your patience. Thanks a lot.
Dear Professor Dr. Recep Keşli, it was a very interesting subject, and thank you very much for your presentation. If it is possible, can you share your email address in the chat box section? Maybe
Yes.
researchers can contact you for possible future collaborations.
Okay, thank you.
Thank you very much for your presence and presentation. And next slide, next presentation is going to be held by Professor Dr. Fatma Jülide Akbuğa. She is from Medipol University and Pharmacy Faculty. Her subject is about nucleic acid-based therapeutics. Dear Professor, we are happy to listen you.
Teşekkür ederim. Yalnız bende de aynı sorun oldu.
Hocam şöyle yapacağız. Öncesinde sunumunuzu açmış olmanız gerekiyor.
Açtım sunumum, yüklendi.
Şimdi şöyle bir şey yapacağız. Sonrasında ben kapatıyorum hocam. Şimdi o yeşil kısma basacaksınız aşağıdaki. Yeşil share screen ya da ekran paylaşımı kısmına basacaksınız.
Evet.
Orada karşınıza sizin PowerPoint sunumunuz gelecek. PowerPoint sunumunuzun üzerine tıklayın. Ekran paylaşımı geldi hocam. Şimdi aşağıda hocam 75 yazan bir yer var en aşağıda ya da 73. Onun yanında bir tane ikon var. Ona tıklayabilir misiniz? Evet, ona tıklayacağız hocam. Şimdi sizde tam ekran mı oldu, aynı mı duruyor hocam?
Evet.
Sizde tam ekran mı hocam?
Tamam. Görebiliyor musunuz?
İşte şimdi şey var, problem var. Onu çözmemiz lazım hocam. En üste gelin şimdi. O tam ekranın üst kısmına gelin hocam. Orada yeşil bir işaret var. Tekrar paylaş ya da reshare yazıyor. Yeşil. Bu sefer aşağıdaki buton yukarıya gitti.
Evet.
Ona tıklayın hocam. Şimdi karşınıza sunumunuzun 2 tane farklı şekli gelecek. Siz sunum olanı seçin hocam. PowerPoint olanı değil, sunum olanı seçin.
PowerPoint değil.
Sunum şeklinde olanı var. Ona basacaksınız hocam. Evet, geldi hocam. İsterseniz bir ileri geri yapın. Değişip değişmediğine bakalım.
Evet. Yapıyorum.
Değişiyor. Tamam. Süper hocam. Teşekkür ederiz.
Ben teşekkür ederim. Değerli Ulaş Hocam. Eğer sürede herhangi bir aşım olursa lütfen beni uyarın.
Tamam hocam.
Kongrenizi etkilemeyeyim. Ben size yeni demeyeyim de farklı bir ilaç grubundan bahsetmek istiyorum. Ancak konuşmama başlamadan önce 2016 yılında Nobel kazanan, fizyoloji ve tıp Nobel'ini kazanan Osumi'nin çok beğendiğim bir sözüyle konuşmama başlamak istiyorum. Hayat, proteinlerin senteziyle parçalanması arasındaki denge durumudur diyor Osumi. Gerçekten de doğru. Hani şimdi ilaçlar daha çok organizmanın temel molekülleri, pathway'ler üzerinden geliştirilmeye çalışılıyor. Yani eski görüş yerine, eski klasik görüş yerine yeni bir görüş geçerli oldu. Gerçekten proteinler temel moleküller. Çok eskiden beri kullanılan preparatlardı. Önce biyolojik temelli proteinler vardı. Bugün rekombinant DNA teknolojisiyle üretilen proteinler kullanılıyor. Ancak bunların formülasyonunda, hastaya takdiminde ciddi sorunlar da var. Bu nedenle uygulama bugün için parenteralle sınırlı kaldı. Oysa bazı durumlarda hastanın yaşam boyu çok sık kullanması gereken preparatlar. Dolayısıyla da ciddi sorunlar var. Sorunların en önemlisi de büyük molekül olması nedeniyle hücre membranlarından geçme sorunları var. Bunun hareketle şöyle bakıldı. Her zaman için dediğim gibi organizma çok iyi artık takip ediliyor. Yolaklara bakılıyor. Protein organizmada acaba üretilebilir mi? Proteinle ilgili bu sorunları gidermek mümkün mü dendi. Ve oradan hareketle de nükleik asit ilaçlarına ilgi başladı. Bunlar yeni bir grup ilaç. Yeni diyorsam aşağı yukarı 40 yıllık bir geçmişleri var. Ancak son 5 yıl pandemiyle de birlikte rönesanslarını yaşıyorlar. Yani fazla sayıda onay alan preparatları oldu. Biliyorsunuz mRNA aşılarıyla kullanımları genişledi. Ve giderekten ikinci jenerasyon teknolojisi üzerinde duruluyor. Çok geniş bir hastalık grubu var. Ancak bu hastalık grubu küçük moleküllerle tedavisinde zorlanılan hastalıklar, genetik hastalıklar, otoimmün hastalıklar, kanserin küçük moleküllerle ilaçla tedavisinde karşılaşılan ciddi sorunlar, kistik fibrozis gibi yaygın hastalıklar, değişik kan hemofilya gibi kan hastalıkları, nöromüsküler hastalıklar ve tabii AIDS. Son zamanlarda tabii buna bir de tedavide yeni ilacı çıktı. Doğuştan olan körlük durumu. Burada nükleik asitlerin tarihçesine bakarsak 47'den itibaren çalışmalar var. Ancak bir ara durgunluk yaşıyor sorunlar nedeniyle. Sonra giderek bir hızlanma gördük. Niçin bu ilaçlar giderekten önem kazanıyor? Çünkü organizmada biliyoruz gen ekspresyonunun proteinler, temel maddeler, bunları üreten de genler. O halde burada bir takım değişiklikler yaparak acaba tedavi edebilir miyiz hastayı? Bir diğer önemli avantajları spesifik moleküller, yani farklı bölgelere değil hedefleyebiliyoruz. Sürekli bir tedavi sağlıyorlar. Adeta hastaya tekrar ayağa kaldırabiliyorlar. Bu nedenle de hem genetik hastalıklarda hem de sonradan işte kazanılan kanser gibi hastalıklarda giderek ümit vaat ediyorlar. Çok fazla çalışmaya başlanılan ilaçlar. Farklı yönlerle tedavide kullanılıyorlar. Şöyle ki, geni inhibe edebiliyorlar, yani susturabiliyorlar. Gen ilave ederek, nükleik asitleri ilave ederek çalışmayan bir veya çalışmasında hata olan bir hatayı düzeltmek mümkün. Yine biraz önce de söylediğim gibi, daha sonra da açıklayacağım, düzeltmeler yapmak mümkün. Skipping dediğimiz teknolojilerle kesip çıkarmak mümkün. İlk tedavi daha önce başladı. 1970'lerde bir takım çalışmalar var ama ilk nükleik asitle tedavi 1990'da French Anderson tarafından bir bebeğe uygulandı. Adenozin deaminaz enzimi eksikliği olan bir bebeğe uygulandı. Ex vivo bir uygulama, yani dışarıya alındı. Burada nükleik asit tedavisi yapılıp retroviralle tekrar bebeğe verildi. Şimdi bilmiyorum, görebiliyor musunuz? Bu yaşam 1990'dan sonra bebek ayağa kalktı, büyüdü ve şu anda da ergen normal yaşamına devam ediyor. Şimdi madem ki bu kadar önemli moleküller diyoruz, yapısı bilinen bir yapı, hidrofilik bir özelliği var. Bu özel önemli. Bir de negatif yükü var. Bu da formülasyon açısından, ilaç oluşturmak açısından önemli. Her ikisi de, yani DNA'da, RNA'da nükleotitlerden yapılmış bir 5 karbonlu şeker yapısı var. Buna bağlı fosfatlar var. Bir de bir seri purin ve pirimidin bazları var. Burada nükleik asidi, dezoksiribonükleik asit ve bildiğimiz RNA'yı görüyoruz. Yapıları çiftlikler. Hemen hemen bazıları ortak, yalnız biri timin taşıyor, biri urasil taşıyor. Bir de şeker yapısında çok ufak farklılıklar var. Ama ne olursa olsun her zaman karşımıza bu sistemi aldığımız zaman neler yapabiliriz diye bakıyoruz. Bir kere DNA'yı kullanabiliriz protein üretimi için. RNA'yı kullanabiliriz protein üretimi için. Transkripsiyonu durdurabiliriz. Bu da önemli. Translasyonu durdururuz, protein üretimini durdurabiliriz. Özellikle kanserde bu son derece önemli. Bir de son zamanlarda geliştirilen aptamerler var. Büyük ümit vaat eden monoklonal antikorların yerini alacağını söylediğimiz yapılar var. Bunlar küçük, tek iplikli yapılar, ancak spesifik olarak DNA'ya bağlanıyor, RNA'ya bağlanıyor, proteinlere bağlanıyor, küçük moleküllü ilaçlara bağlanıyor. Hatta toksinlere bile bağlanabiliyor. Tek bir sorunları var, farklı bir teknolojiyle üretiliyorlar. Bu oligonükleotitler, Selex teknolojisi, pahalı bir teknoloji. Bu aşıldığı takdirde aptamerler bugünkü monoklonal antikorların da yerini alabilecek. Şimdi şu şekilden bakarsak proteini üretebiliriz veya üretimini durdurabiliriz veya bir hedefleme yapabiliriz. Peki avantajı ne bu sistem? Biliyoruz biz biyolojik fonksiyonları, hücredeki biyolojik fonksiyonları yöneten sistemler, kontrol eden sistemler. Burada esas olan nükleotidin sekansı, bilgiyi veren nükleotidin sekansı. Bunun dizaynı önemli. Dizayn yaparak, nükleotit sekans dizaynı yaparak değişiklikler yapabiliyoruz. Değiştiriyoruz, yeni hedef genler oluşturabiliyoruz. Bu bize ne kazandırıyor? Bu bize çok önemli bir avantaj sağlıyor, yarar sağlıyor. Şöyle ki, ilaç geliştirme normalde küçük bir molekül ilacı geliştirmeniz için en az 15 hatta 20 yıl çalışmanız gerekir. Bunun sonunda da başarılı olmayabilirsiniz. Oysa bu grup ilaçlarda bu sekans değişiminden yararlanılarak çok hızlı ilaç geliştirmek mümkün. Gördük mü? Evet, bunu gördük. Son COVID-19 pandemisinde çok hızlı mRNA aşısı geliştirildi. Aşağı yukarı SARS-CoV-2'nin genom sekansının bulunmasından 2 ay sonra önemli bir gelişme oldu. Aynı şekilde doğuştan olan körlük durumda bebeklerde de kullanılan ilaç nükleik asitler çok kısa sürede geliştirildi ve çok başarılı bir şekilde bebeklere uygulanıyor. Dolayısıyla ilaç geliştirmenin hızlanması açısından son derece önemliler. Bir diğer avantaj, biraz önce de aynı zamanda proteinin üretimini durdurabiliriz, geni susturabiliriz. Bu da önemli bir avantaj. Özellikle kanser için, kanser tedavisi için son derece önemli bir avantaj. Yine son zamanlarda kodlamayan RNA'lar üzerinde çok duruldu ve bunlardan ilaç geliştirildi. Yani protein üretmeyen RNA'lar eskiden nedir bunun fonksiyonu diye üzerinde duruluyordu. Şimdi çok güzel bu RNA'lar kullanılıyor. Bu şekilde pek çok biyolojik proseste işlem gören bu proteinlerin susturulması veya çalıştırılması bugün için söz konusu. Bir diğer mekanizma, biraz önce de söylediğim splicing dediğimiz mekanizma. Özellikle birden fazla mutasyon olan genetik hastalıklarda tabii tek bir mutasyon olsa tedavi edebiliyorsunuz ama birden fazla olduğu zaman o zaman splicing profilinden hareket ederek biraz sonra tekrar çok kısa bahsedeceğim. Mutant olan kısmı kesip çıkarıp iki ucu bağlamak bugün için mümkün. İlaç, bu konuda da geliştirilmiş ilaç var. Peki sorunları ne? Tabii sorunları proteinlerde olduğu gibi bunlar da, onlar proteinazda, bunlar da nükleaz enzimle degrade oluyorlar, parçalanıyorlar. Bunun yanında immün aktivasyon söz konusu olabiliyor. Fiziko-kimyasal özelliklerine bağlı bir takım değişiklikler oluyor ve tabii hücre tarafından alımlarında sorun var. Bu şekilde özellikle bu sorunları görebiliyoruz. Proteinle etkileşmesi, nükleazlar tarafından parçalanması, res tarafından alınıp uzaklaştırılması, renal clearance'ın çok hızlı olup hemen kanı terk etmesi. Bu da yetmiyor. Bir de hücre içinde önemli sorunu var. Bir kere hücreye girişi birinci sorun. İkinci sorun endozomdan kısa sürede çıkması lazım. Biz buna endozomal kaçış diyoruz. Eğer bu gerçekleşmezse tabii lizozoma gidip parçalanabilir. Dolayısıyla ilacın hiçbir etkisi olmaz. Aynı şeyi bu iki durumda da görüyoruz. Ekstraselüler ve intraselüler bariyerler, sistemik kullanıldığı zaman nükleazlar, endotelyal hücrelerin çevresindeki yapılar, plazmadaki, restteki atılma, Ve hücreye girdiği zaman da karşılaştığı ciddi sorunlar, çok erken bir şekilde endozomu terk etmesi gerekiyor. Peki bu sorunlara yaklaşım nedir? Çözümler var mıdır? Evet, bu biraz önce saydığım sorunlarda bugün iki çözüm var. Birincisi, nükleik asit yapısında kimyasal modifikasyon yapabiliriz. İkincisi de uygun bir taşıyıcı sistemle taşıyabiliriz. Burada gördüğünüz gibi çıplak bir nükleik asit, kimyasal modifikasyon yapabiliriz, konjugasyon yapabiliriz ve değişik taşıyıcılarla, biraz sonra bahsedeceğim, özellikle katyonik olanları tercih edilir. Polimerik, lipid katyonlar, siklodekstrinler ve bir de çok önemli eksozomlar bu sorunun çözümünde önemli olacaklar. Kimyasal modifikasyon dediğimiz zaman iskelette modifikasyon yapılıyor, şekerde modifikasyon yapılıyor, bazılarda modifikasyon yapılıyor. Tabii bir de konjugasyon yapılabiliyor. Bu şekilde değişiklikler ne kazandırıyor? Yeni bir özellik kazandırıyor oligonükleotide. Enzime karşı daha dayanıklı hale geliyor. Yani daha stabil hale geliyor ve hedefe bağlanma afinitesi de artabiliyor. Bu değişiklikler bazen belli tek olabiliyor veya kombine oluyor. Biraz sonra göstereceğim. Birden fazla grupta değişiklik yapabilirsiniz. İlk jenerasyonlar fosfatiyatlar dediğimiz fosfat iskeletlerinde olan modifikasyonlar ki bunlar artık çok yaygın kullanılıyor. İlaç halinde de kullanılan ilk jenerasyon oligonükleotitler. Endonükleazlara dayanıklılar, afiniteleri, renal clearance biraz geciktirilerek biyoyararlanım arttırıldı. Afiniteleri de arttı. Bu açıdan da önemliler. Ancak hedef RNA'ya olan afinitelerinde azalma görüldü. Bu nedenle de çalışmalar devam etti. İkinci jenerasyon geliştirildi. İkinci jenerasyon riboz modifikasyonuydu. Bu şekilde hem afinitesi hem hibridizasyonu, hedef mRNA'ya olan hibridizasyon stabilitesi arttırıldı. Stabilitede iyileşmeler elde edildi. Yalnız bunlarda toksik etki sorunu ortaya çıktı. Üçüncü jenerasyona geçildi bu nedenle. Burada tam bir değişiklik yapıldı. Şeker grubunda değişiklikler, fosfat bağlarında, nükleotitlerde değişik bağlantılar, değişiklikler yapılarak üçüncü jenerasyon geliştirildi. Ne kazandırdı üçüncü jenerasyon? Stabiliteyi çok arttırdı. Etkinliği açısından. İkincisi enzimlere karşı enzim degradasyonuna karşı direncini arttırdı molekülün. Yani bir anlamda enzim stabilitesini sağladı. mRNA'ya hibridizasyon afinitesinde de artış görüldü. Bu grupta LAK nükleik asitler var, LNA'lar veya peptit nükleik asitler, PNA'lar ve çok üzerinde durulan morfolinofosforamidler var. Gerçekten kullanılan iyi oligonükleotitler. Burada kısaca formüllerini verdim. Birinci nesil değişiklikleri görüyoruz. İkinci nesilde fosfat yapısında veya şekerdeki değişiklikler. Üçüncü nesilde kilitli nükleik asitler, morfolinolar, farklı değişiklikler yapılmış ve peptit bağlanmış, peptit nükleik asitler. Tabii bu da bunun dışında da bir takım değişiklikler yapılıyor. Örneğin pek çok yerde pek çok yapısı adeta değişikliğe uğrayan oligonükleotit sistemleri de var. Şöyle demiştik hatırlarsanız biraz önce, iki yaklaşım var bu sorunları çözmede. Biri kimyasal modifikasyon. Yalnız bunun getirisi şöyle oldu. Hani bazı gruplarda toksitede artma görüldü. İkinci yaklaşım taşıyıcı sistemlerle nükleik asitleri taşımak. Nükleik asitlerin taşınmasında gerçekten taşıyıcı sistemler önemli bir avantaj sağladılar. Burada taşıyıcı sistemlerin özellikleri son derece önemli nükleik asidin etkinliği açısından. Nükleik asitlerin özellikle kendilerinden bağımsız olarak taşıyıcının önemli özelliği var. Burada formülün dizaynı önemli. Yani iyi dizayn etmediğiniz bir formülde istediğiniz sonucu alamıyorsunuz. Bunun yanında bir de multifonksiyonel taşıyıcı sistemler geliştirildi. Biraz sonra göstereceğim. Yani hem hedefliyorsunuz, hem farklı grupları bağlıyorsunuz, kaplıyorsunuz. Değişik sorunlara aynı anda çözmeniz mümkün. Bu taşıyıcı sistemler geliştirilirken farklı amaçlar önemlidir. Nedir bu amaçlar? Her şeyden önce oligonükleotidi bu hemen olabilecek degradasyondan korumak, enzim degradasyonundan korumak. Hem dışarıda ilaç geliştirirken çok dikkat etmek lazım. Hem de organizmaya verdiğiniz zaman bu ön parçalanmalardan korunması lazım. Diğer önemli özellik etki süresini uzatmak lazım. Etki süresi önemli, yani çok kısa etkili bir bazı durumlarda kısa etki istenebilir. Bazı durumlarda ise etkinin çok uzun sürmesi istenir. Yani artık hasta tamamen normal hale dönmesi istenir. Bir diğer önemli husus hedefleme. Hedefleme son derece önemli. Hedeflemenin de mümkün olduğu kadar rahat bir şekilde yapılması istenir. Şimdi bu istekleri karşılayan taşıyıcı sistemleri biz iki başlık altında topluyoruz. Bir grup viral taşıyıcılar ki ilk nükleik asit uygulamaları, ilaçları bu viral taşıyıcılarla oldu. İkinci başlık ise non-viral dediğimiz viral olmayan taşıyıcılar. Bunların bir kısmı kimyasal dediğimiz taşıyıcı sistemler. İlacı nasıl taşıyorsanız bu sistemi de bu şekilde taşıyabilirsiniz. Diğeri de fiziksel sistemler, yani bir anlamda hücre membranından geçemeyecek kadar büyük olan bu molekülleri dışarıdan kuvvet uygulayarak membranda ufak delikler açarak hücre içine girmesini sağlamak. Bu ilaçların uygulanmasında iki yöntem var. Biri in vivo olarak, yani enjeksiyon yoluyla uygulayabiliyoruz nükleik asitleri. Intravenöz, subkutan uygulamalar var, intratekal uygulamalar var. Intravitreal, özellikle göze uygulanan preparatlar var, körlük tedavisinde. Intraretinal preparatlar var ve bu şekilde çalışmayan bir geni fonksiyonunu sağlamak, susturmak veya düzenlemek mümkün. Yani hastalıkların tedavisinde özetle bu şekilde gösterebiliriz. Taşıyıcı sistemler, ki bunların içinde partiküler sistemler diyoruz. Bu partiküler sistemlerin katyonik yapıda olması tercih ediliyor. Çünkü hücre yüzeyi biliyorsunuz negatif. Nükleik asitlerden negatif olduğu için bu da ayrı bir bariyer teşkil ediyor. Dolayısıyla katyonik yapıda olanların tercihi söz konusu. Bu şekilde hem hücre membranından kolaylıkla geçebiliyorlar. İntraselüler trafik dediğimiz endozomu terk etme çok daha hızlı ve düzenli bir şekilde yürüyor. Bu şekilde dozda da azalma sağlayabiliriz. Niçin? Tam hedefe ulaştığı için bu tip ilaçlarda çok rastlanan hedef dışı etkiler minimize olacaktır. Aynı şekilde toksik etki de minimize olacaktır. Ve bu dozun tümüyle verilen nükleik asit dozunun tümüyle hedefe ulaşmasını, farmakolojik hedefe ulaşmasını sağlayacaktır. Diyebiliriz. İlacın terapötik indeksinde de iyileşme sağlanacak. Burada virüs sistemlerini göstermeye çalıştım. Lentivirüsler, adenovirüsler, adeno-asosiye virüsler. Bir de herpes simplex vardı. Burada gösteremedik. Lentivirüs bir retrovirüs ailesidir. AIDS virüsüdür. Cidden çok ilginç şeyler yapılıyor. Örneğin çok patojenik olan bir virüs bir ilacı taşımada kullanılabiliyor. Bu da tabii bilimin önemli gelişmelerinden biri. Şimdi kimyasal sistemlere bakarsak katyonik lipidler var, katyonik polimerler var, peptitler çok önem kazandı, proteinler var. Bunlarla ne yapabiliyoruz? Değişik lipopleks dediğimiz, bakın çok farklı şekilde taşıyıcı sistem geliştirebiliyoruz. Polimerlerle, başta kitozan olmak üzere polimerlerle polipleksler geliştiriyoruz. Bunlar da pozitif gördüğünüz gibi ve yine peptit proteinlerle suni virüse benzer yapılar oluşturuyoruz. Virüsün hücreye girişte kullandığı proteinlerden yararlanıyoruz. Bunlara da suni virüsler hazırlanabiliyor. Bugünkü yaklaşım daha çok hibrit yaklaşım. Yani her taşıyıcı sistemin iyi yönlerini alıp bir araya getirip bir hibrit hazırlamak. Bunu sonda gösterecektim ama burada da göstereyim. Son zamanlarda egzozomlardan çok yararlanılıyor. Egzozomları tanıyoruz. Hücreler arası bilgi alışverişinde kullanılan çok küçük yapılar, veziküler yapılar. Acaba bu veziküler yapılara şu anda kendi mirnalarını taşıyorlar ama dışarıdan bir nükleik asit ilavesi mümkün mü? Bu konuda iyi çalışmalar var ve egzozomlara geleceğin taşıyıcısı olarak bakılıyor. Tekrar bakarsak adenovirüsler, adeno-asosiye virüsler, retroviral vektörler ki bunların içinde antiviral vektörler var. Çok iyi bir taşıma sağlıyor, yüksek bir etkinlik sağlıyor. Herpes simplex var. Herpes simplex önemli, özellikle beyine ilaç taşımakta, nörodejeneratif hastalıklarda nükleik asitlerle tedavisinde iyi bir taşıyıcı olduğunu biliyoruz. Pax virüsler de son zamanlarda kullanılmaya başlandı. Viral olmayanlar için de katyonik lipidler, biraz önce bahsettim, katyonik polimerler, mikro nanopartiküller, inorganik moleküller var. Fiziksel yöntemler içinde biyolojik yöntemler bir altın partikülün üstünü kaplayıp tabancayla vermek, helyumla vermek, yani bir basınç uygulayarak membranda delik açıp oradan girişini sağlamak, jet enjeksiyonlar. Ultrason var. Bunlar içinde elektroporasyon, özellikle iyontoforetik sistemler hem küçük ilaçlar için hem proteinler için hem de nükleik asit ilaçları için çok üzerinde durulan sistemler, yani akım uygulayarak küçük porlar açıp oradan hücreye girişi sağlamak. Bunun dışında hidrodinamik enjeksiyon hayvanlarda iyi sonuç veriyor ama insanlar için bilemeyeceğim, biraz şüpheli diyorum. Şimdi burada bir taşıyıcı sistemi görüyoruz. Kargosu çok değişik kargoları olabilir. Bu kargo içinde kompleksleri de yerleştirebilirsiniz. Lipidlerle zengin yapılar oluşturabilirsiniz. Özellikle katı lipid partiküller çok kullanılıyor bu amaçla. Bu yapıya değişik fonksiyonlar kazandırabilirsiniz. Fonksiyonel gruplar ekleyebilirsiniz. Örneğin kandaki kalın süresini uzatmak için ekleyebilirsiniz. Değişik şeker lipid konjugatları ekleyebilirsiniz. Hedefleme için hedefleme molekülleri ekleyebilirsiniz. Özellikle kanserde tümör yüzey reseptörlerine değişik hedeflemeler bu amaçla yapılıyor. Nükleik asitlerin tip ve karakteristikleri üzerinde çok kısa durursak, fonksiyonlarına bakarak çok farklı nükleik asit var. Plazmik DNA ilk kullanılan, ondan sonra mRNA var, mikroRNA var, cRNA var, ribozimler, antisens oligonükleotitler, aptamerler ve bu sırayı daha da geliştirirsiniz. Çünkü çok hızla gelişen bir sektör haline aldı. Neler oluyordu? Gen replacement yapabilirsiniz. Yani yerine koyabilirsiniz. Bir mutant genden dolayı protein üretilemiyordur. Hastalık ortaya çıkmıştır. Burada şöyle bir ifade var. Doğru proteinin doğru yerde, doğru zamanda üretilmesi lazım. Eğer üretilmezse bir patolojik durum ortaya çıkıyor. O zaman bunu gerçekleştirebilecek fonksiyonel bir geni ekleyerek hastalığı tedavi edebilirsiniz. Biraz önce ilk gen tedavisinde gördüğünüz gibi. Bir diğer durum, gene eklemeler yapabilirsiniz. Örneğin yine bir hastalık durumu vardır, genetik faktörlerden kaynaklanan. Burada bir geni ilave ederek, yani hiç protein ekspresyonu yoktur, hastaya tedavi edebilirsiniz. Geni susturabilirsiniz. Yine mutant bir gen vardır. Yalnız ürettiği protein ya gerekli zamanda üretmiyordur, ya çok üretiyordur, ya yanlış üretiyordur ve bir hastalığa neden olmuştur. Bu nedenle bir gen susturucu ilave edersiniz. Gen susturucu molekülle, sirnalar, mirnalar, antisens oligonükleotitlerle düzeltebilirsiniz. Hasta sağlığına kavuşabilir. Bir de son zamanlarda çok gelişen gen editing dediğimiz, bu bir tamir. Bu hemen hemen nükleik asitlerde bir çığır açtı. Şöyle ki eskiden bu hatalı genin yanına yeni geni ekleyip çalıştırıyordunuz. Şimdi değiştirmek mümkün. Yani onu çıkarmak, onun yerine doğru geni yerleştirmek mümkün. Hücre tamir mekanizmalarından yararlanılarak bu teknikte gelişti. Bu konuda biraz sonra tekrar göstereceğim. Moleküllere kısaca tekrar hatırlarsak RNA'lar var, antisens oligonükleotitler var. Bunlar daha çok RNA'ya komplement olan sistemler, bu şekilde RNA'nın yarılmasını, çalışmasını engelleyen sistemler. Bu şekilde de hastalığa neden olan genlerin ekspresyonunu engelliyorlar. RNA dediğimiz zaman bugün çok sayıda RNA molekülü var ilaç olarak kullanılan. Antisens oligolar var. RNA'nın temel molekülü sirnalar var. Mirnalar var. Aktive RNA'lar var, small aktive RNA'lar var, ribozimler var, aptamerler var ve guide RNA'lar ki her ikisi de demin bahsettiğim bu genin editing sistemlerinde kullanılıyorlar. Bunu şunun için gösterdim bu tabloyu. Bunların etki yerleri farklı. Özellikle mRNA aşılarında bazı yanlış şeyler söylendi. İşte hücreye giriyor, hücrenin yapısını değiştirir vesaire. Hayır, mRNA dediğiniz zaman nükleüse girmez. Sitoplazmada etki eder. Sirna da öyledir. Sirnanın hedefi mRNA'dır. mRNA'yı parçalar, sitoplazmada etki eder. MikroRNA'lar yine hedefleri kendi bozuk olan mikroRNA'lardır. Sitoplazmada etki ederler. MikroRNA'ların yerini alırlar. Antisensler hem mRNA'da hem mRNA'larda etkili olurlar. Bunlar da sitoplazmada, hücre içinde, sitoplazmada nükleus için çok farklı bir yapıyı düşünmemiz lazım. mRNA ve mikroRNA'ları parçalarlar. Bir diğer önemli özellikleri splicing'i inhibe ederler. Aptamer yeni bir yapı demiştim. Hücre dışında tamamen etki ediyor, proteinlere etki ediyor, bağlıyor. Biraz önceki şekilde görmüştük. Tedaviyi sağlıyor. Proteinin fonksiyonel inhibisyonunu sağlıyor. DNA ilk kullanılan yapı. Tamamen proteini hedefli, nükleoste etki ediyor, transkripsiyonu inhibe ediyor. Ribozimler RNA'dan kaynaklanıyor. Etkilisi RNA'nın yarılmasını yapıyor. Hücre içinde sitoplazmada etkililer. Bir diğer pek şu anda aşıları konuşmadığımız için konumuz olmasa da gittikçe çok kullanılan CpG'ler var. Bunlar da kısa tek zincirli sentetik DNA'lar. Farkları ne? Sitozin, guanin arasında bir trifosfat deoksinükleotit dizisi içeriyorlar. Ne sağlıyor bu dizi? Bu dizi immün sistemin TLR reseptörünü uyarıyor ve bu şekilde çok ciddi bir immün potansı var. Yani immün yanıtı arttırıyor. Aşılarda antijenin yanında kullanılabilen. Burada özetlemeye çalıştım. DNA'dan transkripsiyon sonucu ne olabilir? mRNA'dan protein üretimi olur. Bu normal işleyiş mekanizması. Transkripsiyondan sonra ribozomal değişik enzimlerle mRNA'nın parçalanması ve üretimin durdurulması. Veya antisenslerle burada gördüğümüz gibi mRNA'ya bağlanarak RNA'yı parçalıyor ve yine blokaj oluşturuyor. Daha sonra sirna'da da aynı şekilde risk kompleksine bağlanıp mRNA'yı parçalayarak translasyonu bloke ediyor. Tekrar vermemin nedeni şu, bakın şekillerinde farklılıklar var. Her birinin şeklinde farklılık var. Etki mekanizmalarında da bazılarının daha farklı bir etki mekanizmasına sahipler. Splicing'den bahsetmiştim. Önemli çünkü bu pek çok genetik hastalığa rastlıyoruz ve böyle hastalıklı çocuklar belli bir süre sonra yaşamlarını kaybediyorlar. Düşen var, düşen musküler distrofisi var. Yaşamları çok kısa, bir süre sonra hayatlarını kaybediyorlar. Buradaki sorunlar biraz önce de söyledim, birden fazla mutasyonlar var. Bu mutasyonlarda splicing mekanizmasını kullanıyoruz. Eksonları kesip çıkarıyoruz. Şurada gördüğünüz gibi 49 numaralı eksonu kesip çıkarıp 48'i 50 ile bağlıyoruz ve bu şekilde de sağlığına kavuşabiliyor. Hasta sağlığına kavuşabiliyor. Bu tabii tek bir mutasyon. Bunun farklı tipleri var. Farklı uygulama şekilleri var ve geliştirilmiş ilaçlar var. Onay alan nükleik asitler, burada özellikle üzerinde durmak istediğim, bakın retinitte var, değişik kanser tiplerinde geliştirilmiş, onay almış ürünler var. Arteriosklerotik periferal arteriyel hastalıklar var, değişik ailesel hastalıklar, yani genetik hastalıklar da var. Düşende, demin söylediğim gibi düşen musküler distrofisi var, yine melanomalar, değişik immün hastalıklar, immün yetersizlikleri son zamanlarda çok geçen SMA diye de kullanılan ilaçlar var. Birden fazla ilaç var hatta bu konuda. Değişik retinal hastalıklar var. Şu anda 21 tane ilaç FDA ve EMA'dan onay almış durumda. Bunun dışında bir de CAR T-cell terapileri var. Özellikle kan kanserinde, lenfomada kullanılan immün sistemde yapılan bir takım değişiklikler var ki bunlar içinde CAR T-cell tedavileri, lentivirüsler, retroviral vektörlerle taşınan, onay almış ürünler var. Sözlerimi biliyorsunuz Nobel kazanmış bir Japon bilim insanının konuşmalarıyla başlamıştım. Bitirirken de yine 2020 yılında Nobel ödülü kazanan 2 bilim insanıyla bitirmek istiyorum. Çünkü hakikaten bundan sonraki ilaçlar bu 2 kişinin buluşlarından hareketle hazırlanıyor. Birisi Dutna, diğeri Fransız Charpentier, biri Almanya'da çalışıyor, diğeri Amerika'da çalışıyor. Biraz önce söyledim ama nükleik asit değil de nükleazlar olduğu için konuşmamın içeriğini almadım ama çok önemli ve yarının teknolojisi bu genom düzenlenmesi teknolojisi. Yarın neler olacak? Gelecekte neler bekleyeceğiz? Ciddi gelişmeler var. Hakikaten yakalaması çok zor. Değişik biyoanalitik yöntemler var. Genom sequencing'de ciddi gelişmeler, hızlanmalar var. Manipülasyon teknolojileri var. Demin ifade ettiğim splicing'e switch de deniyor. Yani açıp kapatabiliyorsunuz eksonları çıkarıp koyarak. Taşıyıcı sistemlerde gelişmeler bekliyoruz. Tabii bunlar içinde aşılar var. Kanser immünoterapisi önem kazandı kanserin tedavisinde. Biraz önce söylediğim gen edisyonu da bunlardan biri. Yalnız yol çok uzun ve çok zahmetli bir yol. Zirveye ulaşmak için henüz başlangıcın sonundayız. 2. kademeye geçtik. Bilim insanlarına çok iş düşüyor. Bu nedenle de çok çalışmak lazım. Konuşmamı bitirmeden önce ilginiz için teşekkür ederim. Ayrıca özellikle Ulaş Hoca'ya ve organizasyon komitesine çok farklı bir grupla bir arada olma şansı verdikleri için ayrıyeten teşekkür ederim.
Hocam biz kıymetli vaktinizden ayırıp tecrübelerinizi aktardığınız için çok teşekkür ederiz. Dear respected Professor Dr. Jülde Aktuba, thank you very much for your presentation. It was nice to listen you. If it is possible, can you write also your email address in the chat box? Maybe interested researchers may contact with you for possible research activities. Thank you very much again. And the next presentation is going to be held Professor Dr. Kadir Öymen Hançerlioğlu from Giresun Medical Faculty. He is a pediatric surgeon, and his subject is about the approach to common pediatric surgery problems. Dear Professor Dr. Kadir Öymen Hançerlioğlu, thank you very much for accepting our offer. It is nice to listen you.
Thank you very much, Dr. Acarız. Hello to all participants, researchers, and presenters. I hope you are all fine. First of all, in this Congress, the participants are more than from 50 countries, and it's very important success. First of all, I want to congratulate you and your team, Dr. Ulaş. I am Associate Professor Dr. Kadir Öymen Hançerlioğlu. I am working at Giresun University, Faculty of Medicine, Department of Pediatric Surgery. Now I can start my presentation. In this presentation, I want to introduce pediatric surgery. Because of the time constraint, I will give a summary about pediatric surgery. What does pediatric surgeons do? Which diseases commonly seen in pediatric surgery? And lastly, I want to talk about inguinal hernias, which is mostly seen in pediatric surgery clinics. And what about its approach? Okay.
It is full screen now. Just try to change the slides, dear professor.
Full screen'de mi hocam?
Evet hocam. İleri geri bir denerseniz.
Şöyle.
Yeah, everything's fine.
Okay. In pediatric surgery, we are responsible for the treatment and prevention of surgical conditions in especially fetus 25 weeks of gestation to adolescent at puberty. In pediatric surgery established in about 1940s. What about pediatric surgery specialities? We look general surgery. It's important. Then the next one, neonat surgery, orthopedics, urology, thoracic surgeries and cardiac surgeries, neurosurgery, and also consists oncology. What about when we look in pediatric surgery, we saw a lot of congenital abnormalities. When we give some examples, especially in defects in abdominal wall, what about these? Diaphragmatic hernias, gastroschisis, omphalocele, and also neurological system effects. They are brain and spinal cords and etc. some things. And also in congenital abnormalities, we mostly seen cardiovascular and pulmonary abnormalities, malformation of digestive systems, and also limbs, vertebral abnormalities. All are addition to these congenital abnormalities, and also malformation of urological and reproductive systems. And also in congenital abnormalities, we can see some special diseases, which are hypertrophic pyloric stenosis, intestinal obstruction in also neonates, congenital posterolateral diaphragmatic hernias, congenital esophageal atresias, and also addition to tracheoesophageal fistulas. Malrotations, Hirschsprung disease, intestinal atresis, and some intestinal stenosis, congenital malrotations of intestinal system. And also, we mostly seen anorectal malformations in neonat periods, which is hydronephrosis, which consists uretropelvic junction obstructions. And we can see common pediatric surgery diseases. What about these? I can show you alimentary tract duplications, appendicitis, which seen mostly in pediatric surgery clinics, biliary atresias and colorectal cysts in hepatobiliary system anomalies, congenital defect of abdominal wall defects, for example, omphalocele, gastroschisis, umbilical cord hernias. And also, we can see gallbladder diseases with hepatobiliary system disease. And in pediatric surgery, when I will, next slide, I will show you, and I will introduce what about the specialties, inguinal hernias and hydrocele, interceptions, McGill diverticulum, mecanum ileus, mesenteric and omental cysts, and also jejunal atresis and stenosis. And also, we can add some tumoral activities, neuroblastoma, rhabdomyosarcomas, ulcerative colitis, Crohn's disease, necrotizing enterocolitis, which is seen in a very critical position in neonat position, and also understand the testis, torsion, and varicocele. I want to talk about inguinal hernia, which is mostly seen in pediatric surgery clinics, and what about its approach. Inguinal hernias and hydrocele incidence of inguinal hernia is 0.84 to 4.4%, but premature incidence is more than this, 16 to 25%. 30% of them were seen before age 6 months. And also males is more than female babies, 3 times more than. There's no, but there's no sex predilection among premature babies, they are same. And also we can see this inguinal hernias, especially the right side, you can see 60%, but the bilateral side is 10% because of late descent of right testicle. And also it's very important family history. In family history, we can see 11% of the patients, we look family history. What about the inguinal hernias clinical features? In the clinic, about increased intra-abdominal pressure, we can see intermittent bulge on the groin, labia, or scrotum. But in inguinal hernias, we see it usually asymptomatic. Older children complain groin and inguinal discomfort during exercise. It's very important. The history is very important about family and children history. And the emergency situation is incarceration is the entrapment of bowel at the level of internal ring. This can cause intermittent pain and irritability. Bowel obstruction, which is emergency situation, occurs and distension. It comes to clinic. Vomiting and obstipations are seen age 5 years. What will we do when we solve the baby? We place the baby supine position and undressed in the warm room, put the testis in the scrotum by the help of fingers and palpate the inguinal canal. In older children, we can perform a Walser maneuver, which is a special maneuver in pediatric surgery. And also, we look, especially spermatic cord, we can palpate, determine the thickening of the spermatic cord. We say this 6-time string. To palpate the spermatic cord, lay a finger on the pubic tubercle. Also, an examiner can feel the sensation, which we say this plastic bag sign. It's a plastic bag with few drops of water in it. It comes like this. And if the examiner couldn't detect a hernia sac on the palpation, first I say a good history from the parents or the previously examination of an other physicians is appropriate for the surgical treatment. It's very important in inguinal hernias, parents' history and the children's physical activities. They say we feel this. It's very important. And we also inguinal hernias in radiologic investigations. In the past, we look herniography was performed by injecting a water-soluble contrast material. We add this intraperitoneal cavity via infraumbilical fluoroscopic guided injection. This technique helped to detect contralateral hernia also, and also posterior recurrent hernias. But it has some complications. What about these complications? It's very important. This technique includes intestinal perforation and intramural intestinal hematoma. Maybe it can come. If it gets intestinal perforation, we immediately take this to operation room. It's emergent situation. And also intestinal hematoma and allergic reactions comes because of the contrast agents. But first, I say diagnosis of inguinal hernia is based on examination, very important, very, very important, and also history, family history. We use ultrasonography accuracy like 94%. In ultrasonography, upper limit of inguinal canal is 4 mm normally. Diameter of inguinal canal, if more than 9 mm, like 5 mm, shows a patient processus vaginalis, and more than 7.2 mm is associated with true hernia. We say this situation by ultrasonography. If we see this situation, we can say hernia. Inguinal hernia, it's very important, never resolves spontaneously. You cannot wait. And surgical closure is always indicated. It's very important. Repair should be done expeditiously because of herniation. If the repair is done in the first month after diagnosis, complication can be avoided in 90% of cases. You can see, if not, it gets babies or children come emergency operations, emergency situation. So when we expect this situation, when we saw herniation, we immediately, not emergency, but immediately, we must take a surgical closure. What about in the surgical procedure, in which anesthesia we can use in inguinal hernia babies? We can use local anesthesia, regional anesthesia, and general anesthesia. We use regional anesthesia is as safe as general anesthesia because of baby's situation. In addition to this, caudal block can be used for especially postoperative analgesia. Caudal block is only for analgesia. We can repair recommended soon after diagnosis. Some recommended time of repair before discharge from the hospital for the premature babies if they become minimum 2 kilograms. If less than 2 kilograms, we must wait because the anesthesia situation. In early surgery, in premature infants are associated with recurrence. What about technique we can use in inguinal hernias? We say high ligation of the sac. It's our operation procedure. There's a special two names. Mitchell-Bank repair is the high ligation of sac via internal ring without opening the external oblique, and also Ferguson repair, which is external obliques firstly opened and reconstruction was performed without altering the relationship between spermatic cord to the inguinal canal, but it's only the male situation. What about the females? What can we do? It's simpler than males because there's no need to identify and preserve a spermatic cord and addition to scrotum something, testis. But in females, in the sac, we can see ovary, tuber, or mesosalpinx. It's usually in the sac. If there's no content in the sac, it's possible. It should be divided and double ligated at the level of internal ring. The sac is not routinely attached to the conjoint tendon to re-establishment the normal support for the uterus. We say this special name, bastionale maneuver. And also, lastly, the inguinal hernia repair technique, we can use laparoscopic procedure. You know, in the surgery now, laparoscopy is mostly seen in operation technique, but it has also advantages and disadvantages. What about in inguinal hernia repair laparoscopic procedure advantage? It can less pain, earlier return to work or not work, we can say for children to their home and school. Also repair of bilateral hernia through the same port you can use. It's very easy. And easy repair in recurrent hernias. These are advantages, but also it has disadvantages. What about this? Increased costs, economic some problems, longer operating time, and also prolonged learning curve. However, the laparoscopic procedure, recurrence rates are much more. Usually, laparoscopic repair is not used in males, but the technique laparoscopic inversion ligation can be used in females. Thank you very much, Dr. Ulaş, and the participants.
Dear respected Professor Dr. Kadir Öymen Hançerlioğlu, thank you very much for your nice presentation and for your presence here. It was very nice to do something together. And dear respected participants, if you have questions to the presentation of Professor Dr. Kadir Öymen Hançerlioğlu, you can write them in the chat box, or you can just press on the reaction buttons and we can open your voice. For possible future collaborations, you can write down your email address if you find it suitable, dear Professor Kadir Öymen Hançerlioğlu. Maybe the researchers would like to contact you for possible research activities.
Thank you, and also the participants and researchers and presenters have babies, children. Is there any questions for not only the inguinal hernia or other something? They can ask. I can write my mail address. If they have any questions or something they want to ask, they can send a mail. No problem. Okay.
It is very nice of you. Thank you very much. Thank you very much.
Thank you very much.
And the next presentation is going to be had Assistant Professor Dr. Şebnem Alanya Tosun. She is from Giresun University Medical Faculty. Her presentation is about the vitamins, supplements, and nutrition during the pregnancy. Dear Professor Şebnem Alanya Tosun, thank you very much for accepting our offer. We are happy to listen your presentation. If you need technical assistance, I can describe the procedure. Firstly, you have to open your PPT. We are not able to hear your voice. Maybe there is some problem with your microphone. Your microphone is open, but the voice is not coming.
I think we have a problem on the next computer we are coming.
I have problem with my computer. I think I will open from here.
Okay.
Okay. Do you see the screen?
We can see you and your video, but still we are not able to see the PPT. First, you have to open your presentation. Afterwards,
Okay, I opened my presentation now, and I will do. Sorry.
Then you have to go to Zoom window. Then at the bottom there is a green button. Just click on it. Then you will see in the next window your presentation. Just select it. Hocam sunumunuzu açtıktan sonra Zoom penceresinin alt kısmında yeşil bir düğme var. O ekran paylaşımı. Ona tıklayıp
orada birden fazla katılımcı aynı anda paylaşım yapıyor. Aynı anda bir katılımcı paylaşımda bulunabilir diye şeyler çıkıyor.
Onun yan tarafına basacaksınız hocam. Oka değil de yeşilin kendisine basacaksınız. Ortadan basacaksınız.
Basalım. Tamam. Tamam.
Orada sunumunuz gözükecek hocam.
Tam ekran yapacağız önce ondan sonra. Tamam. Hocam görebiliyor musunuz?
Şimdi açıldı. İlk sayfa beyaz gözüküyor yalnız hocam. İsterseniz bir slide sonra geçtiğiniz zaman değişiyor mu bakalım. Ha, geldi hocam. Hı-hı. Süper.
Tamam, slide'da değişiyor. Aha.
Evet hocam.
Aha, bir saniye. Ben bunu minimalize edebilirsem eğer. Tamam. Okay. Sorry for late. Have a nice day. First of all, I would like to thank to the organization committee, especially to Dr. Ajaros for this nice congress. I'm Assistant Professor Dr. Shemna Tosun from Giresun University Faculty of Medicine. I'm in the Department of Obstetrics and Gynecology, and today my subject is vitamins, supplements, and nutrition in pregnancy. As we know, the intense fetal growth and development during pregnancy requires maternal physiological adaptation and a change in nutritional needs. So all the pregnant ladies should get adequate maternal intake of macronutrients and micronutrients. If there is an undernutrition or overnutrition, like obesity, there will be adverse maternal and newborn outcomes. These can be miscarriage, some congenital anomalies, hypertensive disorders of pregnancy, gestational diabetes, preterm birth, small for gestational age newborns, or there can be neurocognitive, there can be suboptimal neurocognitive development of babies. So when we say that human being is like a DNA software, we have all codes, and also pregnant lady has a code, and during the intrauterine time, code should be well written, and also codes should not be broken. These things that can break the codes can be stress, toxins, drugs, and also it can be inadequate diet or some nutrients that can modify epigenetic modifications. So the fetal environment, which is affected by maternal nutritional status, causes epigenetic modifications that impact gene expression and influence subsequent development of disease in offspring, both in childhood and adulthood. So codes that have broken down can be transmitted to new generations as hereditary diseases. So we don't want any other hereditary diseases. So the nutrition during pregnancy and the adequate supplements is very important during pregnancy. As we know, ideally, all individuals should be at a healthy body mass index and have optimal nutritional status when they conceive, when they plan to get a baby. However, in reality, also in our country, in Turkey, approximately 50% of females in high-income countries are overweight or obese, or in low-income countries, they are lean. Many females have inadequate micronutrient status, especially iron and folate is very important during pregnancy, and we know that many ladies who are planning to get a baby are lack of these micronutrients. In obstetric polyclinics, in every visit, we also check their weight gain. Appropriate gestational weight gain is very important. We do not like extremes. We do not like the upper weight gains, and we do not want the inadequate lower weight gain. We want an optimal weight gain during pregnancy. Weight gain is not only a fat. We should not think that all the weight gain is fat as a fat. When a lady, pregnant lady, gets the 40 weeks old pregnancy, she has 3.5 kilo of fetus inside her. She has 1 liter of amniotic fluid. She has half kilogram of placenta, and she has 1 kilo of uterus, and she has also 1.5 kilo of excess blood volume. Only 2.5 or 3 kilo of fat should be in her body, not more weight gain. So totally, ideally, with normal body mass index before conception, a pregnant lady should get 12.5 kilo in all pregnancy. And the recommendation differs if a pregnant lady is lean before conception or overweight before conception. If a lady is very lean before conception, we recommend her to get more weight gain, like 12.5 kilo to 18 kilo. Or if a lady comes with overweight, lady comes and wants to get a baby, we recommend her to gain less kilo, like 7 kilo, 9 kilo. If a lady is obese, BMI is over 30, we recommend her to gain only 5 to 9 kilos. And during the pregnancy, the kilo, most of the kilo is gaining during the second and third trimester. During the first trimester, the lady should only get 2 kilos. After the first trimester, I mean after the 12 weeks of the gestational age, the lady can get half kilo per week. But if she is overweight lady, she should get 0.25 kilo per week. She should get less if she is overweight. And energy intake differs during the pregnancy. For example, normally a lady without pregnancy needs 2,000 kilocalories per day. And normal BMI pregnants only needs an additional 340 to 450 kilocalories per day. Not eating for two. I mean, the old adage, eating for two, neither accurately nor appropriately depicts the increased needs of pregnant women. For underweight BMI pregnants, an additional intake, energy intake, should be a bit more, like 450 kilocalories per day. But for overweight BMI pregnants, it should be a bit less, like 260 kilocalories per day is suggested. And for obese BMI pregnants, there's no need to get additional calories for them. And there should be a general dietary principles for optimal pregnancy outcome, and the ladies should obey these rules during the pregnancy. They should eat all types of vegetables, dark green, red, orange vegetables. They should eat all fruits, especially whole fruits. They should eat grains. At least half of which should be whole grains, and they should eat dairy. Fat-free or low-fat milk, yogurt, cheese, or they can use also lactose-free versions of the milk or yogurt. And they can eat protein. They should eat protein foods like lean meats, poultry, eggs, seafood, beans, peas, nuts, seeds, and also they should get oils. But not unhealthy ones. They should get healthy oils like vegetable oils and oils in foods such as in seafood, nuts. These are good oils. They should get this. And they shouldn't get processed food. They should get unprocessed food and beverage, but in appropriate amounts. And they should limit to consume added sugars, saturated fat, and sodium. These are unwanted nutritions. and they should avoid alcohol, and they should avoid also from mercury. Mercury is in seafood, fish. I will deal in later slideshow. I will deal it in seafood. There's mercury, so we do not want deep sea fish. We just want the fish with low risk of mercury. And they should limit caffeine intake. I mean, it should be 200 milligram per day. It means that it can be 1 cup of coffee, or 2 cups of black tea, or 3 cups of green tea. And they have to take appropriate vitamin and mineral supplementation according to the guidelines. As I said before, there's macronutrients and micronutrients, and they should be all in balanced. Macronutrients are proteins, carbohydrates, and fats. I will first start to tell about them. And then micronutrients are folic acid, vitamin B12, omega-3, and other elements and antioxidants. First, very popular macronutrients is protein. As we know, the fetal placental unit utilizes approximately 1,000 grams of protein, with most of this requirement is in the last 6 months of the pregnancy. I mean, the second and the third trimester of the pregnancy, they will require more. And the recommendation is to take a minimum daily 71 gram of protein. Normally, a non-pregnant lady needs 46 grams per day, but it's, as you see, we recommend to the pregnant lady 71 grams per day protein. But as I told you before, also we increased their total calories a bit, like 350 to 450 additional calories. So the percentage of the protein remains same as 10 to 35% of total kilocalorie. What about the sources of the protein? It can be lean meats, poultry and eggs, seafood, beans, peas, nuts, seeds, and soy products can be. But we do not recommend highly processed meats to pregnants, and we do not recommend the deep freeze. We do not want it. We want fresh foods because of the infection risk in deep freeze meats. In a study of 120,000 pregnant people in Denmark and Norway, high protein intake did not appear much, did not affect so much about birth weight, and did not lower the low birth weight, and it's associated with increased risk of preterm birth. So the excess is not good also. Excess protein is not wanted, adequate protein is wanted. And another macronutrient is carbohydrates and fiber. Carbohydrate requirement increased to 175 grams per day. Normally, a non-pregnant lady needs 130 grams per day. But the percentage to the total calorie does not differ. It's 45 to 65% of total calorie. They have to take also fiber. Many pregnant ladies comes to our polyclinics with the completion of constipation. And to prevent constipation, they need to take adequate fiber, and they need to take adequate fluid intake. And also fiber is necessary to decrease the risk of preeclampsia and dyslipidemia. And they also control blood glucose. So pregnancy is a period for a lady that can be go to the gestational diabetes complications. So it's very important if they take adequate fiber. It can control both blood glucose, lipid profile, so it's very good. And the sources can be fruits, vegetables, and whole grains. Highly processed carbohydrates is not wanted again. We do not want highly processed carbohydrates because we want to manage the weight gain. We do not want extreme weight gain, and to avoid high postprandial blood glucose levels, we do not want highly processed carbohydrates. And another macronutrient is fats. Fats should be 20 to 35% of total energy. And it's important that the saturated fat should not exceed to 10% of it. Daily goals for essential fatty acids of 13 grams per day of linoleic acid and 1.4 grams per day of linolenic acid is very important. So they have to get essential acids, but they have to consume less saturated fat. Approximately 75% of pregnant people exceed the recommended limits for saturated fats. This means that they consumed lots of meat, dairy, coconut, palm. We do not want these things because they have negative metabolic consequences. And what about the healthy sources? Healthy fats are olive oil, nuts, and especially fish. We want them to consume fish, but there is a limit to consume fish. We think that one serving of fish is 150 grams. So 2 times per week means 300 grams of fish, and it is adequate consumption per week. They can take enough fats from that fish, but they will not exceed mercury. If they exceed 2 times a week, they will get lots of mercury. We do not want that. So 2 times per week for fish is good. So we have some supplements for micronutrients. These are helpers of us in our obstetric polyclinics. These are folic acid, iron, vitamin D, calcium, magnesium, omega-3, and multivitamins. First of all, folic acid, it's very popular. Obstetricians like very much. It's folate B9 vitamin, the natural form called B9 folate, and it's water soluble. And we can normally get folate from beef liver, green vegetables, avocado, egg, milk, pea, and beans. But it has also a synthetic form with commercials, and the synthetic form of folate calls folic acid. And this synthetic form is also added to pasta, flour, and rice as a government politics. So the people can get enough folic acid from also pasta, flour, and rice if it is added. So it's also in multivitamin medications and prenatal vitamins medications. But both has an active form of L5-methyl tetrahydrofolate, which is absorbed from the intestinal mucosa. Why folic acid is so important? Because there's a disease called neural tube defects. It is a group of birth defects which has an opening in the spine or cranium, remains from early in human development. During the first trimester, during the first 12 weeks of the pregnancy, neural tube normally closes. If there is not enough folic acid there, it cannot be closed properly. So neural tube defect can develop. So folic acid is when we give folic acid to the patients, we decrease the occurrence and recurrence of neurotube defects by at least 70%, and it's very important number for us. At least 70% we decreased. So what is the recommended intake? We should give a supplement in first 12 weeks of folic acid as 400 microgram per day. And we should start it from the 1 month before pregnancy, and we should continue it in the first 12 weeks of the pregnancy to reduce the risk of neurotube defect. And also it has some additional benefits like reduction in pregnancy-related hypertensive disorders, reduction in occurrence of congenital anomalies other than neurotube defects, and it's a good medication for us. And after 12 weeks, some authors are recommended to continue to folic acid supplementation to meet the growth needs of the fetus and placenta. In my practice, I also continue to give folic acid. And pregnant people at high risk, who are them? For example, individual who had a neurotube defect pregnancy before, who experienced or who had diabetes, or who has to use anticonvulsant medications because of epilepsy, should use folic acid 10 times higher. I mean, they should use folic acid 4 milligrams per day. And we know that in the population, at least 36% of all the pregnants are taking inadequate food to get folic acid. So with the all guidelines, NICE, ACOG, Canadian, American, England, with all guidelines, folic acid intake is recommended 1 month before pregnancy and 3 months after pregnancy. After conception, I mean. And the other thing is iron. Iron is necessary for fetal brain and placental development and to expand the maternal red cell mass. Iron deficiency is the very popular cause of anemia. We saw much, and it is the most prevalent single nutrient deficiency worldwide. In pregnancy, it's ranging from 7% to 30%. And when they are not anemic during the first trimester of the pregnancy, with the growing of the fetus, in the third trimester, late pregnancy times, they are getting anemic. So we have to replace it. Routine iron supplementation in pregnancy results in a reduction in the frequency of iron deficiency anemia at term. And recommendation intake is 30 milligram of elemental iron. It's World Health Organization recommendation. This 30 milligram is for the people who hasn't got anemia. But if they have anemia, it means that if their hemoglobin level is below 11 for the first and third trimester, or below 10.5 for the second trimester, we have to replace it with an additional iron supplement to 60 to 120 milligram per day. So we have to treat anemia also with a higher dosage. So iron supplements should be used also in the postpartum period till the baby got her or his 6th month. So mother should use, because a mother is also having breastfeed during this period, and also giving iron to the baby with the milk. So we have to replace after pregnancy also. we usually give oral, but we can also give intravenously because when we give oral, there can be some complaints like dyspepsia, gastric intolerance, and some people doesn't like iron, and we can give intravenously, and the intravenously form is ferric carboxymaltose, which is the most safe form against allergic reaction. And there's some dietary sources for iron. It's hem and non-hem sources. The most bioavailable form is hem iron. Hem iron is in meat, poultry, and fish. There's also non-hem iron. Non-hem iron is in dark green leaf vegetables like spinach or in legumes, bean, fruits, seeds, eggs has non-hem iron. But as you see in the table, hem iron absorption is more than non-hem iron. So fish or meat is very good for iron supplementation also. These two, folic acid and iron, are recommended routinely to the pregnant ladies by all the guidelines and World Health Organization. What about multivitamin supplementations that contains all the micronutrients? The only indications for them is low socioeconomic status, inflammatory bowel disease, morbid obesity, diabetes mellitus, multiple gestations, malnutrition, smoking, alcohol consumption. It's not recommended routinely to the well-nourished human. And there is also very popular and very good vitamin. It's vitamin D. Vitamin D is very, very important, I think, because as we know, it's it's responsible from growth and bone mineralization, but it's only the part that we see. Also, it has more valuable about the immunity, about cell proliferation. These are very good properties of vitamin D. Because of that, for example, during this COVID-19 pandemics, we use vitamin D for regulation of immune function. And also it has control, it promotes control of cell proliferation, it stimulates cell differentiation, it inducts apoptosis, it regulates muscle calcium transportation. It has many roles. And it's very important vitamin. We can supply this in several ways. The most known way is from the ultraviolet, from sunlight via skin. And it's coming with 7-dehydrocholesterol. And in our body, it metabolizes in liver and kidney, and turns to the active form of 1-alpha-25 hydroxy D3. And also it comes from the dietary sources, from the bowel with absorption. And these can be vitamin D2, argocalciferol, or vitamin D3, cholecalciferol. And the same metabolic patterns are also for them. If the lady has a poor vitamin D status, so it may have short or long-term consequences on bone, immune system, and general health. I mean, the poor vitamin D status can be with preterm birth, preeclampsia, gestational diabetes, and we do not want to face with these complications. But what about the threshold of vitamin D? What is the normal level of vitamin D? We do not know well. It's not well defined. Some guidelines says that you have to check vitamin D and it should be over 20 nanogram per milliliter. Some guidelines says that it should be over 30 nanogram per milliliter. But it's not well defined. But we know that vitamin D deficiency is estimated 40 to 98% of pregnancy worldwide. And I'm living in Giresun. It's in the northeastern part of Turkey. Here is rainy in 300 days of the year. And because of that, we do not have so much sunlight. And there's a research in Giresun, and it says that the 95% of the women, pregnant women, are with vitamin D deficiency, and it's very much. And FIGO recommend routine intake of vitamin D during pregnancy. They recommend 600 international units per day. And FIGO says that if there's a deficiency, it should be 1,000 to 2,000 international units per day. And our ministry get a guideline about vitamin D, and according to that guideline in Turkey, we obey to this guideline, and we routinely start 600 international units per day to pregnant ladies. But if there's a deficiency, we start 1,200 international units per day vitamin D to the ladies. If we do not, if we cannot check that there's deficiency or not, we routinely start with the dosage of 1,200 international units per day. But we are starting to give this supplement from the 12th week of gestation. And after that, if the patient complains about noise, nausea, vomiting, or constipation, polyuria, polydipsy, so we should check calcium levels. And if there's hypercalcemia, we should check also the vitamin D, and we should send the patient to the endocrinology. If there's no hypercalcemia, we have to give the vitamin D till to the postpartum 6th month. And we have to be careful that many commercial non-prescription products labeled vitamin D inside them, but some of them contain D2 rather than D3. D3 is more readily converted to the active forms and more effective. So D3 is better, and we have to check the boxes of the commercials if it's D2 or D3. And also dermal synthesis is very important, and we should recommend to our pregnant patients say that every day, if it's sunny, hold your palms to the sunlight at least for 15 minutes per day. And also another thing is vitamin D is in the fatty fish livers. Another thing is calcium. Calcium is main source for bone mineralization, nerve impulse transportation, muscle contraction, and cell signaling, and maintenance of enzyme and hormones. So fetal skeletal development requires approximately 30 grams of calcium across pregnancy, primarily in the last trimester. There may be a benefit for preeclampsia prevention in high-risk populations, but in our guidelines, calcium is not recommended routinely to every pregnant patient. It's just recommended to the patients who has a risk for preeclampsia. For example, they can experience pregnancy before with hypertension, or they have a family family history, or they can be obese. So we are going to give calcium for them to prevent preeclampsia. Otherwise, we do not give calcium routinely. What about the recommended dosage of calcium to prevent preeclampsia? It's 1,000 to 1,300 milligram per day in pregnant and lactating individuals. And this is the WHO World Health Organization guideline 2018. And for that, 1 gram of elemental calcium equals to 2.5 gram of calcium carbonate. And it says that obesity, previous preeclampsia, diabetes, chronic hypertension, renal disease, autoimmune disease, neuropathy, advanced maternal age, adolescent pregnancy are risk factors, and you can give to them to prevent preeclampsia. Otherwise, to a normal pregnant woman, it's not recommended routinely. And magnesium is another nutrient. It regulates thermoregulation. It's important in DNA and protein synthesis and nerve and muscular stimulation. And it's important in vasomotor tonus adjustment. And it has antagonistic effect with calcium. And it's a vasodilatator. And as you see in this table 1, normal female needs 310 to 360 milligram of magnesium, and our pregnant ladies also needs 350 to 40 milligrams of magnesium. It's almost the same, a bit more. And this is a Cochrane review. In this Cochrane review, it says that there's not enough evidence to supplement dietary magnesium with as a medication routinely during the pregnancy. So during the pregnancy, magnesium is not a routine supplement for pregnant ladies. Omega-3 is very popular because it's long-chain polyunsaturated fatty acids, and they are essentials. They cannot be produced in our bodies, also in pregnant ladies' bodies. And as you see in the table, there is 2 types of omega here. One, the green one is omega-3, alpha-linoleic acid, and the red one is omega-6, linoleic acid. When it's metabolized, omega-6 goes to arachidonic acid. Arachidonic acid is related with inflammation and thrombosis. And we do not want inflammation and thrombosis in pregnant ladies, of course. But omega-3 goes to the EPA and DHA at the end. And docosahexaenoic acid and eicosapentaenoic acid are 2 omega-3 long-chain polyunsaturated fatty acids derived primarily from fish and marine-based sources. And especially DHA is for normal development of the fetal brain and retina. It's very important for visual things and for cognitive things. It's very important for our offsprings. So we prefer, of course, DHA for pregnant ladies, and we do not prefer omega-6 because of its inflammation and thrombosis. And for to get omega-3, we have fish oil. We have to say that you need to take fish oil. It can be canned tuna, salmon, sardines, or mackerel. So it will have anti-inflammation effects. It will have good effects to the cardiovascular functions, and especially to the fetus. DHA will get good effects about cognitive functions and visual functions. For fetus, it has benefits to prevent preterm labor. to have visual and cognitive development and to prevent allergy. And omega-3 has also benefits to mother to prevent preeclampsia, hypertension, and to prevent postnatal depression. So we recommend DHA to all pregnant women. But if pregnant consume 2 servings of seafood per week, they do not need to take another supplement outside. 2 servings of seafood is enough to get DHA. But if they could not eat fish, so we have to recommend DHA prep medication. It should contain at least 200 milligram per day DHA. And we should recommend at this time. And the other sources for DHA are fish and adequate amounts of fish. And another sources are available: yogurt, milk, eggs. Also, they can contain DHA. You should check the boxes of the yogurt, milk, and eggs. Some of them is fortified with DHA, and DHA supplements are available either from fish oil or synthesized by algae. Also, algae preparates are okay for us, and some prenatal vitamins also contains DHA and EPA. And these are safe fish. Safe fish mean low with mercury. So these are salmon, shrimp, canton, catfish, sardine. And there are some fish with higher mercury. They are living mostly in the deep side of the sea. These are shark, mackerel, swordfish, deep water fish. We do not want them to prefer these fish. And there is also long-term effects of omega-3 deficiency. If there is omega-3 deficiency, asthma, depression, diabetes mellitus, cardiovascular disease, dementia, macular degeneration, and cancer grows. And also for omega-3, there should be it should be from the fish body, not from the fish liver, because fish liver contains vitamin A, and we do not want to give more vitamin A because it can be teratogenic after a dosage. Iodine is an essential mineral necessary for production of thyroid hormones, and daily iodine intake of 220 microgram during pregnancy and 290 microgram during lactation is recommended. But we can take this from the iodized salt. So if the lady has a good nourishing lady, so we do not need to give extra additional iodine. But if she has malnutrition or she's living in a place that she cannot reach to the iodized salt, so we can give her additional iodine. And there is some supplements and dietary intake that can be harmful. The most popular one is preformed vitamin A in supplements. Consumption of vitamin A greater than 1,500 microgram retinol appears to be teratogenic. So we have to check the boxes of the medications, and we have to see that it should be beta-carotene. If it is beta-carotene, it's not related with congenital anomalies, but if it is retinol, it's related with congenital anomalies. And vitamin A is in liver, so limiting the intake of liver during pregnancy also recommended, especially in the first trimester of the pregnancy. We limit to take to consume the liver for pregnant ladies. And also iodine can be teratogenic, and excessive intake of iodine can cause fetal goiter. This excessive intake of iodine means the greater than 10 times of the recommended dietary allowance. So if it is 10 times higher, it will cause fetal goiter. And also processed trans fatty acids should be minimized or avoided. We do not want processed trans fatty acids because they have adverse effects on fetal growth and development, and they can replace maternal intake of cis essential fatty acids. We do not want this. And there's some supplements with no or uncertain pregnancy benefits. Vitamin E, vitamin C, vitamin B6, there's no benefit, no harmful. It can be inside the medication boxes, supplements, they can take with multivitamins. They have no benefits, they have no harmful. And probiotics, this is a popular item, as Professor Dr. Recep told us. Some evidence suggests that probiotics used during pregnancy may have beneficial maternal effects, such as reduced risk of inflammatory events and preeclampsia and improved maternal glucose metabolism. However, in a Cochrane review, it's a very recent review, probiotic treatment for patients with gestational diabetes concluded available evidence was too limited. So it's not in guidelines now, it's not recommended routinely, but there should be more research about the probiotics. And lastly, fluid requirements. As all the individuals, pregnant ladies also need water or other fluid requirements. It should be approximately 10 cups. And also additional water is consumed in foods and other beverages. So totally adequate intake of 3 liters per day is needed. And fluorids, for fluorids, if there is a water without fluoride, there's no need to replace more fluoride to the pregnant lady. This is my last slide. These are the take-home methods. Total calories vary according to the BMI, but it should be in balance with carbohydrates, fat, and protein. And at least 3 liters per day water intake should be recommended to the pregnant ladies. And they should consume 2 or 3 servings per week, fish or seafood, but they should choose safe fish. They should be careful to mercury, and they should get 200 milligrams per day DHA for neurologic cognitive development. But if they eat fish 2 times per week, omega-3 supplement is not necessary. But if they do not eat fish, so omega-3 supplement is necessary. And for all pregnant ladies, folic acid 1 month before the conception and 3 months after the conception is necessary. And iron prophylaxis for 30 milligram per day is necessary for all women. If they have anemia, additional 60 milligram per day is recommended. And for vitamin D, if there's a high-risk pregnant woman, we should give 1,000 to 2,000 international unit vitamin D. And for ladies with high-risk patients, pregnant, we should give also calcium. This is elementary calcium, 1 milligram per day for high-risk pregnants. Thank you. This is my university, and this is Giresun. Thank you very much.
Dear Professor Dr. Şebnem Alanya Tosun, thank you very much for your valuable presentation. If you allow us, maybe we can share your presentation also in the YouTube channel, because there were very important information for during the pregnancy. It can be very useful for the pregnant women all over the world. And also, if you can share your email address, maybe the academician who wants to work with you or asking questions to you. Thank you very much again for your valuable contributions. It was very nice presentation.
Thank you very much. Thank you.
And the next presentation is going to be held from Romania, from Associated Professor Dr. Laura Shumulek. She is from Faculty of Agriculture and Banat University of Agricultural Sciences, Veterinary Medicine. And 2 months ago, we have been with Damla in Romania in this university. It was very good experience in our life, and they have accepted our offer to make a presentation in our Congress. Thank you very much for Dr. Raul and for Dr. Laura Shumilek. We are happy to listen your presentation about the sustainable development and environment, dear Dr. Shumilek. It is your turn.
Yes, hello everyone. Thank you for just a moment. Thank you for inviting me to this conference. I hope you all seen my presentation. You can see it.
We can see your presentation, but it is not full screen. Can you make it full screen?
Okay, just a moment.
Yeah.
Now, how it is?
Now, there are some things you should do. Just go bottom of your presentation. You can click on the icon to make the full screen. It's gone again.
Here now?
Yes, we have now.
Yes, you can see it.
Can you try to change the slides? Sometimes it is also
yes.
not possible.
Oh, I cannot. Just a moment.
Yes.
Yes, you can.
Now everything is fine.
Okay. So my presentation is regarding environmental sciences. It's one of the topic of your conference. I will present now the impact of climate changes in Western Romania. Let me show us a brief presentation of mine. I'm vice dean at the Faculty of Agriculture. I'm also associate professor and Erasmus Department coordinator and also coordinator for international activities, having in the present 25 Asian partner universities. Regarding my background is founded on agriculture, global environmental and climate change. I have bachelor in both agriculture and engineering of agriculture environmental. Also, my PhD is focused on the climate changes. My work experience, including water resources, hydrology, climate changes, and I publish more than 100 scientific papers regarding this paper. These topics. I'm also a member of different associations. One of very important is the European Network of Environmental Professionals, and also I'm a secretary of Romania Environment Association and member of Romanian Water Association. Back up to my topics. What is climate change? Are you having climate change? How do you know that there are climate change? Scientific evidence for warming of the climate system is unequivocal. So this graph, based on the comparison of atmospheric samples containing ice cores, provides evidence that atmospheric dioxide of carbon has increased the industrial revolution. As you see, here is the level after the industrial revolution and the current level. Most of these changes, climate changes, are attributed to very small variation in Earth's orbit that change the amount of solar energy. But there is no question that increased level of greenhouse gases must cause the Earth to warm in response. So we can see here the global temperature rise Earth's global surface temperature in 2020 was the second warmest since modern record keeping beginning 1880, and now we can see this rising of the temperature. This graph illustrates the change in global surface temperature relative to 1951-1980 average temperatures. The year 2020 tied with 2016 for the warmest year on record since record keeping began in 1880. Observing warming over the past 50 years show rapid warming in the Arctic, while the surrounding ocean around Antarctica was warmed little, if at all. Regarding melting glaciers, the Greenland and Arctic ice sheets have decreased in mass. Data from NASA Gravity Recovery and Climate Experiments show Greenland lost an average of 280 gigatons per year. Since Antarctica mass variation lost 147 gigatons per year. What about the mounting glaciers? Glaciers are retreated almost everywhere around the world, including the Alps, Himalaya, Andes Rocks, Alaska, and Africa. Because snow is so reflected, it plays an important role in regulating climate. It reflects incoming sunlight back into space, cooling the planet. Shows also support lives, so it's very important to have snow currents. About sea level is increasing primarily by two factors related to global warming: the added water for melting ice sheets and glaciers, and the expansion of seawater as it warms. This graphic traces the change in sea level since 1993 as observed by satellites. Extreme events such as hurricanes, tornadoes, fires, heat waves, drought, and affected all over the planet. Since the beginning of the Industrial Revolution, the acidity of surface ocean water has increased by about 30%. This increase is the result of human emitting more carbon dioxide into atmosphere and hence more being absorbed into the ocean. The amount of carbon dioxide absorbed by the upper layer of the ocean is increasing by about 2 billion tons per year. Greenhouse effect is the process by which radiation from a planet atmosphere warms the planet's surface to a global to a temperature which is above the normal. And we can see in this image that the human enhances greenhouse effect. Now we are back in my part of Romania, in my part of study, in Western Romania. The Banat Plain is located in Western Romania, and it is the largest landform in Banat, occupying about half of this area. The Banat Plain is one of the eastern part of the large sedimentation basin called the Pannonian Depression from Pannonian Sea. The subsidence movements are translated in the landscape by the unraveling of the rivers, the degradation of the water courses, the existence of the large swampy areas of lakes and alluvial plains. The deplorable state of extensive swamp and permanent flooding of the uncontrolled river in the Banat Plain in the early 18th century led the provincial administration to take measurements to drain this swamp and regularize the river, especially the Bega channel. The special impact of the hydromelioration works in Banat Plain start in 18th century determined the lowering of the groundwater level and the entire area of the plain. The slow descent of the ground level under the influence of the first amelioration works, it is maintained for a long period of time, over 200 years, at critical depth. But since 1950, land improvement works are resumed in Banat based on large national project and with increased intensity. Groundwater levels are rapidly failed below critical levels, and the remaining swamps of the past are drained rapidly. Currently, the Banat plain is completely drained. We study also the evolution of thermal regime to see if we have climate change or no. And the study was made on the data recorded at the Timișoara weather station for a period of 123 years. Between, as you see in this graphic, 1897 until 2020, starting the due that the first war records are missing between 1917 until 1920. Figure shows the evolution of annual temperature. It's a slightly upward trend, especially in the last decade. We try to separate it on the vegetation period and the cold periods. And we can see if in the cold period, it's not, we cannot see a trend, but in the vegetation period, we can see a temperature distribution has although been carried out, and it's in the trend, a slight trend of increasing temperature. Due to the fact the oscillation between the annual values were quite large and the tendency to change is not very representative, we resorted to representing the evolution of temperature by average of 10 and 50 years. From the evolution of over periods of 10 years, there is a sinusoidal oscillation that follows the same trend in the 3 period considered. The last decade records the highest temperature in the period study. So October, September, March, October, and April, September. The average values for 50 years are not so representative as those for decade, and show very small variation, but with a clear upward trend, especially in the last 50 years. In the context of the manifestation of global climate change that also affects Romania's climate, it is necessary to study the quantitative variation of the rainfall regime in the Banat Plain, being a priority indicator of the climate. The following analysis was the evolution of rainfall over a period of 147 years. Figure shows the evolution of annual rainfall, and the slight downward trend is observed, as we can see in this image. Analyzing the distribution of precipitation during the vegetation period, it was noticed that in the warm period of the year, the decreasing tendency is higher than in the cold period. The analysis of precipitation during the warm period is very important because it is the period when the water demands of plant is highest, and is the period when the condition of aridization more and more accentuated in the global context. This figure also shows the average precipitation values over a period of 10 years. There is a sinusoidal oscillation of precipitation over time, but with a visible downward trend. The average annual for a value of 50 years, although oscillation shows the downward trends for the agriculture years, as well as the cold period of the year. We also calculated the evapotranspiration, but we want to see how does the climate change influence the management in the Western period. So the potential evapotranspiration was calculated with the Thorne weight method and can be calculated with the data provided by the Timisoara weather station. It is noted that in the last decade, we have the highest values of evapotranspiration, which denotes high water demand of plants, which is we cannot be covered by rainfall, which are declining. Regarding the potential evapotranspiration during the vegetation period, there is also a ground trend more prominent than in the case of annual transpiration. The high-low diagram indicates in the form of areas the differences between evapotranspiration and precipitation accumulated in the warm period of the year. It is visible that the values of evapotranspiration are higher than precipitation. Moreover, in 98% of the period under analysis, the precipitation evapotranspiration ratio was subunit. For the entire period, 1898, 2019, the average values of cumulative evapotranspiration was 714 millimeters. At the same time, the average values of accumulated rainfall through the year were lower, only 610 millimeters. The tendency of the report between precipitation and temperature is a slightly decreasing one, but still superior to the limit of 0.5, under which an area is described to be a semi-arid one or even arid. However, the high number of the years in which evapotranspiration exceeds quantitative rainfall, which is an unfavorable ratio, are the years in which considerable damage to agriculture occur. It is understandably the need for concrete measurements by regulating the water balance. Here we have, because of the climate change, some runoff of our important rivers. We have also floods, the big floods on Banat River, which was in 2005. And you can see here the highest level of the river. The zone occupied by the floods was very big floods. Here we can see the pump station, some images with the floods in Bennett's area. So the question at the final of this presentation is not if there is a climate change. The question is, when do we start to live sustainably? Thank you for listening to me.
Dear Professor Dr. Laura Shumulek, thank you very much for your valuable presentation.
Thank you.
If you can share your email address.
Of course.
If you can share your email address, the researchers can contact you.
Thank you.
We thank you for your presence and contribution. And the last invited speech of today is going to be held by by Dr. Raul Pascalau. Normally his presentation was yesterday, but due to some health issues, we will listen him today. And he is also at the same time the Erasmus coordinator of Romania University. And Thank you very much, Dr. Raul, doing together.
Hello, everybody. Thank you, Dr. Ulash, for this university, and congratulations for all the organizations. We are very happy to contribute as members of the Banat University of Agricultural Sciences and Veterinary Medicine, King Michael I of Romania. And it is also an honor for us to be together here with you. Yesterday evening, I watched a few materials that my colleagues have sent, because Corina was present yesterday for a while. So my presentation also is related to the environmental area. Do you see the slides?
Yes, we can see.
Okay. Let me see, how can I just make it
full screen? It is at the bottom of your presentation. There is an icon. Just click 2 times on it, then it is going to be full screen.
Not this one. At the bottom of the presentation.
At the right side.
No, it's not this
one.
Not
this one. This one.
Okay.
Yep.
So once again, bilingual education in environmental sciences. Of course, we all know that environmental sciences as a large area includes also our university, because we are a life science university, and in all our partnership around the world, we try to match and to partnership with colleagues and partner universities within this area, either only environmental sciences or also comprehensive universities who include also environmental sciences. Life Science University, of course, has also the veterinarian sector, the animal science, the bioengineering of animal resources. First of all, I would like to say that I am an assistant professor. I'm teaching, I teach foreign languages, and I also manage the Erasmus program, being the head of the Erasmus office, presently having 100 non-European universities and 200 from 57 countries all over the world, and 283 European partners within the frame of the Erasmus Plus, where Turkey also is part of the European partners, Turkey and Serbia. So why bilingual education? Because I have chosen the sector keeping on the trend of my colleague, Associate Professor Laura Șmuleac, within the climate changes, because climate changes is part of the environmental sciences, and we are very concerned about what happens at the global level, trying to bring a small contribution to the education within this area. So we believe that teachers face a difficult but imperative task. All of them have to figure out how to talk about the complex forces behind climate change, but also the effects on cultural safety, prosperity, and progression. They have to teach youngsters, in this case our students, their possibility and the community's ones, how to act and become an important pillar in reducing the magnitude and the intensity of climate change. Because we are not only educational providers, but we are also part of the active life within the economic and social background. Environmental sciences represent an important area in one's life and activity. As I said, we are a life science university and one of the 4 existing in Romania, but we all have a mutual goal. And of course, our mutual goal is also yours, education. Environmental sciences represents an area important worldwide because it is connected to life itself. Bilingual education is the key, not only to recognize performance, but also to information acknowledged from all over the world. Being able to possess and to speak several languages and having the required information or data acquired in a professional way makes all the students future professionals, having a better chance after graduation to be easily inserted on the international labor market. Educators must act in advancing the school's overall response to climate change, which involves focusing on climate and sustainability in the curriculum. In this regard, it is essential that the program contains strong and clear guidelines for the future. Teachers need to help learners to adapt to climate change and the climate conditions of the future. Education has a crucial role to play in this difficult task. Its position and role is crucial in this area. In the first place, it is very important to act for the development of an attitude supporting the fight against global warming projections in the near future. Secondly, skills development. It is crucial as well as capabilities and attitudes to adapt to the obvious and imminent climate impacts. And thirdly, it carries out a permanent position in stimulating and strengthening understanding and attention to the evident facts of the global warming. The education for the global warming targets a clear, strong direction, namely a determined commitment of the academic community in HAES, higher education institutions. It is also highlighted by the diversity of the variety of educational methods and approaches. Teachers who experience the Teachers' Climate Change Education course will be involved in a wide variety of ways that will use both intelligence and training skills. Their mission is a very complete one, and the result must be seen in the consequence of their students' actions and attitude in the near future. Because as we know, it's not enough just to teach and then to check the impact. We also have to follow the final aim. UNESCO aimed in the last 10 years to target the development of educators on all levels and cycles of study, of course, prioritizing climate changes and global warming, acknowledging that education and training within this area of environmental sciences must be redefined and reoriented so that the future must be a sustainable one. If we are thinking at a program or a schedule of a course in bilingual education for environmental sciences, we should include 4 distinct characteristics. Why? Because it gives the possibility to the educators to have a better idea and understanding on the causes and the impact of global warming, embracing a holistic approach. If the teacher cannot understand very well this idea, how can he teach something that he just masters in theory, but does not understand the holistic approach? A second one, educators and teachers face several pedagogical challenges, using different techniques that they are experimenting, being able to develop more the entire educational process that might be adapted and used in all professional backgrounds. This might also include and content their engagement and that of their students in the whole university and university and community approaches. Because as I previously said, we are not just a university for the students, we are also a university for the society, for the community. We try to build partnership with the local community and build a strong defense against climate changes. 3, educators will develop and ensure the student learning within the community through the traineeship that companies offer in the area, through their support, to their scholarships, because we have company providing scholarship for the students in the area of the environmental sciences, or a traineeship period in the summer, compulsory in the cycle of bachelor, for example. And 4th, teachers need to develop transformative and forward-looking capacities to serve as facilitators in learning about mitigation, global warming adaptation, and disaster risk reduction. Of course, it is very important to include education for global warming prevention and sustainable development growing, especially within teenager and youth generation. That is why UNESCO and EU, the European Union, the European Commission, support all programs related to environmental protection, admitting the most important position and role that education and awareness raising must play in reducing the threat that climate changes poses to a sustainable future. If you take a look on the website of the European Commission and you just Google green future, you will see a lot of programs. All these programs promoting the green concept are meant to educate, are meant to support financially, to support alliances at the European level or international alliances to apply for projects within this area. So mainly the priorities of such programs supported by the EU are reinforce the ability to support the education at all levels, especially among students, the future professional within this area, in order to create a better awareness of global warming, to support and strengthen new pedagogical approaches so that teaching global warming becomes a must. It is not enough that we have, and we know that we must teach. We also need to know how to update to current trends and how to teach the current trends into the classroom, not just stick at what we learned during our pedagogical formation and just transfer the information. We have to update it to the current trends. Increase the acknowledgement of climate change and strengthen non-formal education programs using even the social media networks. Why? Because teenagers spend a lot of time on social media networks. So if they keep on seeing everywhere things about the climate change, things about green, about measures meant to decrease the impact of climate change, they will start also within these social media networks engaging maybe in discussions or taking the attitude. Education for sustainable development requires imagining change while looking at the past, present and future in the same time. The vision is that of the trans-curricular and interdisciplinary treatment of percepts and principles of sustainability. Because if you understand what sustainability means and we take it from the past through the present towards the future, I think we'll come to a happy end. Why does learning about climate change require a local dimension or a worldwide dimension? Real and practical experiences allows for dynamic learning. This is the reason. This is a reason enough to include the local perspective in learning about global warming prevention. Of course, there is a more important reason, which is our lives themselves. So why it is so important to have this bilingual education? One may say it is enough we provide it in one's native language, but the advantages of a bilingual education within the area of environmental sciences, namely climate change, it has some advantages. The student is familiarized with specific vocabulary, both in the native tongue, but also in one of the relevant international languages, English, French, Italian, Spanish, and so on and so forth. The student is able to get information or data directly from the EU portals related to climate or environmental sciences, but also to read all the published scientific articles, which are, of course, in English mainly, usually. And also may have a better idea of an overview on the already studied area in his native language. The student also will have the advantage comparing with the others who are getting the info only in their native language when applying for a job. Every employer will look for a better employee, being able to work, communicate, and act with professionalism, and also understand EU or global policies in the field, which are mainly in a foreign language. So they will have clearly a definite advantage on the other competitors. And here, because I'm also managing the Erasmus program, I give it as an example because through Erasmus Plus, the student may develop several competencies and skills forced by the context, being in another country, of course, to learn bilingual, to study bilingual, and also to travel and meet new professionals and colleagues from similar area, different cultures and religion. The Erasmus Plus program is the best opportunity for a bilingual education, regardless the field. They may be students in veterinary, bioengineering of animal resources, agriculture, food engineering, mathematics, arts, it doesn't matter the area. Once they undertake an Erasmus mobility, they are forced to, of course, between the brackets, because they are not forced, they reach another country, they cannot study in their language. So they have to study in a foreign language. This bilingual education, because they are coming, for example, from Turkey, they speak Turkish, they are coming in our university, they don't speak Romanian, they will learn and they will be taught in English. So this bilingual education helps them to compare what they have learned, to acquire new information, and then to become better in their field of expertise. Which are the objectives of the bilingual education or bilingual course, for example, in environmental sciences? To learn and appreciate nature, the magnitude and importance as part of both self, but also personal development, and of a context learning interdisciplinary. To develop the confidence, some teachers, of course, and their capacity at facilitate one learning oriented towards the future. The objectives are not only for the students, the objectives are also for the teachers. It allows teachers to teach the change climatic in frames, environmental, economic, social, and cultural. Allow the teachers to teach dimension keys of the change climatic, the risks of catastrophe, justice climatic, and also it gives the teachers the capabilities to animate discussion and reflection on each dimension in class, but also in practical commitment outside the class. It showed the teachers how to elaborate one dimension of partnership, local and communal, important in this area. But it also proves the teachers the means to integrate the voice and the prospects for the whole world, especially of the youth in the speech of the change, on the change climatic in class, and also regarding the question of the realization of justice climate. It also developed the confidence and the capacity of some teachers to work with the pessimism, the cynicism, when feeling powerless in the face of the change climatic, and to encourage skills, empowerment, and resilience. And it reinforced the skills and the confidence in some teachers for facilitated, interactive, and participatory learning. A very keen organization of debating and involving the students in mutual solutions might be the most difficult within all the competence. Why? Because the educator, which is also a trainer, so we call it the educated trainer, must set an example and show what the practice of good restitution consists of, drawing the attention of the participants to the means and the reasons for carrying out restitution according to particular modalities. A practitioner capable of conducting a reflection, a good organization of participatory learning, asks the teacher to think about how he animates it, to identify what has worked well and the improvements that could be made in the way he delivers its course in the future, to make it more attractive. It is an excellent practice for the teacher trainer to be seen as personifying a practitioner capable of leading a reflection, showing the participants what this entails in practice. In order to become aware and conscious and determined while supporting this action, WAS must be very organized and structured in the entire process, following a conductive atmosphere, obtaining in the end an instructive feedback from all the participants. The scope and the complexity of climate change pose enormous challenges for educators. His understanding requires them to cross the traditional boundaries of school subjects and address topics such as geoscience, the media, and to know the process of change and the understanding of the dynamics of the system. The way in which society is currently responding to climate change provides an ideal context for learning from it. Global warming, of course, we all know, represents one of the biggest challenges humanity is facing in the last decades, and why might say even centuries. And it could be prevented or diminished in its effects, firstly by becoming and making students and pupils aware of its challenge, of its importance, causes, but also solutions. So it is highly recommended that all educators, teachers, trainers, how you want to call us, having classes within the area to try to teach the importance of nature's protection and their own protection, facing this global warming challenge, and at the same time to give them clear example and advices, even solutions, so that together we might create a potential community aware of the most dangerous effects of the global warming. The purpose of the global warming prevention is huge. To understand global warming and its results, we will need to understand the many related systems, including physical systems, glaciers, rivers, sea level, as you have already seen in the presentation of my colleague. Biological system, terrestrial, marine, and human system, agriculture, energy, health, economy. Changing worldviews and addressing climate change requires challenging many of society's norms, including our definition of progress based on systematic perspective, takes into account local and global factors, and cultivates respectful ways of addressing controversial opinions, approaches that are all transferable to support student development in other fields. A comparison between conventional and transformative learning, conventional teaching, relying on the transfer of the information and the discovery of the right answer, does not suit well the complexity of teaching climate change. All the procedures and the current strategy often involve understanding the experiences that the students have had. Educators and trainers, facilitators, who are themselves grappling with this issue of climate change, act as facilitators and guides for students. School learning is connected to the real world, allowing learners to cultivate their creativity and innovation when confronted with the complexity of life. Learners, especially students enrolled in specific study programs, will develop a proper attitude and skills needed to address problems for which there is yet no solution. The same skills they will need to be successful as individuals, citizens, and entrepreneurs. Mastering the information and having the knowledge in one or more than one languages and being able to use it at a professional level will also contribute at the internationalization of the HAES, of the higher education institutions, having competent ambassadors in the area of environmental sciences. Thank you for your attention.
Dear Dr. Pascalau, thank you very much for your nice presentation. If it's possible, can you share your email address in the chat box?
Yes.
Maybe
Yes,
the
right
people
now.
interested both in your research area, also for the visiting the Romania University in the with the Erasmus activities.
In fact, Dr. Ulas, because right now I still have the floor, I take advantage to mention about the two conferences in the last week of May. Erasmus Plus Internationalization Ambassadors Together for a Green Future, and the Multidisciplinary Conference in Life Science and Sustainable Development of our university. So two conferences in Monday, Tuesday, Wednesday, Erasmus, and after that multidisciplinary.
Perfect.
So you are welcome to attend it. Thank you.
It will be our pleasure. Thank you very much again.
Thank you.
Dear respected participants, we are closing now the invited speaker section. Without making a break, we will continue directly with the second section of today. And this section is going to be moderated by Assistant Professor Dr. Şebnem Alanya Tosun. And thank you very much again for your support for the moderation of the section, dear Dr. Alanya Tosun. And it is your turn, and I am opening your voice. And
thank you very much, Dr. Ajaros. I appreciate. This is the section for oral presentations. I will open it.